Good morning, everyone, and welcome to the second edition of the VHIO-BBVA Foundation International Symposium in Cell-Based Therapies in Oncology. My name is Elena Gross, and I lead the Tumor Immunology and Immunotherapy Group at Barcelona Institute of Oncology. And I also co-direct the Advanced Cell Therapies program, particularly leading the translational research and development. And today I'll be speaking on behalf of the Vall-d'Hebron Institute of Oncology. I would like to begin by thanking VHIO for giving us the opportunity of organizing this event, and the BBVA Foundation, who very generously sponsors not only the symposium, but also supports VHIO's comprehensive program of cancer immunotherapy and immunology, also known as CIMI, including our cell therapy program, which has been slowly but steadily growing over the last 9 years. A special thank you to our organizing committee. Natalia, this goes for you, who have worked tirelessly over the last months to make this event possible. With over 400 attendees registered, it is a full house today. There's many attendees that are actually not physically present, but participating online. It's truly a pleasure to see this room full of scientists, clinicians, innovators of all sorts. I'm really, really excited to see a lot of friends and former colleagues, a lot of familiar faces. We couldn't have chosen a more fitting venue. The Aula Magna of La Casa da Convalescencia is a masterpiece of Catalan modernism, designed by Lluís Domènech i Muntaner. A place that once brought care and healing to patients is now hosting discussions on the next generation of cell therapies that promise to do the same. Our logo, which you can see on the screen, also carries a special symbolism. It's inspired by Barcelona's most iconic panot, the tile that lines many of our streets since 1900s, and that many of you have stepped on your way here today. In our version of the panot, it now symbolizes the power of cell therapy, a cancer cell surrounded by therapeutic T cells from all sides. Over the next 2 days, we'll hear from leading experts who are pushing the boundaries of the field. We'll discuss some of the biggest challenges we are facing, how to make cell therapies scalable and accessible, how to overcome the barriers that still limit their success in solid cancers, and how to design smarter, safer, and more durable treatments. We'll also explore how to bring these breakthroughs to patients faster through better manufacturing, innovative clinical designs, and collaborative efforts across disciplines and borders. But beyond the data and the presentations, this symposium is about connection. The breakthroughs in science frequently come from collaboration, from scientists, clinicians, engineers, and patients coming together with a shared vision. So I encourage you to take these next 2 days not only to listen, but especially to engage, to ask questions, exchange ideas, and build partnerships. This symposium is not only about celebrating the progress in cell therapies, but also about facing the challenges and forging the next steps together. So I thank you all for being here, for sharing your ideas, your data, and your passion, and I hope you will enjoy. A few housekeeping and practical notes before we move on to the first session. We'd like to ask you to please help us respect this beautiful historic venue. The Wi-Fi details you will find around in the room, and also the QR codes that you will find on the screen for the abstract book and the program. If you need any assistance, please approach anyone that has the organizer badge. We'd like to ask the speakers to please stick to your assigned presentation times. We're already starting with a bit of a delay, and that's okay. And just before lunch, we're planning to take a group picture, so we hope you can all make it. Thank you. We can get started.
Good morning, everyone. Thank you very much, Elena. Along with Dr. Martí Ferreira Sal, I will be chairing the first session on advance in gene and cell therapies. It's a pleasure to introduce our keynote lecturer, Dr. Sal Gill. He's got an MD and a PhD in immunology from the University of Melbourne in Australia. And then he moved to Stanford for his postdoctoral fellowship in cellular therapy. He joined the University of Pennsylvania in 2011, where he's now associate professor of medicine. She's the scientific director of the Center for Cell Therapy and Transplantation, co-director of the Parker Institute for Cancer Immunotherapy, and his clinical practice is in leukemia and bone marrow transplantation. He will be discussing about a dream towards in vivo CAR-T therapy. So thank you very much, sir.
Thank you very much for the invitation. Hopefully everyone can hear me okay. So I'm going to talk to you about in vivo generated CAR-T cells, which I am very excited about. This is a topic that I think is very timely, novel, and I think about to explode in the field. So I look forward to your questions and comments and your suggestions at the end. As many of you know, this is the way we currently make CAR-T cells. We'll call that the current standard, ex vivo manufacturing, where patients undergo apheresis of peripheral blood mononuclear cells. And then on the bottom row, what you see there is that those cells undergo activation typically with beads against CD3 and CD28, which activates those cells and makes them receptive to transduction with typically lentiviral vectors, typically VSVG pseudotype lentiviral vectors. Those are expanded for about 9 to 10 days, formulated for and prepared for release, and viably cryopreserved, and then shipped, if made centrally, shipped to the clinical site and then infused into the patient after lymphodepleting chemotherapy. And this is what we have been working on for a few years now, initially in my academic lab and then in a company that I founded, which I will mention a little bit later on. There is a possibility of modifying T cells directly in vivo using lipid nanoparticle carriers carrying mRNA. That's not what I'm talking about today. I'm actually talking about use of targeted viral vectors, and I'll explain to you, hopefully with some clarity, how we do that. And the idea was, and in fact appears to be a reality in the clinic, that we can infuse these viral-like particles into patients and then they will specifically transduce T cells directly in the body and make them into effective CAR-T cells to treat disease. What is the problem to be solved? Well, in the United States, the price of FDA-approved CAR-T cell products is considerable, let's say $450,000 to $550,000 a shot, and that appears to be increasing. I'm sure the price is not cheap in Europe either and is not available in many jurisdictions, both due to the price, but also due to the complexity, the clinical and logistical complexity of this that I illustrated in the previous slides. Why is that? Well, the factors that contribute to the high cost of CAR-T cells are illustrated here on the left side of the slide, which is basically what I outlined to you before. There are multiple steps from vein to vein, if you like, from the minute that the cells are extracted from the veins of the individual until they can be reinfused. And so those multiple steps lead to high costs and various other barriers. And if you think about it, because this is a personalized product, it's a little bit like a cottage industry. Okay, so on the right side of the slide is a picture of how one would have made a shirt, maybe in the 15th century. Okay, so various artisans were sitting together in a room, Imagine this is our clean room in the hospital, our GMP room. We don't normally have dogs in there, of course, but nonetheless, this is the concept of personalized therapy, which I would argue is not fit for purpose. And what we want to do is move it into the industrial revolution such that CAR T cells are no longer individualized, but rather begin to look a bit more like a drug. And my dream when I set this up was that I would be able to write a prescription for IV administration of a CAR T vector that would go down to the pharmacy. The pharmacy would ship up to the patient's room a vial of lentivector that would be injected intravenously and make CAR T cells a little bit later. And you'll see how it started, and then I'll tell you a bit about how it's going. But I want to tell you a little bit about my thoughts on how cell therapy diverged from gene therapy. After all, these days we call it cell and gene therapy, and actually it's different. It's cell therapy, engineered cell therapy, and gene therapy is something else. But I think with the advent of in vivo CAR T cell therapeutics, we're actually getting back to much more similar cell and gene therapy. Hello, Sonia. And so how did cell therapy diverge from gene therapy? Well, these are the assumptions in the field and how we and you have broken those assumptions. The first assumption was that was about efficacy, and we've learned that younger is better. Rather than having more cells, we want better quality cells. The second assumption that led to why cell therapy diverged from gene therapy is biology, that T cells need to be activated, indeed strongly activated, for transduction to occur. And the final one is safety, that integrating viruses are non-specific. Well, I'll show you that they actually are very specific. And some of this is now in the clinic. I'm not showing you results here, but they're published. Actually, the first author of this paper was a co-fellow with me in Australia at Peter Mac, Michael Dickinson, who published a paper with the new process from Novartis, which is their T-Charge platform. And if you compare the process, the length of the process of the previous Kymriah or tisagenlecleucel to their new T-Charge platform, what ends up happening is that the ex vivo manufacturing is about 2 days instead of about 9 days. You end up with far fewer cells, of course, but each one of those cells is much more potent than each one of on a cell per cell basis is much more potent than the older standard commercially approved product, such that you can get, and this, I'm not showing you the clinical results, but you can get the same degree of complete responses in diffuse large B-cell lymphoma when you do that with much fewer cells. And so that tells us the younger is better. What about biology? Well, T cells need to be strongly activated for transduction to occur. This is an assumption. It's mostly true, but it's not entirely true. And the way this came about is by realizing that when cells are resting and you put a lentiviral vector, let's say, expressing GFP, you get very little, if any, transduction. There's very low level expression of the LDL receptor, which is the receptor for the VSV glycoprotein. And that this is an active process because it does not occur at 4 degrees, but occurs at 37 degrees. But whereas when the cells are activated on the bottom row, you can now see very nice transduction. And associated with that is upregulation of the LDL receptor. So do T cells need to be activated for transduction to occur? Well, this is data from a colleague and friend of mine, Saba Gassemi from the University of Pennsylvania, where each one of these plots represents a cohort of mice that are injected with a NELM6, an ALL cell line, followed by the injection of CAR T cells. On the y-axis is the tumor burden. Each individual line represents an individual mouse. And you can see that in this particular case, 3 million CAR-positive CAR-19 cells were not able to control disease when those cells were made in the usual fashion, which is about 9 days of activation, followed by resting and cryopreservation. In contrast, Saba showed that you can transduce resting cells using a variety of approaches. This is published. And what you can see is that even if you go down to certainly 2 million, but even if you go down to 0.2 million cells, which is less than a log-fold lower than the standard activated case on the right, you have a better tumor control. So what this tells us is that younger is better, less activated is better, and the T cells that are resting can be activated. This, even if you just take this at face value, this opens the possibility, notwithstanding release testing, the possibility of real point of care genetic modification. So what about safety? Is it a crazy thing to propose to inject lentiviral vectors into a human being? Aren't those extremely non-specific? Well, I would argue that they're exquisitely specific, because think about what happens when HIV enters the bloodstream of an individual. It requires CD4, it requires CCR5 if it is an R5 tropic virus. After all, if you don't have CD4, you don't get HIV. If you don't have CCR5, you don't get HIV. So these viruses are exquisitely specific. And so this opens the way towards using these approaches in a safe manner in vivo, which I'll show you how we do that in a second. What about insertional mutagenesis? Don't CAR T cells cause T cell malignancies? Well, rarely. Okay, so there was about 2 years ago, there was a lot of hoo-ha from several cases of T cell lymphomas occurring after CAR T cell therapy. In the fullness of time, it appears that there's probably about 3, maybe 5 bona fide cases of T cell malignancies that are caused by integration by true insertional mutagenesis, whereas most of those cases just occur sporadically. Patients who get B-cell lymphoma sometimes get T-cell lymphoma. So those have mostly not been related, despite the infusion of probably 35,000 people all over the world. So there are some bona fide cases, that is true, but very rarely. Sometimes insertional mutagenesis is actually a positive salutary thing, as you can see from a famous case from my institution with Joe Ferreira as the first author, showing that in some cases, when the CAR integrates into a locus, like TET2 in this case, that actually led to the proliferation and expansion of just one single T cell, which was able to eradicate chronic lymphocytic leukemia. So that case illustrates 2 things. Number one, sometimes insertional mutagenesis can be a positive thing, rarely. And sometimes, and it also illustrates, I think, that the most impressive thing from this paper shows us that a single T cell, if it's a really good quality T cell, can actually eradicate disease. And recently, our group published the long-term safety of lentiviral or gamma-retroviral gene-modified T-cell therapies in hundreds of years of patient follow-ups, showing not a single case of insertional mutagenesis. So what are the basic requirements for a safe and effective in vivo transduction platform that I will describe to you? Number one, you need a sufficient number of high-quality cells. Number two, you need to solve the biology of whether non-dividing T cells are receptive or not to genetic modification. And thirdly, you probably want to, in fact, I would argue strongly that you need to specifically deliver the transgene only to the candidate cells, your cells of interest, let's say T cells, and or to ensure specific expression of the transgene within the candidate cell. So what's a sufficient number of high-quality cells? Let's do a little back-of-the-envelope calculation, okay? So Yescarta axicabtagene, the dose is 200 million CAR-positive cells. What percentage of your T cells in the body do you need to transduce to get to 200 million? So in a healthy human being, let's say me, hopefully me, I have about 10 billion T cells in my blood with several assumptions, okay? The assumption is a 5-liter blood volume with CD4 and CD8 counts in the normal distribution. So I need a 2% transduction efficiency to get to 200 million. Let's make it a little bit of a tougher case, a patient who has maybe lymphoma, and maybe has had chemotherapy and lymphopenic and has only 1 billion cells. Here the transduction efficiency needs to be about 20% in the blood. However, I would argue that when you're looking at in vivo transduction, you have access to cells not just in the blood, but potentially all over the body. And so it raises the question, how many T cells are in a human body? Well, we don't know exactly, but we think approximately 4 × 10⁻11. So to get to 200 million CAR-positive cells, you only need a transduction efficiency of about 0.05%. And the reason I'm trying to strike home this point is because once you start to modify the VSVG, you really reduce, and I'll show you data, you really reduce the efficiency of the process. It's highly specific, but you really lose efficiency. And so it's important to consider that when thinking about whether this modality can be clinically relevant. And here's the distribution of T cells in the body, 2% in the blood, 40% in the bone marrow, and about 40% in the lymphoid organs, including lymph nodes and spleens. So this is I think, why we can consider that assuming the viral-like particles that I will show you traffic anywhere and everywhere in the body, it's actually quite feasible to get a transduction of a good number of cells that might mediate a potent anti-tumor activity. And this also opens a really interesting immunological question that, to my knowledge, has not been explored yet, which is, are tissue-resident immune effector cells better suited or equally well suited to undertake cancer immunotherapy than those in the blood if you could access them? After all, when we do apheresis, we can only access the ones in the blood. What is the function and potential function of T cells that are elsewhere in the body? So, efficacy, we need a sufficient number of high-quality cells. Biology, we need non-dividing T cells that are receptive to genetic modification. After all, we can't put anti-CD3, CD28 beads intravenously into the patient in order to stimulate T cells. People have tried that. when an agonistic anti-CD28 antibody was injected into human beings in 2008, most of those previously healthy individuals almost died and went to the ICU. So we cannot stimulate T cells in the body. We need to develop a system that can transduce resting or essentially resting cells. And as for safety, we do want specific delivery of the transgene to the candidate cell and or specific expression, as I said. So what are the tools at our disposal? We have, and I'll go into this in a bit more detail in a second, we have viral or non-viral approaches. I do believe that we want permanent genetic modification of T cells if we want to fight cancer. That may not be the case for autoimmune disease, we shall see. But in terms of viral therapeutics, we need to use an integrating virus like a lentivirus. If we want non-viral approaches, we have potentially the ability to use non-viral cut and paste mechanisms such as CRISPR or transposon-transposases. So this is a busy slide, which we're not going to go into. I just want to take you through from zooming in from high level to more granular. In terms of vectorology, viral vectors are represented on the left, non-virals on the right. The viral vectors are targeted lentiviral vectors, or from a family of pseudotyped with vesicular viruses, like VSVG, or AAVs. And for non-viral vectors, we have the potential for targeted LNPs or targeted polymeric nanoparticles. In terms of the targeted lentiviral vectors, again, these are pseudotyped not with the routine HIV typing, of course, but rather with vesicular viruses. The concept here is we have our let's say anti-T cell scFv that redirects the viral-like particles, and then inside the viral-like particles we have our transgene as usual. These viral-like particles bind to a cell surface protein on the T cell. Let's say it's CD3 or others. I'll show you some data momentarily. And then can enter the cell via pH-dependent entry or pH-independent entry for different pseudotyping strategies, and thereupon to undergo reverse transcription, integration, transcription, and translation. So I'm going to focus on viral vectors with pH-dependent entry rather than the other ones, though we've studied the other ones as well. Among the vesicular viruses, there is the VSVG, as I mentioned, or coccal protein. And when it comes to the VSVG, several mutations which change residues, such as K47, for example, actually can blind and remove the tropism of the VSV glycoprotein for its receptor, the LDL receptor. So this is really important. The mutants K47Q and R354A render the VSVG unable to bind to the LDL receptor, but crucially are not required for fusion. That's been published by others. What this means, the implication of this, is that it is possible to uncouple G-fusion activity and receptor recognition. And it is thus crucial and necessary to functionalize the virus with the ligand that triggers internalization upon binding to target cell receptors. So the concept is here. Our wild-type VSVG will infect any LDLR positive cell, which is many cells in our body, of course, not just immune cells. It will not infect and transduce resting T cells because, as I showed you, well, as you probably all know from your lab experiences, under most cases, but it will bind with activated T cells. The mutant will be blind completely and unable to infect any cell at all because it has no longer any tropism. Whereas if you use that blinding strategy and then add to that, supplement that with a targeting ligand, you can now transduce resting T cells and activated T cells. So what antigens should we go after? The antigen expression on T cells before and after bead activation is shown here, with the number of antigen molecules per cell shown on the y-axis for CD45, CD3, CD8, CD4, CD5, CD7, CD2, etc. And then with the inset, using a different scale, some other molecules, including the IL-7 receptor, CD123, CD28, and the LDL receptor, before and after. So I did found a company, Interis Biotherapeutics, which is now a clinical stage biotech. However, the company has been recently acquired by Kite Gilead, and so I don't actually have any conflict of interest anymore. They don't even talk to me anymore. So, but nonetheless, the reason it was acquired is because there are some interesting clinical results that I will share with you. Together with the company in the early days, which is about 4 years ago, we decided to go after CD7 rather than CD3. CD7 is a marker on most T cells and most NK cells as well. And so the strategy that I showed you before, rather than going after CD3, we went after CD7. We can talk later a bit about why and why not. And so the fusogen was blinded, like I show you, to the LDL receptor, and the transgene encapsulated therein is a fully human ofatumumab-based anti-CD20 CAR. Why CD20 rather than CD19? Lots of discussions, lots of thoughts, pros and cons, but we considered that probably most patients who go onto these trials will have failed prior anti-CD19 CAR T-cell therapy. Some of those patients have lost or downregulated CD19, not most, but nonetheless, it's at least a consideration. So I'm skipping through in vitro data and through mouse data, going straight to the monkey data. And another one of the reasons why we selected CD7 and CD20, CD7 to target the viral-like particles, VLPs, to T cells, and anti-CD20 CAR as our transgene, is because these reagents are cross-reactive between human and monkey, whereas CD19 is not. So that's another reason why we went into CD20. And I'm showing you two animals here. One is a control that received vehicle, and the other one received a single infusion of an anti-CD20 CD7 directed, so targeting CD7 positive T and NK cells, CAR20 bearing lentiviral particles, a single infusion. On the y-axis is the number of B cells in the blood of the animals, and you can see over 300 days, about actually a year, that there was no change in the number of B cells in the blood of the control monkey, whereas there was B cell aplasia for a year in the animal infused with the CD7-directed CAR20. And then the spleen was removed from the animals at necropsy. You see B cell follicles on the left. You see no B cell follicles on the right. And then when we look for the CAR T cells or CAR NK cells, the control animal is shown on the left, and the animal receiving CAR20 in vivo lentivector is shown on the right. Blue is DAPI nuclei. Green is for CD3 T cells in the top, and NKG2A on the bottom for NK cells. And then in red is the CAR, showing that we have CAR T cells and CAR NK cells. Next, I'll show you. Oh, I'm sorry. And so those were that was the single best monkey. Okay, so first I showed you the single best monkey. About 20 animals were treated with vehicle, with drug, if you like. 18 of those 20 animals developed anti-CAR protein immune responses by 7 to 14 days after vector injection, so it was very immunogenic in the monkeys. And I have to say, when I saw this data, I was a little bit concerned that the clinical trial would not work. I think it has worked, as I'll show you in a second. Perhaps an explanation might be that these are essentially human reagents injected into a monkey. You might imagine what would happen if monkey reagents were injected into a human being. Nonetheless, you can see that several of these animals did have B cell depletion, maybe aplasia for a short period of time, and then the one animal I showed you in the previous slides had, for whatever reason, it was not immunogenic and had a year of B cell aplasia after a single infusion, and this was safe with no toxicity at all in the animals. This leads to another open question for us all to consider, which is, what is the role of lymphodepletion that we do for standard ex vivo CAR T-cell therapy in minimizing immune rejection of engineered cells? What is the role of B-cell depletion in that as well? No B cells, maybe no antibodies. And of course, with this technology, I didn't explicitly say that, but this technology, of course, you cannot do lymphodepletion, because if you did, you wouldn't have any T cells to transduce. So this is a first-in-human study, and if you don't mind, please don't take any photos of this, because it's unpublished. This is a first-in-human study of CD7-directed CAR20, exactly the same thing that went into the animals. It's open at 2 sites in Australia so far, with the hope that, or a plan for it to open in Europe actually soon as well. Though after the acquisition, who knows what Kite plans to do. The trial is led by my colleague Michael Dickinson at the Peter MacCallum Cancer Centre in Melbourne, and was open to patients with relapse refractory B-cell malignancies, including ALL, but really all patients treated so far have been adults with lymphoma. CD20 has to be documented. I'll skip past this, but for those of you who are clinicians in the audience, you'll see that these are highly relapse refractory patients with mature B-cell malignancies. I will tell you that there's been no CRS, no neurotoxicity, and no infusion reactions in these patients. These are the first 3 patients, and then I think I'll show you another case after that. So the question is, what is the dose? How do you know what the dose is? Well, we have no idea what the dose is to infuse, because the drug is not cells, the drug is viral particles. So based on transducing units, the first 2 patients, dose level 1, were infused with 200 million transducing units, and nothing happened to their B cells. The first patient treated at dose level 2 received 6.3 million transducing units and achieved B-cell aplasia, which lasted for, well, I'm showing you 28 days here, but several months. This patient had mantle cell lymphoma with circulating disease, as mantle cell lymphoma sometimes does, and so his circulating mantle cell lymphoma cells were also eradicated. And this is the, I'm skipping through several patients to show you a particular indicative one, now at dose level 4 at 1 × 10^10 transducing units, a patient with diffuse large B-cell lymphoma who'd had multiple prior therapies. Sorry, I should have told you that on the previous slide, the patient in dose level 2 had received prior CAR-19 and had failed prior CAR-19. This patient elected not to get ex vivo CAR T cells, but wanted to go directly to the study and basically achieved a complete response with PET negativity and with elimination of this fairly unusual lymphoma mass in his leg, before therapy shown on the top, after therapy at day 28 shown on the bottom, with B-cell aplasia shown on the left, and with a PET scan, which admittedly is low-level disease, showing a PET negative CR. But this company is not the only one to be treating patients with in vivo CAR T. There's another company that treated patients in China. This patient is published, so you can take photos if you like. And it was published in The Lancet. It was a correspondence. I think it's a beautiful paper. This is in vivo BCMA-directed CAR T-cell therapy for myeloma. I think 4 or 5 patients were treated. The concept here is that, again, BCMA CAR is the transgene. They used a nanobody similar to the one used in Carvykti. Again, they mutated the VSVG to blind it. They also modify the producer cells in a number of ways, which we are also working on in my academic lab, including knocking out class 1 and overexpressing CD47 to reduce phagocytosis of these cells. And I'm going to run a little bit over time, so I apologize for that. But basically what this shows you is the baseline and then a day 28 PET CT scans of 2 different patients, one on the left, one on the right, showing a PET negative CR. And this was associated with quite considerable expansion of the CAR T cells in the blood of the patients, again, in vivo, CD3-directed CAR BCMA therapy. And in this case, unlike the case with the patients treated in Australia with a different drug, the Interius drug, in this case there was severe cytokine release syndrome with sort of ICU-level toxicities. And we can talk a little bit about why, but I suspect the difference is related to the fact that this VLP's targeted CD3. When you cross-link CD3, of course, you get very profound T-cell activation. When you cross-link CD7, that is not the case. But resting T cells are difficult to transduce, and now I'm showing you some in vitro preclinical data from my lab using BFP, blue fluorescent protein, as the reporter, with UT meaning untransduced negative control, VSVG being our usual lentivector positive control, and anti-CD7 means the blinded, redirected to CD7 vector. You can see high efficiency in stimulated T cells, and in unstimulated T cells you can see a teeny tiny population of BFP positive cells, especially in the anti-CD7 and maybe a little bit in the VSVG. This is 6 days after addition of the virus. And I'm showing you this to be quite transparent about the difficulties and challenges of this modality, but also to get you all thinking, as we are, about how we can improve it, because I think this is a really interesting area of investigation for people who want to get into the field. There is lower titer and lower infectivity when you modify the VSV glycoprotein, okay? And so that leads to a lot of potential for how to improve the system. And in the last few minutes, I'm going to show you just one of the ways, and you can be extremely creative. What I find really exciting about the field is that if you agree that it looks like there are some legs to this modality, then I think it opens up a huge area of investigation to be creative in how to further improve it. Because it needs to be improved. There are clinical results, not just from my group, others as well, and several companies are in the field as well, but we can do better. So one of the approaches, if you think about the basic concept, which is that resting T cells are minimally transducible, they are a little bit, minimally though, and we cannot inject anti-CD3, CD28 beads or antibodies into a human being. Well, how can we stimulate T cells in a human being? Maybe by vaccination. We do that all the time, yes? So we thought about what potential viral-specific T cells we could stimulate using vaccines in a person. And this is a busy slide. I'll just skip to the point, which is we thought that the SARS-CoV-2 vaccine might be a good one. And the reason is that there is broad immunization in the population. that there are, and there are vaccines for it. And in addition, a really interesting paper published in Nature recently showed a possible improvement in the in quality T cells. These are melanoma patients who received or did not receive the COVID vaccine and then received checkpoint inhibition for the treatment of melanoma. And you can see progression-free survival improved in those patients for some reason after they received the COVID vaccine in addition to the checkpoint blockade. So this tells us that maybe those T cells are very high-quality T cells. So we studied this in vitro by taking cells from the blood of healthy donors at the University of Pennsylvania, pulsing PBMCs with a peptide pool of SARS-CoV-2, and then seeing what happens. So DMSO is negative control, peptide pool is our cells of interest, and then activating beads is the positive control, using CD25 and CD69 to indicate T cell activation. And then you can see that whether you use VSVG or the blinded CD7-directed viral-like particles, you get activation, especially CD25 of cells 6 days after exposure to SARS-CoV-2 peptides. And then you can see, this is the more important slide, you can see, again, using blue fluorescent protein as our transgene of interest. In the DMSO negative case, we get very little, 0.23 transduction efficiency. With beads, you get very high expression, but in the middle there, you can see that when you use the peptide pool, you can get a considerable expansion, 1.5 log or something like that, in the number of cells that are transduced. So this tells us that there might be a possibility of just using something that is commercially available and routine, just to give patients some of these vaccines and to increase their transduction. I'm going to skip because I'm going to run over time, but this is basically data from, I think, five different healthy donors showing that this is indeed a robust result. And how we think we might do that is about a week or so, or maybe 2 weeks after the injection of a vaccine, we would then inject the lentiviral particles. We're now studying this in syngeneic models. I think it's important for this, particularly for this field of in vivo, to consider at least using syngeneic models. And we've now made several binders, which are not as easy to find as human binders, for mouse CD8 and for mouse IL-7, showing, I have to say, so far they're not exquisitely specific like the human reagents are, so we've got a little bit of work to do on that. And as for those of you who've tried using lentiviral vectors on mouse T cells, really difficult. Well, this technology actually allows you to get really high transduction of using lenti in mouse T cells, which opens up a range of possibilities. And finally, we're working with the John Wherry lab at Penn, which has extensive expertise with T cell exhaustion, of course, in viral models, to basically take mice that have been previously infected with LCMV and rejected that, so using the Armstrong strain, cleared the infection, and then 6 or 9 months after infection, you can see on the bottom left a control is clean, representative sample of T cells from the blood of the mice showing 4.56% of those cells are specific to LCMV, and you can see those are mostly T cell memory and T effector memory on the right. And what we propose to do is to basically vaccinate them or reinfect them with the virus, but basically a vaccination model, and then to infuse into them viral-like particles redirected against mouse reagents like CD8 to see whether our hypothesis that we showed in vitro in human cells is correct. So my summary slide is here. And again, I won't spend much time on it because of time, but basically there are different attributes for ex vivo and in vivo therapies. Maybe I'll focus on the fact that we know relatively little about the in vivo approach so far, but I think there are indicators that maybe this can work in patients with lymphoma or myeloma. We don't really understand the pharmacokinetics. They might be quite different to what we've come to expect from ex vivo CAR T cells. We don't know that much about the toxicity. There is a higher risk of off-target genetic modification, it's true, has not been seen yet, but very early days. And I think immunogenicity may be a particular problem for this and something that we and others are working on. So, okay, I'll stop there and thank you for your attention.
Okay, we have time for questions. Yeah, Sonia. Is there a microphone? There. Sorry.
No?
Yeah.
Hi.
I can repeat the question.
Okay. Thank you, sir, for a truly pioneering work. Congratulations on dosing one of the first patients with in vivo CAR T-cell. I think the field is going towards that direction. We all know that. So thanks for pioneering this and for this very beautiful talk that was very
Pleasure.
interesting. So I was wondering, in terms of co-stimulation, when you transduce naive T cells and they get activated through the CAR for the first time, does co-stimulation matters in terms of, can we go straight forward with 4-1BB? Should these CARs contain the CD28? And then about do you redose with the lentiviral vectors? And what do you expect in terms of immunogenicity?
Yeah, it's kind of hard to hear here because of the sound waves, but I think so your first question was about I think it's a great question. And basically, it is entirely possible that, as Sonia is postulating, that there is sort of an initial entry of the lentiviral particles into the T cells that may then secondarily activate them. Maybe even the pseudotransduction may activate them initially. And so it is possible that using CD28 or other co-stimulatory molecules may enhance this. I don't know. We actually haven't tried that, but I think it's a really good suggestion. Your second question, I think, related to immunogenicity of injecting lentiviral vectors. So I think we will have to see in humans what happens. I think the monkey results were pretty clear, but I'm not sure that they are representative of what happens in humans. We can study that in mice as well. But I think an active area of investigation, a very fruitful and important one, is to consider modifying the viral-like particles to make them hypoimmune, as is done in other fields.
Thank you very much for that impressive talk, Elena Garralda from Valdebrun. So from a clinical perspective, I'm really curious about how you selected the dose of the phase 1, and if you believe this is something that could be repeated as a follow-on, or it's only one dose that you're going to be giving, and if you believe this could be personalized due to the patient, or you're just going to do a normal dose escalation, like if it was just a regular therapy. I'm just curious about how you do this.
Yeah, great question. Thanks very much for that. So maybe I'll take the last one first. As with anything, we have to find what the maximal tolerated dose, if there is one, and then figure it out, just like we do for small molecules, etc. And I think tangentially, even in the field of small molecules, there's probably not the right dose for everyone. It's just that's what the approved dose is, and that's what we do. How do we come up with the dose? The maximal dose used in monkeys. There was no MTD, by the way, but the maximal in monkeys go down a few logs, start there with humans, and then escalate from there. Just don't know until such time as we. And it does appear preliminarily as though there is a dose response association.
That's very interesting. Thank you.
I think Elena had a question.
Thanks for such a beautiful talk. So it's quite established in the field that we need lymphodepletion for these T-cell therapies to work, but your data clearly challenges this idea, right? I'm wondering whether you think it could be a challenge when moving towards more solid cancers, or what are your thoughts on this?
Yeah, I don't think any of this directly helps solid cancers. That's another, to me, actually, the way I think about it, that is that if this becomes a reality, it just allows, because it's cheaper, faster, it just allows us to iterate faster and to get to the point where it can help with solid tumors. I don't at all propose any of what I showed you today helps with solid cancers, but it may sort of make things more feasible for us to iterate faster. Your first question was, yeah, okay. So, and I think that, so I just think that if it works, it's just going to be much more feasible for us to invest. Think about the clinical trials that we've been engaged in. Think about what we do in terms of, for example, here in Barcelona with cells made here. How expensive is it? How time-consuming and how logistically complicated is it? It's time to make things simpler and faster.
I was wondering, I was wondering from the regulatory perspective, when you went with the package to ask for the clinical trial, did they ask you for something that you found very challenging in this respect? Because I know that the integration of the viral particle, you addressed this discussion, but I was curious to see, because this is one of the first cases in which you were going with this kind of approach.
No, actually no. I'm not saying it was easy at all, but it was very practical and realistic. The company talked to, so, I personally advised the company to open in Australia with a different regulatory environment, that there is high quality, very high quality, but just not the FDA. And maybe it was the right decision, maybe it wasn't the right decision, but I thought it was really important to get into the clinic as quickly as possible and see whether this has any legs, whether this works, and then we can, talk to the FDA. So it was actually a conscious decision to, and maybe the FDA wouldn't have been more difficult than the Australian agencies or the European agency, which, as far as what I hear, was very reasonable in their discussions. It was appropriate, responsible, but not a crazy amount of questions.
On the types of T lymphocytes that are infected by the virus in your animal models? Are they resident or only
right,
circulating?
So, great question, thank you. In terms of what the types of T cells that were transduced in vivo, in humans, no idea. It's way too early. In monkeys, in the monkeys where we looked later, I showed you the example from the standout case. But in the other monkeys, there were just so few cells at the time of necropsy that we really couldn't comment on that. And so that's another reason why I'm focusing on immunocompetent mice in order to find that out. Thank you. Thanks very much.
Yeah. One last question.
Thank you for this talk. It's really inspiring. I was again thinking on, as Elena, in solid tumors, and I was wondering if maybe the vectors might reach the tissues, and there that could be the transformation, and if we have room in engineering on the vectors in reaching the tumor and occurring there the CAR formation.
Yeah, I think it's a great question. I think you should study that. It's a great question. Thank you.
Thank you very much. Okay, our next speaker is Dr. Uday Kamula. He is a surgical oncologist and tumor immunologist with over 25 years of experience in cancer immunotherapy. He was trained at the Johns Hopkins University in Maryland and in the Memorial Sloan Kettering Cancer Center. He's currently the director of the Solid Tumor Cellular Immunotherapy Program, and he leads efforts to develop next-generation TIL therapies to hard-to-treat solid cancers. He will be, in fact, discussing about precision TIL therapy with poorly immunogenic metastatic cancers. So thank you very much.
Thank you so much. And it's a great privilege to, sorry, jet lag voice. It's a great privilege to be here in Barcelona and to see so many familiar faces. So what I'd like to share with you is our effort at the University of Pittsburgh to develop a precision TIL therapy strategy for what I'll call poorly immunogenic metastatic cancers. And the first question, of course, is what exactly is a poorly immunogenic cancer? And as you'll see by the end of this presentation, that's a bit of a tricky question, but at the beginning, I think we can use a very pragmatic definition based upon two metrics that are plotted here, summarizing 75 clinical trials, 29 different tumor types, over 8,000 patients who all received what is arguably our most effective immunotherapy for solid cancer, checkpoint blockade of the PD-1, PD-L1 axis. And of course, you see this compelling relationship between tumor mutational burden and the objective response rate with this very elite, and I'll say very small group of solid cancers that have an extraordinarily high mutational burden, that have an equally high responsiveness. And these are the familiar cancers that we know so well, such as advanced skin melanoma, a subset of lung cancer, and then the cancers that still keep getting into the news but are extremely rare, the mismatch repair deficient cancers. Now, the prevailing theory, as all of you are aware, is that the very same mutations that give rise and help propagate these cancers may in fact serve as the Achilles heel, the weakness, in that a small subset of these mutations become true neoantigens that are processed and presented, recognized by the immune system, and exploited by checkpoint blockade. It's a very beautiful theory, but it has a rather ugly consequence, and that is that the vast majority of patients that we will see in our clinic with solid tumors do not fall into that high category. They fall here in this blue category, having a terribly low mutational burden and an equally terrible probability of responding to currently available immune therapies. And quite tragically, these are the common cancers that we see in our practice: garden variety colorectal, breast, pancreas, prostate, and so forth. So the focus of our research is really to develop more effective immune-based therapies for this large group of patients that are in desperate need. And it was for this reason that we began exploring, of course, the topic of this great symposium, the use of adoptive cell therapy. Now, what makes this strategy so powerful is that unlike checkpoint inhibition or cancer vaccines or cytokine therapy, which have to work within the body, in essence, they have to play by the rules of the body, with adoptive cell therapy, we can bend and even break the rules. Of course, it starts by removing the immune cells with deference to SAR for a brilliant discussion. The advantage of removing the cells from the body is we have great potential to activate them, expand them, select them, use a series of pharmacologic and genetic tools to improve them, and then we can take these cells and infuse them as the drug, as the cancer treatment, which is why this approach has been given the moniker of administering a living drug. Now, a variety of cell therapy products are on the agenda of this great symposium, and they fall into two main classes. You heard a very elegant discussion about peripheral blood cells, which normally don't have intrinsic anti-tumor capability, but one can confer tumor specificity by introducing the genes encoding either a chimeric antigen receptor to form a CAR T cell or a T cell receptor to form a T cell receptor transduced cells. Elegant biotechnology, but as far as the use in solid tumors, they have limitations. Typically, they target a single antigen, although there's some new generation products that might target two, but if the solid tumor diminishes or loses expression, these become quite limited. In the case of T-cell receptor transduced cells, they additionally require MHC restriction, and tumors are well known to downregulate either specific alleles or beta-2M, which make these therapeutics largely ineffective. And although they represent really exciting technology, they've shown a very limited impact thus far in solid tumors. The second main class, which I will focus in on, are called tumor-infiltrating lymphocytes. As the name would imply, these are the naturally occurring immune cells that exist in both primary and metastatic tumors, and pathologists have been describing them for well over 100 years. And you see a beautiful illustration of this sea of lymphocytes that appear to swarm around this tumor. Well, studies over the last 40 years have revealed that these T cells, unlike the genetically engineered cells, can recognize a wide array of both shared and highly personalized tumor antigens. And what's garnered a lot of attention in recent years is the ability of TIL to recognize those somatic mutations. And of course, they do so already completely MHC matched. So you can imagine if we can harness these TIL, they may represent the ultimate in a personalized precision therapeutic. We, of course, know that that's a lot easier said than done because as these T cells exist within the tumor, within the body, they're largely rendered suppressed and anergic due to the tumor microenvironment. And this is exactly where TIL therapy comes in because we can break these rules. It starts by excising a tumor. I'm a liver surgeon, so I tend to take out a lot of liver metastases, but we've successfully grown TIL from lung, soft tissue, lymph node. And once these tumors are in our laboratory, we can liberate the T cells from this toxic microenvironment. We can select different subsets. We can activate them, expand them in these bioreactors, and then we can do one thing that completely breaks all the rules. We can wipe out the patient's own immune system to repopulate them with these cancer-fighting cells. This is not a novel concept. In fact, Elena and I spent many years together at the surgery branch of the National Cancer Institute, where Steve Rosenberg pioneered this treatment. But for much of that time, it was in a single cancer, that, of course, being cutaneous melanoma. Quite logical choice. Cutaneous melanoma was one of the first to respond to high-dose cytokine, interleukin-2, anti-CTLA-4, anti-PD-1, anti-LAG-3, highly immunogenic tumor. But it served as a very logical basis to develop this treatment. Subsequent to that, centers around the globe replicated the efficacy of TIL therapy in cutaneous melanoma, which ultimately led to the very first FDA-approved agent called Amtagvi. It was a little bit of an interesting product in that as a first-gen product, it didn't really have a potency assay reflecting tumor specificity. This is very important that I'll come back to, but the FDA did give it approval based upon a response rate of about 32% in refractory melanoma. Well, as you can imagine, on the heels of this approval, everybody has been trying to develop the next great TIL product, and they're using the same playbook, which is quite logical. Pick a cancer that has a track record of responding to immunotherapy, high mutational burden, and studies are ongoing in lung cancer, as well as cutaneous squamous cell and cervical cancer. Totally makes sense. However, we asked a question that made no sense. We said, could we harness TIL therapy not to treat those highly sensitive cancers, but instead these cancers? Low mutational burden, low response rate to everything. And the reason why we did this is that these are the patients in need. And so about 15 years ago, my lab stopped studying the highly responsive cancers, and we decided to focus all of our energies on these cancers. And the cancer we had chosen so many years ago turns out to have the lowest mutational burden and the worst response to everything. And quite ironically, it was a form of melanoma, but one that no one talks about, called uveal melanoma. And we reasoned that this rare orphan cancer could represent the Rosetta Stone, the blueprint to develop more effective immunotherapies for pancreas, breast, colorectal, so forth. So what is this cancer? It's an orphan disease. It arises from melanocytes within the immune-privileged eye, but how most clinicians know it is because of its aggressive spread. And that's how I, as a liver surgeon, became involved. On the left, you see a normal liver, and sadly, on the right is a liver consumed with uveal melanoma. It's tense, it's black because of the pigment. These patients die a miserable death due to liver failure. And quite tragically, I told you we have an unprecedented number of effective options for cutaneous melanoma, and you can add TIL to the bottom. We have nothing for uveal melanoma, and that includes this new agent. And this intrigued us. So the best stories in science come from extremes in nature, and this was one of them. Two polar opposites of melanoma, cutaneous melanoma, the poster child for immunotherapy development for 40 years, and uveal melanoma, the cancer no one likes to talk about. And we thought if we could understand the difference, it might reveal important findings. So we conducted a simple experiment. We surgically resected liver metastases from these two types of melanoma, cutaneous and uveal, and we processed them exactly the same way in our lab. A couple of centimeters of tumor, we minced them into these small 1 millimeter fragments. And the reason why is each one of these fragments came from a geographically distinct part of the tumor. We know tumors, solid tumors, are quite heterogeneous. If there was TIL anywhere in the tumor, we wanted to find it. What we noticed was uveal melanoma TIL grew equally well. In fact, there was plenty of TIL in this cold cancer. And when we looked at the growth, we found that it was quite robust. So this is one of those tumor fragments, and you see these colonies of TIL start to grow within a few days. This is uveal melanoma. And then in a couple more days, the TIL will consume the culture. So we knew that the TIL were there and they grew, but the question is, did they have tumor specificity? Could they recognize the cancer? And the assay that we used was one that I think is the only reputable one. You can't use cell lines or PDXs or other alternatives because the antigens that are being recognized have to be in the autologous tumor. So at the time of surgery, we made sure to get enough fresh tumor tissue to serve as targets, and we used rigorous negative controls, which many groups don't. We used monocytes, which are completely HLA matched. They're from the patient. And this is what we saw. So this is cutaneous melanoma. This is the highly sensitive cancer. You see on the x-axis 8 liver metastases. Each dot represents an individual fragment culture, and the readout is interferon gamma. No great surprise, every single cutaneous melanoma patient produced tumor-reactive TIL. And in fact, what was quite remarkable is 80% of the cultures were reactive. So what that meant was everywhere we randomly biopsied the tumor, there were reactive TIL. Not surprising. This is the definition of a hot cancer. This is likely the explanation for why cutaneous melanoma has responded to literally every immune manipulation over 40 years. What we were interested in is, what did this look like in uveal melanoma? Well, this is what we reported a decade ago. Indeed, typical with a cold cancer, we found 40% of the cultures, nothing, just passenger cells with no reactivity. But what we were intrigued to see, by rigorously screening so many cultures from each tumor, we found that there were tumor-specific cultures, and in fact, in some cases, it rivaled the frequency seen in cutaneous melanoma. And we thought this was an important observation because what this suggested was that one of the coldest human cancers that we know was not poorly immunogenic. It was poorly responsive. The TIL were there. They were reactive. Well, we reported this a decade ago, but the dogma around cold cancers is so strong that people thought, can't be true. This is an artifact. We know uveal melanoma is not immunogenic. And so we thought the only definitive way of determining whether these could recognize cancer was to adoptively transfer them. So we conducted the very first ever TIL therapy trial for a cold cancer, and we saw a 35% response rate, which included both checkpoint refractory as well as naive patients. But what was important to me was the nature of the responses. We were not seeing little tumor shrinkage. We were seeing massive amounts of tumor regression. This is a PET scan. Everything in black shows tumor. This is a uveal melanoma involving the bone, liver, peritoneal cavity. And she showed us for the first time, one of the first patients, that TIL therapy could actually cause regression. You can really see this in the sagittal or side view. And then we also learned about durability, the hallmark of immunotherapy. This patient had liver metastases completely gone, and I show you scans at 3 years. And what was important was these same tumors were growing on checkpoint blockade. So this is the growth of those two metastases. You see they were rapidly growing on anti-CTLA and anti-PD-1 therapy, really reinforcing that these therapeutics weren't sufficient to elicit the activity of the TIL, but by liberating them and infusing them, you see this gratifying response. Well, we reported this back in 2017, and one of the tenets of clinical medicine is reproducibility. Could this actually be reproduced, and especially at another center? The NIH is a very special place. It has unique resources, and at the time I was moving to Pittsburgh, and so we built the manufacturing infrastructure within the academic program. We built the nursing and clinical infrastructure to conduct this follow-up trial, aimed to determine were those results reproducible, and could we determine the mechanism, and could we extend it to pancreatic cancer and other cancers? Well, let me address the first question. I reported these results at SITC last week. This is our new UPMC trial. This is a positive result. The new 31 patients are shown in red, and we have matched the original NCI results. And in fact, what has been so striking to me as the PI is that the patients are coming to us far sicker, far more advanced, and we're still seeing good clinical responses. This is a patient turned down for numerous clinical trials due to impending liver failure because his liver was being consumed with disease, hundreds and hundreds of tumors, and you see after infusion of TIL 1 year later, near complete response. Once again, this is uveal melanoma. This is a second patient, thankfully with less disease, but an equally good response. And we have seen brain metastases show regression. Now, of course, the question is, how is this working? And we are, of course, in the process of studying this. We are analyzing the genomics, aspects of the TIL, the patients. The strongest predictor that we have found turns out to be the potency of the infused cells. So what is potency? We took every one of these infusion products and tested them against autologous tumor, to determine their reactivity using standard immunologic criteria, production of interferon gamma, and upregulation of 4-1BB, as well as other activation markers. And when you look at the responding patients, they're all receiving tumor-reactive TIL. Now, this is important because many commercial and academic manufacturing operations don't test for potency, but we were finding this quite striking result. Among 28 uveal melanoma patients who received reactive TIL, the response rate was the highest that's ever been reported, 43%. But more importantly, patients who received in blue, the non-reactive TIL, derived no benefit. This became strikingly apparent on the spider plot. So for those of you not familiar, each patient is a line. When the line goes down, it's regression. The patients deriving significant and durable benefit. Our longest patient is out now 5 years. They're all getting reactive TIL. So what did we conclude? Potency matters. But potency is not tested for. And the reason why most groups don't test for potency, it's a pain. It's a pain for the patient because they have to undergo an operation. Then it's a pain for the manufacturing operation because they have to culture it for weeks. And then they have to do a quite tedious testing. And I can understand that because no one ever wants to tell a patient after a month, sorry, we're not going to treat you. So we thought, could we develop an alternative precision strategy where we wouldn't go to surgery? We would simply do a needle biopsy. And from this needle biopsy, we would analyze it using rapid RNA sequencing, which takes us about 3 days. And then we would try to predict with great degree of accuracy whether a given patient and a given tumor would harbor reactive TIL. Well, I don't have time to go through a lot of the aspects, but we reasoned that this approach would be minimally invasive, time and cost effective. It would help select both patient and tumor, prevent futile operations, and improve clinical outcomes. The first transcriptomic signature that we developed, we reported last year. I don't have time to go through all the details, but this is a gene signature composed of 2,400 both coding and non-coding transcripts that we found could replicate and reproduce the immunogenicity of tumors without really using this quite colloquial cold and hot terminology. And here you see the expression of this UMIS marker, the uveal melanoma immunogenomic score, across 100 tumors. And the question is, how good is it at predicting TIL potency? Well, we reported this data that if you take a needle biopsy and calculate UMIS, not only can it predict which metastases will produce tumor-reactive cultures, but it can quantitate the frequency of those cultures. And importantly, for the patients that I see and I operate on, we defined a threshold below which we don't see reactivity. The patients in the lower left are the ones that should not undergo surgical resection. Well, if this biomarker can predict potency, and then I showed you potency is the strongest predictor of response, then you might intuit that this biomarker might predict clinical response. And that's exactly what we found, where just screening a tumor based on this transcriptomic signature can very accurately predict not just the reactivity of the TIL that we produce, but their clinical response. So we've now started to employ this strategy of precision TIL therapy for uveal melanoma, but we've also extended it to other solid tumors, cold tumors. So one of the advantages of being in a busy surgical oncology section is we procured 500 tumors over the last few years, ranging from some of the coldest cancers that you will ever see, and we've developed a next-gen transcriptomic signature that we're calling TIL score. Now, TIL score, when you look at the expression, this is unpublished data, when you look at the expression across even some of the coldest cancers like liver cancer, sarcomas, pancreas cancer, so forth, are predicted to produce reactive TIL. Nothing would have predicted this. But when we validated this by doing the co-cultures with autologous tumors, once again, we are now predicting and demonstrating tumor-reactive TIL in sarcomas, mesothelioma, pancreas cancer, and so forth. And importantly, we've defined this cutoff below which we don't think that TIL therapy is going to be a suitable choice. Well, we're now testing this strategy in a series of clinical trials that are only being conducted at the University of Pittsburgh, now targeting virtually every solid cancer, and we're seeing encouraging results. So here's a patient with refractory peritoneal mesothelioma. She had 7 lines of prior therapy, including checkpoint blockade. She had a complete response of numerous intraperitoneal tumors causing bowel obstruction at 2.5 years. This is a young college student, 18-year-old with terrible refractory melanoma of unknown origin, who has been living a cancer-free life for 3 years. And we're very excited about pancreatic cancer. This is garden variety, low mutation pancreas cancer. All of these patients are refractory to first-line therapy. She had hundreds of lung nodules and had a complete response at 10 months, all using this precision strategy, biopsy, identifying that the tumor can produce these reactive TIL. Well, here's the data. You're seeing it for the first time. This is our this is our precision TIL therapy approach in refractory metastatic cancer. And we're just learning now. We're getting a handle on this technique and the approach, but we're seeing encouraging results in pancreatic cancer as well as other very cold cancers. So with that, I will summarize and say that adoptive transfer of potent tumor-reactive TIL can induce regression in patients that previously were thought to be poorly immunogenic, but now I think it's best to say they're poorly responsive to currently available immune therapies. But tumor transcriptomics seem to be allowing us to develop precision approaches for these patients. Now, efforts are still underway to enhance the durability, and so we have an active interest in trying to enhance persistence and mitigate exhaustion. Once again, these are efforts done ex vivo and studied in vivo. I'd like to thank everybody at the Hillman Cancer Center. Everything I've shown you was done within the academic environment, which you know in the United States is undergoing a lot of turmoil. It takes a great dedicated team of clinicians and scientists and nurses to do this, and I thank them all, and I thank all of you for your attention.
Thank you very much. We have time for 1 or 2 questions. Thank you. Yes.
Over here? No? To your right.
Here.
Oh, hola.
Here.
Oh, hi. Fantastic talk. I guess, congratulations. This looks incredible. I mean, I work in pancreas cancer, so it's always very encouraging to see. I guess one's more a conceptual question is, what is it about checkpoint blockade that you just break the barrier in these high mutational tumor burdens that like TILs just seem to just go, like power through all these barriers?
Yeah. So I think one of the key aspects is the lymphodepletion. The lymphodepletion that we give, I think, is the magic of many adoptive cell therapies. I can tell you I lived in the era before lymphodepletion at the NCI. The responses and durability are night and day. So what does lymphodepletion do? It's such a black box, so it makes more space for the T cells to proliferate. It gets rid of Tregs, MDSCs. It causes incredible changes both systemically and within the tumor microenvironment. We document drops in Tregs that can be sustained for a year to 2 years. So I think that there's a lot we need to learn, but I think it's this incredible ability to repopulate and change the immune system. And that's why I like this concept of we can bend and break the rules.
And a quick follow-up to that is, your signature, maybe I didn't get it, is it tumor specific?
Yeah, so the first signature, UMIS, was developed in uveal melanoma. The second signature is a cancer agnostic signature trained on that 500 multiple histology. So we use the same signature for pancreas, colorectal. So that's the new one. It's unpublished.
Yeah. Okay, thanks.
Okay.
Yeah, one quick question, please.
Thank you for your talk and congratulations on your great work.
Thank you.
I have more of a curiosity because with all of these poorly immunogenic cancers, even though they're not responsive to other immunotherapies or immune checkpoint blockade, did you find or did you check whether previous treatment with immune checkpoint blockades affects maybe your ability to find with your signature more tumor-specific high-potency TILs?
This is such a great question, and we did not find that checkpoint blockade or bispecifics can alter it, but I think that we're on that track because I think of these core biopsies as a window into the tumor. We're just farmers. We're plucking the TIL at their peak, and so our goal now is to find priming therapeutics to enhance the infiltration of the tumor, and then we go in and procure it. And we think that these minimally invasive biopsies allow serial analysis to find out which priming agents, maybe a cancer vaccine, or potentially other immune stimulatory agents so that we can then procure a more effective T-cell population.
That's very interesting. Thank you.
Thanks.
I've been told that I have been very strict with time, so we have time for one more question.
If I may, thank you so much for the really great presentation and promising result in MUM. Just a few weeks ago in the Presidential Symposium ESMO in Berlin, there was also the fantastic results presented for phase 1 study in TCRT in MUM as well, with almost 70% of ORR for the PRAME addressing tumor. Yeah. So my question is general in how can we ensure that those results currently presented and seen in MUM can be reproducible in the later phase 3 studies, for example? What are the key factors? Yeah, are you talking about the PRAME TCR or TIL? Both. Mostly for TIL, but yeah, PRAME, they also expressed in the hot tumors and cold tumors and cutaneous and synovial sarcoma. So I would say in general, because we are seeing currently really great presentations in MUM. Yeah, and so just for everybody, the question is, how do you scale this and make this common and positive in a phase 3 study? Now, of course, TIL therapy got approved in the United States without a phase 3 study. It was just a massive phase 2 study. I will make a comment about the PRAME TCR, which they reported at ESMO, Sapna Patel, with a really high response rate.
we have to remember that it was restricted to A2 positive patients, and they did select based on PRAME expression. And so the expandability to larger cohorts is still unclear, but it's very encouraging. With most TCR transduced therapies, the question is durability. Given that there's targeting a single antigen, can they in fact see responses that we've been seeing out 5 years? To your question, how do you then determine whether this therapy is only very niche-y for early phase trials? It's a great question. We just have to treat more patients. I think that if you had talked about cell therapy 15 years ago, people would have thought it was a pipe dream. And now SAR is showing in vivo CAR. People are manufacturing this left and right. So I think the technology will advance so that we can treat more patients. The question that we're focused in on is, can we make a better cell and find a better cell? And then I'm sure that the biotechnology industry will make it widely available for big, large studies. I hope that answers it. Good.
Yeah, one last question.
Yeah, so wonderful talk. Congratulations.
Thank
so
you.
my question is regarding the TCR repertoire. So did you study in your patient if there is any difference between the responder and non-responder?
Great. Yeah, so I didn't have time to show that, but we do TCR repertoire analysis for the alpha beta chain as well as gamma deltas in all of these patients. I didn't have time to show it, but if you do repertoire analysis of the infusion product and the persistence, all of the responding patients have striking engraftment and persistence of the clonotype that we give. And so what we're trying to figure out, and I'm surprised Elena didn't ask me, but what are the antigens? We are trying to find the antigens. Now that we know the specific TCR, I can tell you that they're not the product of somatic mutations. And you might expect our median tumor mutational burden is 0.7, less than 1 mutation per megabase. And so the somatic mutation load is so low that we think it's going to be cryptic antigens, the product of RNA splicing. But we are very interested in understanding which cells are doing all the work, and we're doing TCR repertoire to really be precise. And I'll close by saying one thing, and we go back into the tumor to biopsy it. And the one patient who's out 5 years, we had done biopsies, and 80% of the lymphocytes in her tumor were one clone that we had given in the infusion product. So it's a little bit of Darwinian survival of the fittest. They're these clones that outgrow.
Great. Thank you very much. It's a pleasure to introduce now Carlos Fernandez Larrea. He's a hematologist trained at Hospital Clinic in Barcelona. He's currently group leader at the DBAPS, and I have learned recently that he's been promoted to the head of the department. He was a visiting investigator at the Center for Amyloidosis in Pavia, NCI, and Memorial Sloan Kettering Cancer Center, always focusing on CAR T cells for myeloma. Today he will discuss about the academic model for CAR T development, results in multiple myeloma with ARI 0002 and beyond. Thank you very much, Carlos.
Good morning, buenos días, buen día. Thank you so much for the invitation to be in this amazing place with a full house and to talk about this, our academic model for CAR T cell development. We are going to hear more about that during these 2 days, our experience at Hospital Clinic, DBAPS, Universitat of Barcelona. And here I'm going to talk more about our experience in multiple myeloma. I am my principal disclosure and principal investigator on these trials. And we have seen impressive efficacy with commercial CAR T cells in multiple myeloma. However, they are expensive. We have difficulties in the distribution of the products, and there are still a strategy of optimization. We have heard about that with off-shelf products, but are still under development. With all that, we know that the access to CAR T cell therapy in the world remains challenging. Less than 10% of the potential patients are really receiving this treatment. And with all that, we have established a network, here you can see, of administration of these academic products in almost 15 hospitals now in Spain. And we have treated more than 500 patients with RE1, RE2, or RE3 products. That is this is a product that is coming from more than 300 people working in advanced therapies group with several backgrounds for pharmacology, immunology, and hematology, oncology, and go on. The vision of these advanced therapies in the hospital clinic is that we have a more sustainable system with a global access and always with patient-centered therapies. And this is not only limited to Catalonia or Spain. We have now more than 50 proposals around the world, and in fact, clinical trials with a product with RE2H are starting in the Netherlands and in Australia very soon. And the idea with all these projects is that you are now in the classical scientific approach that you request funding, you receive a grant, you publish in a very good journal, and after several years you are promoted to professor, head of department, or these nice things that you have heard. But anyway, we have to think more a drug developer approach, that at the end the crucial point is to contact regulator to develop a pathway to get an approval to reach patients with our clinical and relevant and medical need, and the publication is a secondary endpoint. We started with RE1 cells for against CD19. Our department of immunology is crucial in this development of this point of care strategy to production of the cells in the same center. And we got approval for patients older than 25 years with relapsed refractory acute lymphoblastic leukemia. This is reimbursed in Spain under the hospital exemption regimen, and it was designed as a prime drug by the EMA, and they have a pediatric investigation plan. There is a phase 2 clinical trial reported, and a European clinical trial is under development. But I'm going to focus on multiple myeloma. That is the second most frequent hematological malignancies due to the proliferation of malignant plasma cells. And even we have amazing results in the last years in improving the survival of the patient from 3, potentially now beyond 10 years. It's true that when the patient develops a relapsed refractory disease, the prognosis is very poor. With that, we developed this RE2H, Cell. It's a 4-1BB-based CAR-T with a humanized anti-BCMA scFv that it was developed in our institution, REDIVAS, after in vitro and in vivo confirmation of the activity. And we started a clinical trial sponsored by our National Institute of Health, initially in 5 centers, after in 7 centers. The viral production and the coordination of the trial was performed here in the hospital clinic in Barcelona. There were 2 centers for cell production in Barcelona and also in Pamplona, in the Clínica Universidad de Navarra, and the other centers performed the apheresis and treated locally their patients. Some of the consideration in this trial was the fractionated matter of the administration of the first dose, something that we learned with our first trial with RE1 that can reduce the toxicity without any impact in efficacy. And we explored for the first time this strategy as a booster dose, at least after 3 months, in patients with some degree of response and no relevant toxicities. Here you can see the characteristics of the patients, and mainly was enriched by high-risk features such as extramedullary disease, high-risk cytogenetics, and refractoriness to the main therapies available for multiple myeloma. We have shown that the patient had a response of 95%, almost 60% achieved complete response, and that was related to a high minimal residual disease negative that was analyzed in a centralized manner. That was translated in a progression-free survival, of a median progression-free survival of 20 months, with an overall survival that is not reached after 2 years of median follow-up, and the final results of the clinical trial will be reported next year. We have studies on correlation, for example, the correlative studies, for example, the expression of the BCMA in the surface of the malignant plasma cell. We have seen such other groups that the reduction of the expression could be important at the time of relapse when we compare with the plasma cell at the time of the screening. And we have seen also that some features, for example, the extramedullary disease, particularly those true extramedullary disease, not related to bone disease, are associated with shorter progression-free survival, shorter overall survival, and particularly when it was related to a higher tumor volume measured by PET-CT. The second dose is very safe. We have not seen any side effect, and we are administering now the second dose in the outpatient clinic. And we were able to see that almost 1 out of 4 of the patients improved their response after the second dose. About safety, the majority of patients developed a cytokine release syndrome, but mainly grade 1 or 2. I was also a kind of escalation intra-patient because we were able to stop the treatment after 2 doses in some of the patients, and it was not related to any difference in efficacy. And importantly, as many of you can know, that there is also a problem with late neurotoxicity with some products with CAR T-cells in myeloma. We have a very low grade 1 ICANS initially, and we have not seen any late neurological toxicities. With all that, we were able to describe the results in the main events of the hematology community, also in our society in Spain. And last year, we got the approval by the Spanish agency of the RE2H for patients with relapsed refractory multiple myeloma after at least 2 lines of treatment, and there were patients that were exposed to the main 3 families of drugs: immunomodulatory drugs, proteasome inhibitors, anti-CD38 monoclonal antibodies. We are going beyond this trial, and we have treated now more than 180 patients, initially in a compassionate use and now in the hospital extension program, using our academic point-of-care network in Spain. It's less than 100,000 euros annual reimbursement for patients who meet the criteria after the national committee evaluation, and an academic European trial with the confirmatory with this point-of-care strategy is currently in development. With that, the idea is to improve the access using this academic point of care with a CAR T manufacturing that is equivalent in different facilities with an homogeneous clinical management. Beyond myeloma, we have treated the first patient in the world with our anti-BCMA CAR T therapy with AL amyloidosis. The patient achieved a complete response. It was a patient with renal involvement by amyloid, and I saw her last Monday, and she's still in complete remission, negative for the disease with all the measurements and with renal response also. With all that, we know that we have used these therapies in very advanced phases, and that is to move in earlier stage of the disease. And one of the ideas is to use this CAR T in the most aggressive form of monoclonal gammopathy, that is the plasma cell leukemia. That here in Catalonia, we also started a new definition that is now by the presence of 5 or more percentage of circulating plasma cells in patients with multiple myeloma. And with that, we started the clinical trial this year in the context of the Spanish myeloma group. And we are using the fractionated manner, but in first line. After induction therapy, instead of autologous or allogeneic stem cell transplantation, we are using this CAR T. And we have now more than 5 patients included and 3 patients already infused. Beyond RE2H, we are also working in our research group at IDIBAS. We are working in several strategies to improve the efficacy of this, in particular in patients also exposed to anti-BCMA therapies, using immunocheckpoint blocker, using genetic disruption of some transcriptional factors such as BLIM1 in terms of increased efficacy and the persistence of the CAR-T, in the development of bicistronic CAR-T BCMA. For example, I was working on the memorial with the BCMA GPRC5D. Here we have the experience with BCMA CD229, and also with the modification of some structure of the CAR-T, such as the transmembrane domain, also in order to increase the persistence and potentially the efficacy. We have demonstrated that in vivo and in mice, and the idea is to move forward also in patients. And we have also several correlative studies coming from the results from our patients, trying to predict the activity of the CAR-T cells, new ways to evaluate the response. For example, I have shown about the extramedullary disease, but also the evaluation by mass spectrometry or the minimal residual disease centralized. The impact of gut microbiota that in collaboration with our Department of Immunology and with our Department of Pharmacology to go deeper in this strategy of point of care and how to translate that not only in Spain but also in other regions in the world. And with that, I would like to thank all our funders. Without that help, it's impossible to develop this program, and particularly to all our patients, to all their families, to all the centers that have participated in these strategies for the treatment of several malignant hematological diseases, particularly multiple myeloma, and to all our team at Hospital Clínic IDIBAS, Universitat de Barcelona, that are committed to this very complex process, but that at the end is quite successful, and at the end we are very happy to see our patients living longer and living better. And thank you so much for your attention.
So, questions? Yes.
Hello. Thank you so much. This is really impressive. Although I'm from Barcelona, I now live in the US. And I think that when I hear the Barcelona model and I hear all this, how you guys managed to do this, I think this is truly impressive. And so first of all, I just want to congratulate everyone because I feel like 10 years ago when I left, nothing was here, so congratulations. I actually think about what you call point of care manufacturing. Is that truly point of care? So you move all the patients around and do they all come to Barcelona? Or can you actually just do centralized manufacturing and then just shift the leucas and the final products back and forth?
Yeah, it's a mixed point of care strategy because now, for example, for myeloma, we only have 2 points of care in Spain, one in Navarra, one here. The patient treated in Navarra or here are really point of care because the cells are going from the apheresis unit to the lab and from the lab to the ward. But in other centers, we are using this mix because they are still during the apheresis, like in Galicia, in Sevilla, or in Murcia. They are sending the cell to one of these 2 centers, and the cells are coming back to the regional hospital. And it's more a mix. The idea is that to grow and to have more this point of care for production around Spain and around the world. We have already started in other European countries and even in Australia. And that the idea is that there could be also a wider network for the treatment of these patients.
And so just following up on that, so what does make more financial sense, and maybe not only financial but logistical sense? Is it to have like a huge manufacturing hub where you can basically send back and forth like cryopreserved leucas with cryopreserved final products and maybe manufacture everything in Barcelona or somewhere else and then ship everywhere in Europe? Or does it make more sense that big hospitals have their own
Well, that is a philosophy. If you ask me, I believe more in our model. I think that it's more patient-centered. I think that the criteria in this case is coming from the clinics. What is the patient that needs the treatment the first? It's also, you can adapt potentially to the clinical needs of the patient, and you can release the product. Can release the product as soon as 20 days after the production if the patient requires the treatment. And that is been very important in the clinical trial. I think that saving even some patients, that that is an important number of patients that are dropping out of after the apheresis because they are progressing, for example, in multiple myeloma with refractory disease. And I think that to be close is that our experience with allogeneic stem cell transplantation or other cell therapies in hematology, that the production is close to the... I know that and it's difficult to think how we are going to escalate when we are going to have more access to patients around the world, and I think that is also another point.
Hello, I'm Silvia Martin, also from the VHIO. I also think that the centralized model has some advantages. One of them you were pointing is that the patient has not to travel so much, and this sometimes is a barrier for many families when they have to treat someone. So this is a point that is an advantage. And also I think that the point that, in fact, you are more resilient. I mean, if you have a company that is producing this product and you have only one company, if this company goes away, forget about the product. And this is something that is happening. So I think that the centralized model also has produced more resilience in the system. But I was wondering, because if you go to the centralized production, then you have to establish manufacturing centers in different hospitals or institutions. And I was, at this moment, do you have to make the production exactly the same from one center to another? So it's exactly the same, or you have some flexibility in your strategy?
Well, you have to get the approval of the agency, and you have to go through our validation runs, and that you are very close product. We know that you have to use the same apheresis and to validate that the product at the end is the same. We know that the variability among products is very high because of the patients, but you have to validate that at least 3 brands are the same, and they are exactly you have to learn how to produce exactly the product that's in the initial, and we are also exploring how to escalate that, and that is a point of how to continue growing in this point. And I have to say, I would like to add something that is academic freedom also. You have seen that we are moving, for example, to plasma cell leukemia or to AL amyloidosis or to other monoclonal gammopathies. That is something that is very difficult for a centralized, industry-focused strategy, because they are a small subpopulation, and I think that it's very important to have this possibility to decide also to move forward to specific subpopulations.
Oh.
Thank you. Congratulations as well. I have to say, in the US, we're very envious that you can do this. It's really, really exciting. How do you decide which of your patients will get your product versus Carvykti or whatever, or are those not available here?
Yeah, that is an important question now. Before this year, it was very easy because it was no Carvykti available in Spain, because you know that in Europe, the approval is not the same as reimbursement. You can get the approval in Europe. I had that slide, but in the 27 countries, it's very heterogeneous, the access to the drugs. And in the majority of countries in Europe, for example, Carvykti is not available at all. And however, we have Carvykti right now available in Spain. We have just started, for example, in many centers in Catalonia this year. And the decision is mainly, well, our Carvykti is approved after 1 line of treatment in patients refractory to lenalidomide and exposed to bortezomib. That is carefully analyzed. You know the cost is higher for our public system. And the idea is we have also a very interesting safety profile. I think that with the RE2, particularly in patients that cannot get a deep response before the CAR T, or you cannot predict that a patient can have a good response after bridging therapy, because we know that the mortality related to some of the CARs, like CARVYKTI, could be 15%, and the potentially of toxicities, neurological toxicity in form of Parkinson's could be 6%. That is a complication that until now have no clear treatment. And we think that when we can administrate now RE2H in selected patients in an at-home program, that is, the patient receives the treatment, the infusion in the hospital, but the whole process is performed at home. And I think that is also very important for the sustainability of our system. As head of the department, I know that beds are very expensive, are very limited in Catalonia, for example, and everything that we can do in this regard is crucial.
That's wonderful, thank you. Can I ask a follow-up question? What is the number of patients in Spain that would be eligible for this product, and what percentage of them get the product?
In this case, yeah, initially we had only the program was a clinical trial, and we had 60 patients because it was our possibility, and was also agreed with the Spanish agency, the drug agency. We have a limitation, of course. There is now it's impossible to treat all the patients that we have. We have 5 patients every year, every 100,000 inhabitants, and we have it's not possible to treat all the patients with this. And we think that we have altogether with the commercial option that we have in Spain, with the clinical trial that we are very lucky also to have several clinical trials with new products also here, plus this academic, to consider each patient to what is the best treatment that the patient should receive, knowing that the best treatment that the patient can receive is the treatment that they can receive.
Thank you.
Thank you so much.
And our last speaker before the coffee break is Dr. Gloria González Asedino-Laza. She got her PhD from Universidad Autónoma de Madrid, and she did a postdoc in New York University working on vaccines. She's the former head of the gene therapy for viral hepatitis lab and deputy director at CIMA. And she's currently the coordinator of advanced therapies and diagnostic innovation at IDISNA. She is the co-founder of VIVE Therapeutics and president-elect of the Spanish Society of Gene and Cell Therapy. She will be discussing about gene therapy for rare liver disorders, from the bench to the patients.
Thank you very much for the introduction, and we are going to really completely change gears, because as you see, I'm not talking about cancer at all. But I'm very happy, I'm very glad for this invitation to be in this really wonderful place, and also to be exposed to these wonderful talks. No, I'm really learning a lot because they are really fantastic. So I'm going to talk about the work we are doing in the lab, which is about using gene therapy for rare liver disorders. And I'm going to guide you through the journey we made from animal studies to the patients, and how we are going back to the lab to understand how we can improve these gene therapy approaches. We are focused on rare diseases that affect the liver. So the gene deficit is mainly affecting the liver, and diseases that can be cured by liver transplantation. And the rationale for that is that if liver transplantation is the cure for those diseases, if we are able to modify the genetic content on the liver, we will be able to hopefully change the clinical course of the disease. So this is our approach. Let's bring the genetic material to the liver to cure the disease. And among the diseases that we have identified that can be cured by liver transplantation mainly, we focus on several of them, but here I'm going to talk to you about Wilson disease because it's the first disease in which we were able to reach the clinic. So the vehicle that we are using and has been already mentioned to bring this genetic material to the liver are adeno-associated vectors. And adeno-associated vectors are very, very small vectors of 25 nanometers in diameter and has a DNA, very simple DNA of 4.5 kb of a single-stranded DNA. They have several characteristics that makes AB very attractive for the use in gene therapy, particularly for inherited diseases, such as, for example, they are non-pathogenic, they are not able to replicate by themselves, they need the co-infection by other virus to initiate their replication, and although most of us, we are infected by AAVs, we don't know because this virus doesn't induce any pathogenic disease. So as you can see, these are, for the safety point of view, very, very attractive vectors. And this is the schematic representation of the DNA of an adeno-associated vector. It has only two genes, one that codifies for the proteins involved in the replication, the rep gene, and the other one, the cap gene that encodes for protein involved in the formation of the capsids. And at the end of the genome, they have two inverted terminal repeats that are the sequences that are necessary for the vector, for the genome to be recognized as the genome of a viral vector. So we can remove the rep and the cap gene and introduce our therapeutic material. And just by maintaining these two ITR sequences, we are able to produce recombinant vectors very easily in the lab. So one of the limitations this vector has, and I will explain you later how this impacts the development of genetic therapies, is that it's very small, so it only can fit a certain amount of genetic material. In this case, it's around 4.5 kb. So we started working on Wilson disease because there was a foundation that has a family that was affected for Wilson disease, and we started the development of this vector by 5 people in the group that really take this very seriously. Dani, Ruben, Miren, Oyana, and Christina. So I want to acknowledge them from the beginning. So what we did, what we first did was analyze what Wilson disease is, of course, and Wilson disease is an autosomic recessive disease of the copper metabolism that affects around 30 people per million inhabitants. It is caused by mutation in the ATP7B gene that encodes the ATP7B protein, which is a copper transporter. In the absence of this protein, the copper accumulates in the liver, and this is what causes the disease. After the accumulation of copper in the liver, the cells start dying, and this copper goes into the circulation, affecting other organs, and particularly the brain. So this patient has as characteristics to have liver damage that usually progress to cirrhosis, but also neurological damage and the neurological symptoms like dystonia, cognitive impairment, personality changes. So it's a disease with many different pathological aspects. The current treatment for this disease is daily uptake of copper chelators several times per day. So really the medication is really cumbersome, and the main issue we have is that people don't take usually the medication, so adherence is a problem, and when the patient doesn't take the medication, the disease progresses very rapidly, and when there is a crisis, a Wilson disease crisis, usually is fatal in 100% of the cases. And on the other hand, they can develop other symptomatologies that leads to maybe cirrhosis. So to understand my results, particularly the first part, which is the results in the animal model. We need to understand how ATP7B work and which are the aspects that we will analyze in the animal model. So first, copper is essential for life, so we need to uptake copper every day, and we uptake copper from our meal, and we cannot prevent copper uptake. We can reduce the amount of food that has very high copper, like chocolate or seafood, but we cannot remove copper from the diet. So copper is essential, but has to be very well controlled. As soon as we have an excess of copper, since it's a very reactive element, can cause oxidative stress, and this is what really is involved in the disease. So copper is uptake in the intestine. It travels to the liver, and in the liver, this copper goes to the Golgi apparatus, where it forms cuproproteins. And the main cuproprotein is ceruloplasmin, which is secreted into the circulation and bring copper all over the body. The other function that ATP7B does, on top of transporting copper to the Golgi apparatus and bringing to cuproproteins, is also to remove this excess of copper by binding this copper and bringing them to vesicles that will be, this copper will be secreted into the bile and will appear in the feces. So what happens when there is no ATP7B? First, the body tries to compensate the excess of copper by overexpressing metallothioneins and other proteins that can bind and make this copper safe. But in the case that this is not possible, this copper starts accumulating in the liver hepatocytes. Another aspect is that since we don't have ATP7B, this copper cannot be transported to Golgi, and we don't have ceruloplasmin. So the patients with Wilson disease has low levels of ceruloplasmin in circulation. And this copper that accumulates that cannot be excreted into the bile and eliminated into the feces is excreted into the circulation, and this copper levels increase in the blood and is eliminated through the urine. Okay? So what we are going to see in patients and in mouse with ATP7B deficiency is an absence of ceruloplasmin, the apparition of liver damage, so transaminase elevation, and an increase in copper in urine and a decrease of copper in feces. Okay? So with this in mind, we have to go now to see how we explore the therapeutic efficacy of our vector. And first, we have to construct our vector, and as I said, we focus on liver diseases, so we use vectors that can go to the liver, that can transduce well hepatocytes, and we control the expression of the therapeutic gene by using a liver-specific promoter. As I mentioned earlier, these vectors has the limitation of the amount of material that we can introduce. And this was important in this particular case because ATP7B is a large gene. And ATP7B protein has several transmembrane regions, but also has an N-terminal sequence that bounds to copper. And they have several metal binding sites at the N-terminal region. And when we were looking at this protein in different species and different organisms, we found that depending on the organism, we found different numbers of metal binding sites. And since we have this issue of introducing this gene into the AAV vector, we start playing around with the number of metal binding sites we introduce in the gene. And after several rounds of testing different variants of this mini ATP7B gene, we found that the mini ATP7B gene with 2 metal binding sites that were located in the 5 and 6 position was perfectly functional and fits very well into our AAV expression cassette. So we produced this vector, we proved that the protein was properly localized in the bile canaliculi, and we tested the efficacy of this vector in ATP7B knockout mice, so a Wilson disease mouse model. And we analyzed all the parameters I just mentioned and many, many more. So the first thing we did was inject different doses of this vector in male and female Wilson disease mice, and we compare, for example, liver damage in animals untreated or healthy animals as control. And as you can see, at the 3 doses that we have tested, we were able to control liver damage in the animals, both in male and in females, as shown by a clear reduction of transaminase elevation and bilirubin levels in serum, very different to the animals untreated and very similar to healthy control animals. And also, this accumulation of copper affects the functionality of the liver, and one of the main markers is the biosynthetic pathways of the liver. For example, the production of albumin that is clearly reduced in Wilson disease animal untreated, but that can be restored after the administration of the gene therapy vector. So we reduce liver damage, we improve liver functionality. What happened with the copper? So we analyzed the copper in urine that, as you can see, is significantly elevated in animals untreated and is reduced in a dose-dependent manner after the administration of hardware vector that I forgot to mention is called VTX-801. So we observed this reduction both in male and in females, and at the higher dose, we were able to reach really normal levels. We also increased ceruloplasmin production in the animals after the administration of the vector, again, in a dose-dependent manner. So these animals are able to, again, have copper all over the body because this is what ceruloplasmin does. Then we analyze why this is happening, and of course you will expect that this improvement in liver function and reduction of liver damage will be related to the decrease of copper concentration in the liver, and in fact this is the case. We observe a clear reduction of copper concentration in the liver of treated animals, and again in a dose-dependent manner. And also, apart from having this reduction in the copper in the liver, that is the organ that is expressed in the ATP7B, we were able to prevent the accumulation of copper in other organs, like the brain, although we never reached null bar levels, but we also were able to reduce copper in the kidney, reaching normal levels at all the doses tested. And this is just an image showing how we can really see in an image how we can decrease this copper concentration from the liver after the administration of the treatment. So the next question, and because everything is telling us that this treatment is working and maybe we can bring this to patients with Wilson disease, was how many cells do we need to transduce with this vector to achieve a therapeutic effect? Because it's true that in mice we really can reach 100% of the hepatocytes with the AAV vector, but this will be very difficult to achieve in a patient which has a much bigger liver, and probably the dose that we need to administer will be difficult to reach and can have some safety concern. So we analyze how many cells do we need to achieve to really have a clinical benefit on those animals. And we analyze the amount of messenger RNA in the liver of those animals by in situ hybridization, and we found that already with 10% of the hepatocytes transduced, we can really improve the hepatic pathology in those animals. You have seen that we are able to reduce completely liver damage by transaminases. We are able to improve ceruloplasmin elevation and reduce copper from the liver. So we were really excited about this, and we thought, okay, let's move to the clinic, but probably we will need, doing liver biopsies in the patient will be kind of difficult, and maybe we can develop a system that will allow us to determine if our therapy is working in non-invasive manner. So what we did was use radiocopper and PET analysis to be able to demonstrate if we are able in the animals to recover a proper copper metabolism. And what we did was treat animals with our vector at different doses, and after 3 months, we administered to the vector radiocopper. The radiocopper is administered at very, very low dose, but the signal that you can detect it by PET, as you can see, is very high. So what we observed is that, as we were expecting, in a healthy animal, copper is eliminated through the feces, so you can detect the copper in the intestine and is not accumulated in the liver. While in the animals of Wilson disease, the copper is retained, the radiocopper is retained in the liver. And as soon as we treated the animals with the vector, you can see how the increase in doses resulted in the apparition of this copper in the feces, no? So it's going to be eliminated into, it will appear into the feces, and it's reduced from the liver. And when we analyze these feces, you can see how with the higher doses, the levels of copper in the feces are very similar to a wild-type animal. On the other hand, this technique also allows us to detect radiocopper in the blood, and since this radiocopper should be bound to ceruloplasmin when you have ATP7B, the pattern of the presence of copper in circulation is different between untreated animals, treated animals, and wild-type animals. So you can see how the kinetic of the presence of copper in circulation is completely different from healthy animals in green and untreated animals in red, and how the administration of our vector changed this kinetic. And there is a very important or very useful ratio of the detection of radiocopper in the circulation, which is the ratio of radiocopper at 24 hours versus 2 hours. So it can clearly differentiate wild-type animals versus untreated animals, and this increase with the administration of our vector. So we use this ratio to analyze patients, Wilson disease patients, healthy individuals, and patients that are heterozygous. And as you can see in the graph in the right, we can really discriminate between healthy volunteers that has higher ratio as well as mice, and heterozygous patients that are like in the middle. They have half of the expression of the protein of the ATP7B, but are perfectly healthy, and patients that with Wilson disease which has lower ratio. And we use these parameters for the clinical trial to discriminate if the patient has response after vector administration. And this is the clinical trial we are pursuing now. We have 9 sites in Europe and the States. The planned number of patients is 16, and the follow-up of the study will be 5 years. So this is the schematic representation of the trial, but I'm going to go very fast. We will administer the vector at day 1, but first we will check the radiocopper profile in those patients because we are not going to remove the background therapy of those patients. We will remove this background therapy, this chelator therapy, only if the patient is responder. So only if the vector is working and the protein is being expressed in the liver. So we will analyze copper at baseline and 3 months and 1 year later. So this is a escalating the dose study. So I'm going to show you here the results of the cohort number 1, but we are going to introduce 3 different cohorts depending on the efficacy result in the different groups. So we have treated the first 2 patients, cohort 1, and we have already observed some adverse events in those patients that are usually very common, in AAV clinical trials, but are adverse event that can be easily managed by the clinician, like no infusion-associated reaction, transient lymphopenia. ALT elevation is a very common adverse event in patients treated with AAV vectors that usually are managed by the use of corticoids. So they are in general, they were general in self-resolving, and this is the first patient has 4 different severe adverse events, and the second patient has only an elevation of transaminases the day post-infusion that was self-resolved. Then, when we analyze the therapeutic effect in this patient, what we observe is that ceruloplasmin levels were increasing after time, but this increase was, let's say, moderate. It didn't reach normal levels. Regarding copper in urine, what we observed that one of the patients reached normal levels, while in the other the copper urine was fluctuating. So we have some sign of efficacy, but they were quite poor. Then when we the capacity or the possibility of doing a biopsy was optional, but the first patient agreed to give a biopsy before and after. And what we observed is that at baseline, we have some fibrosis in this patient that was absent 1 year later. And also we observed an accumulation of iron at baseline that disappeared 1 year later. However, when we analyze copper concentration in this patient, the copper concentration was unaltered. Another thing that we were able to do was to analyze how many cells were transduced in the liver of this patient. And you can see here, this is DNA and RNA from the vector, and the levels of this transduction were, according to the analysis performed by the CRO, were around 50 to 75% of the cells after 1 year. So this is the first cohort summary. The treatment was safe and was well tolerated. There was a slight but not significant improvement in copper-related biomarkers, and we were a little bit puzzled because, as you can see, we have 50% of the cells transduced, which were more than the cells we have in patients treated in mice treated with the same vector. So just as a reminder, with 10%, we have a very good clinical benefit. So now what we are doing in the lab, we are going back to the lab, trying to understand why this therapy is not working as well as in mice. We are trying to see if, since our mice is cow, if the problem is that the expression of the mutant protein in the patient is altering the activity of the recombinant protein, and also if there is changes in the functionality of the protein depending on the species. And just to finish, because I'm running out of time, this is my last slide, which is just to thank all the people that has worked in this whole project, from the preclinical data performed at CIMA University of Navarra and Vive Therapeutic, which was the sponsor of the clinical trial. And thanks for your attention.
Yes, over there.
Congratulations. It's a very important study, especially for these metabolic deficiencies. So one of the main challenges with AAV constructs are neutralizing antibodies in the human. And it looks like your liver expression was robust. Have you measured neutralizing antibodies pre and post? Because if you're going to envision repetitive treatment, this is certainly going to be an obstacle.
Oh yeah, absolutely. Yeah, you are completely right. I mean, one of the exclusion criteria in all AAV trials, or nearly all AAV trials, is the presence of neutralizing antibodies. So those patients are negative before treatment, and highly positive after treatment, I can tell you. However, in anticipation for that, we are already working on strategies to remove antibodies. There is IgG depleting, cleaving enzymes that can be used just prior AAV administration, and that we hope will allow readministration, particularly in those patients that has received a suboptimal dose of the vector. Said that, we believe now that there are other aspects that make this therapy not working as well as they do in mouse models. Yeah. Yeah. Okay. The last question, because I don't. In the last question, these AAV vectors are episomal vectors. So if the cell divides, the genome is lost. So if you target on stem cells that we exactly don't know which are stem cells in the liver, the division of the stem cell will result in the elimination of the vector genome and the efficacy. However, the first patients treated with an AAV vector targeting the liver in hemophilia has been followed up for 13 years. And the expression of the factor 9 is still there, sustained levels. So it seems that in the liver, at least, the AAV can stay longer. So I hope I answered your question. And the first one was? Oh, yes, of course. We check the immune response, the T-cell immune response, both in mice and in patients also. And we have not observed a T-cell response against the transgene. And we have observed a T-cell response against the capsid in one of the patients treated in the cohort number 2, in the second dose cohort.
Thank you very much. So with this, we move to the coffee break. I want to thank all the speakers for these wonderful talks. And we are supposed to be back by 11:45, I guess. So thank you very much.
Sí. Vale, ara baixo el senyal, ¿vale? No, està igual? Ara pujo a tope. Igual? Vale, pues no és aquesta... No.
good coffee. It's time for some good science. Yes, welcome back. Perfect. So it's my pleasure to introduce the next speaker and also chair of this session, Dr. Joaquin Arribas. Let's make it properly now. Perfect. Welcome back, everyone. It's my pleasure to introduce the next speaker, Joaquin Arribas, who is a PhD in biology from Universitat Autónoma de Madrid, and then he moved for a postdoc at Memorial Sloan Kettering Cancer Center, studying growth factor processing, and he's currently director of research at Hospital del Mar and its research institute, and research professor at VHIO, where he leads the growth factors group. And today he will be talking about novel targets for immune redirection. Joaquin, the stage is yours.
Thank you very much, Martí. And I would like to start by thanking Fundación BBVA for funding this wonderful program. Of course, the organizers, Natalia, is there taking care of something, and the rest of the committee, and the rest of my colleagues at VHIO, where it's a real pleasure to work. So these are my disclosures, and I will be discussing basically three main projects. We are focused on trying to find novel tumor-associated antigens or tumor-specific antigen against which redirect lymphocytes via bispecific antibodies or CAR-T therapies. We are cell biologists, but our aim is to take these to the clinic, and we are learning how to do it the hard way. Hopefully, with these overlapping projects, what we are learning with the first one, we are applying to the next one, and instead of taking decades from the initial finding to enter into the clinic, we hope to spend only a few years to do it. Anyway, starting with the first project that started decades ago, I'm sure all of you know that approximately 4% of cancers are driven by HER2, amplification of the gene. When we hear HER2, we immediately think about breast cancer or gastric cancer, but in fact there's a huge number of other tumors at lower percentages that are driven by HER2. So this is data that is constantly being updated at the CVIO portal. So I'm going backwards, sorry about that. So we discovered and other groups many years ago that in these HER2 amplified tumors, in addition to the full-length receptor here depicted by this molecule, we could find in tumors truncated forms of this receptor, basically truncated forms that lack most of the extracellular domain, but are very active signaling. They are a very potent oncogene. They are not generated by alternative splicing. They are generated by alternative initiation of translation. That means that the mRNA that encodes full-length HER2 also encodes P95 HER2. So there's no way to detect the expression of the truncated form, but to use antibodies. So what we did was develop specific antibodies. This truncated form, this is difficult, too difficult for me. So this truncated form unveils an epitope that is completely hidden in the full-length molecule. So antibodies against this N-terminal epitope do not recognize the full-length receptor. So with these antibodies, we have been able to show that approximately in 40% of tumors driven by HER2, we can detect the presence of this truncated form. So in addition to the full-length receptor, these tumors express this fragment truncated form that we call P95 HER2. And this is true not only in breast and gastric cancer, but as you can see here, in a whole variety of different tumors. So what is the importance of this fragment? We believe that is a truly tumor-specific antigen. And as you know, there's only a handful of truly tumor-specific antigens that are not generated by somatic mutations. So, as you know, HER2 is overexpressed in these tumors, but is expressed at detectable levels in multiple healthy tissues. This is shown here with this immunohistochemistry on healthy human tissues, showing the expression of HER2, but we cannot detect the expression of P95 HER2 whatsoever, so making it a tumor-specific antigen. With this information, several years ago, we partnered with Roche to develop a bispecific antibody to redirect lymphocytes against P95 positive cells. They use their platform of these bispecific antibodies that have 3 antigen binding domains. 2 of them bind to the tumor-specific antigen or tumor-associated antigen, and 1 to the CD3 subunit of the T-cell receptor. So it recruits the T-cell against tumor cells. These antibodies were effective. These are classical analysis of the activity of bispecific antibodies using humanized mice. In these mice, we implant tumors from a patient-derived xenografts, and then we take white cells from these same patients, and we inject them into these mice. So before they develop graft versus host, you have a window to try and see if your antibody has tumor efficacy or not. And that is shown here. A tumor that expressed P95 HER2 could be controlled by these bispecific antibodies, while a tumor that did not express P95 HER2, these bispecific antibodies had no effect. So because of company reasons, and because this bispecific antibody competed with some of the pipeline of Roche, they decided not to develop it in the clinic, but we had at least shown that the redirection works. So we decided, with no experience whatsoever, to develop CAR-Ts several years ago. And we were very happy to show that the CAR-Ts were far more efficacious than the bispecific antibodies. These are mice with a cell line. This is a cell line, not a tumor's patient-derived xenograft. And as you can see, treating with the P95 CAR-T is extremely efficacious, and it really cures the mice. This is almost more than 1 year follow-up. And more importantly, these mice are protected from additional rechallenge, because if we implant again these tumor cells that express, these are cell line, as I said, that overexpress HER2, they do not develop tumors. So this is the kind of response that when you see, you think that you have something in your hands. However, when we used these P95 CAR-T cells in patient-derived xenografts, the efficacy was much lower. This is an experiment similar to that I have shown with bispecific antibodies. As you can see, there's a tumor response, but nothing as we had seen with a cell line. So we wanted to improve this. Once you have seen these kind of responses, you want to pursue them. So we aim to further arm these CAR-Ts with additional anti-tumor therapies. And inspired by the work of Marcela Maus, what we did is to add a second component. In this case, bispecific antibodies, BiTEs, against HER2. The trick here, because HER2 is not a tumor-specific antigen, was to modulate the affinity of these HER2 binding moiety. It had been described by the group of Carl June several years ago that introducing certain mutations decreases the affinity of this HER2 binding domain to HER2. And as the practical consequences of this is that these antibodies only bind to cells overexpressing HER2, and they do not bind whatsoever to cells. I'm sorry about this. I mean, this is the format has gone wrong, but sorry about that. They do not bind to cells expressing normal levels of HER2, and we tested that in many normal cells expressing physiological levels of HER2, and in fact, these bispecific antibodies do not bind whatsoever. So we are now attacking, so to speak, the tumor with P95 CAR-Ts and a bispecific antibody that binds only to overexpress HER2 and not to cells expressing normal levels of HER2. And sorry about that. This is just an in vitro assay showing that these cells that express P95 CAR-Ts and what we call TEC2. TEC2 is just a name for the HER2 BITE, but as you know, BITE is a registered word from Amgen and we cannot use it. So these cells, and sorry again because of the format. Anyway, we could show that in fact they are as efficacious and they are safe. These are mice in which we implant in one side cells expressing normal levels of HER2, the levels that you find in normal tissues, and in the other side, a patient-derived xenograft, one of those xenografts that did not respond completely to the P95 CAR-T. And as you can see in the results, these CAR-T cells expressing P95 and the bispecific antibody completely remove the tumor, the patient-derived xenograft, but do not have any effect on cells expressing normal levels of HER2. And we have repeated this experiment several times. So now, using these armed CARs, we went to a panel of PDXs, and now we saw the responses that we really liked with cell lines. And as you can see, in most of them, the response was complete, durable, it lasted months, and in many cases, you can consider these mice cured from these tumors. We have repeated this not only with breast cancer, all I have shown so far are breast cancer cell lines, but also with even pancreatic cancer cell lines, and patient-derived xenografts, and also with gastric cancer cell lines. So basically, we think that is not dependent on the cell of origin of the tumor. If the tumor expresses high levels of HER2 and or P95 HER2, they can be targeted by these CAR-Ts, and they can produce total responses. This is a mess, sorry about that. This is supposed to show the specificity and efficacy of these CAR-Ts. Those brown things that have gone wrong are the bispecific antibodies. They decorate the cell that produces the CAR-T, but also a part of them, approximately 20%, are secreted and can recruit T cells that have not been transduced. So they have kind of a bystander effect. And they are efficacious not only against cells that express HER2 and P95 HER2, but also to cells only overexpressing HER2. And we have shown that with the pancreatic cancer that I have just mentioned. So we published this 1 year ago, and we thought that with these preclinical results and also the funding that we got from the Spanish Association Against Cancer and Instituto Salud Carlos III, we could go ahead and immediately take this to the clinic in a clinical trial on patients with HER2 amplified tumors. Of course, we are cell biologists and we didn't know anything about regulatory steps and the production. Fortunately, we partnered with the BST, the Banco de Sangre y Tejidos, from here, Barcelona. They have co-developed the fabrication in GMP of this CAR-T. It has been difficult, but we have transferred the technology to be produced in GMP conditions, but we didn't know that regulatory events were going to be so difficult. So it's been 3 years of going back and forth with the Spanish regulatory agency. And finally, we think that we will get the approval before the end of this year. But we thought that 2 years in advance of publishing the preclinical data was going to be enough. It was not. It's a complete new field for scientists, and I recommend that you start as soon as possible with the regulatory issues that have nothing to do with the scientific issues that we are usually dealing with. One of them, and it's just to know your opinion in the coffee break, was that they are completely against retroviral vectors. As you can see, and you know there's in the field the consensus that lentiviral vectors are the future, but no one knows exactly why. They are self-inactivating, that's fine, and probably for stem cells therapy, that's very, very important. But this is data, real world data, showing, and this paper was mentioned in the first talk, showing the follow-up of patients treated with retroviral and lentiviral vectors. And out of 3,002 years, only one T-cell malignancy was developed, and it was from a lentiviral vector. So I think that as a field, we should reconsider why there's this consensus. Where does it come from? In addition to theoretical risks of the retroviral vectors. We chose retroviral vectors because we thought it was a safe possibility because they have been approved. We have used exactly the retroviral vectors that are being used to treat patients as we speak. But that was not enough, and it has taken almost 1 year to go back and forth with the Spanish regulatory agency. Anyway, that was a relief, I had to say it. so sorry about that. So anyway, we got yesterday news from the agency. I think that the trial is going to be approved pretty soon, and this is the Catherine trial that it will take place between Vall d'Hebron and Hospital del Mar, Barcelona. But the production will be done at the Banc de Sang i Teixits, and the treatment will be done at Vall d'Hebron. We hope to test this therapy pretty soon during next year. So what we have learned here, we hope is going to help us to expedite additional projects, and I'm going to tell you about another one that is quite advanced, that is the development of antibodies against a tumor-specific antigen in endometrial cancer. I mean, I have not disclosed what this receptor tyrosine kinase is. I mean, it's not secret, but the patent is ongoing, is the FGFR2, but it's not written. And there's a very, very prevalent mutation in the extracellular domain in FGFR2, curiously enough, in endometrial cancer. Almost 5% of patients express this mutation. And as you can see here, we have developed antibodies against a mutant peptide, a peptide that contains this mutation. And as you can see here, we have developed a whole bunch of monoclonal antibodies that bind to the mutant receptor, but not to the wild-type receptor. We truly believe that this is a tumor-specific antigen, and that is going to be very safe to redirect T cells by any means against these tumor-associated specific antigens. This is a Western blot just to show the quality of these antibodies. They are very, very specific. In the first panel, you have the commercial antibody against this receptor tyrosine kinase receptor, FGFR2. Of course, these two cell lines have been transfected with the receptor. And as you can see, two of these antibodies do not see the receptor, the wild-type receptor. They only detect the mutant receptor. This is also true by flow cytometry. Again, extremely specific. Sorry about the format. Extremely specific. And even if we increase the levels or the doses of the antibody, we don't see any signal in cells overexpressing the fullerene receptors. So using these very specific antibodies, we've already generated CAR-Ts with the SFBs of some of these antibodies. And again, we can show that cells expressing the wild-type receptor are not targeted at all by these antibodies in green and blue. They are targeted with antibodies against commercial antibodies against FGFR, as you can see, as expected. But look at the efficacy on cells expressing the mutant receptor. So again, we are facing a tumor-specific antigen, very specific. We have done already the humanization of these antibodies. We have sequenced the SFBs, the single chain fragment variables, and we have humanized them, the heavy and the light. We have tested different combinations, and the take-home message is that indeed the humanized antibodies are as efficacious in vitro as the murine antibodies. This is, again, the cell line that we use to test the specificity. These are HEC2 overexpressing the wild-type receptor of the mutant receptor, and as you can see, no effect of these CAR-Ts on cells expressing the wild-type receptors. Very effective expressing the mutant receptors, and the efficacy is similar in the case of the humanized receptor, the humanized antibody that produces the CAR or the murine antibody. And also importantly, these are cervical cancer cell lines, endometrial, cancer cell lines. As I have mentioned, this mutation is not so rare, it's 5%, so it's not difficult to find endometrial cancer cells that express these mutations, and this is data on cells expressing these mutations, not overexpressing them. And as you can see, our CAR-T, humanized CAR-T, is as effective, again, as the CAR-T derived from the monoclonal antibody. So we are hoping also to do the in vivo analysis, and actually it's true that this is gone much faster. We know all the steps that we have to follow now: antibodies, specificity, humanization, in vitros, and we are going to improve the in vivo assays just to try to expedite this as soon as possible to the clinic. We are also comparing, or will compare, the efficacy of bispecific antibodies versus CAR-Ts and so forth. So in the last 3 or 4 minutes, I'm going too fast. So in the last slide, I want to discuss what I think is the future. We have done a search of tumor-specific antibodies. We think that they are crucial to safely redirect lymphocytes against cancer cells. But as you can expect, there's a limited repertoire of tumor-specific antibodies expressed at the cell surface. So we really think that the future is trying to target peptides presented by MHCs. And then we will have all the repertoire of tumor-specific antigens that we can find. We can do that by identifying the TCR that binds these complexes, and Elena, I'm sure that she will do it in the near future. But we are specialized in antibodies, so we are trying to develop antibodies against these complexes. So this is the pathway that you all are familiar to. I mean, basically, at any time point, the cell line expresses at the cell surface a representation of everything that the cell line is expressed. And that, as I said, it's theoretically a much larger repertoire of tumor-specific antigens. So in collaboration with Elena, as you know, her laboratory is doing immunopeptidomics. We have identified a given protein that is, sorry, I cannot disclose it, because again, we are filing a patent, a protein that is expressed by an haplotype of MHC that is quite common in the Caucasian population, approximately in 20%. So she was able, and her group, she was able to determine the sequence of these peptides presented on this protein that is upregulated in many tumors, massively upregulated in many tumors. And then we have followed this protocol. We, in collaboration with the company, of course, we have generating recombinant proteins, the MHC with the beta-2 microglobulin in the presence or in the absence of the peptide. So we recover the MHC bound to something else or empty, or MHC coupled to this peptide of interest. With that, they humanized rabbits, and they extracted splenocytes of these rabbits. These splenocytes are much enriched in antibodies against this MHC coupled with the peptide. And with that, they develop, or they produce a library of SFBs with all the combinations. with many different combinations. And they started doing panning with in phage displays of phages that recognize that MHC haplotype bound to the peptide, but not to the MHC bound to something else. And after many of these passages, we have been able to take many different candidates. As you can see here, these are ELISAs control in gray, or against the HLA bound to something else in blue, or at the HLA bound to our protein of interest. As I said, this is a protein that is overexpressed in some tumors at very high levels. And as you can see, the antibodies are quite specific, binding that haplotype bound to the peptide, but not the haplotype empty or bound to something else. And I want to call your attention to the fact that that is a logarithmic scale, and in a linear scale, these antibodies against these proteins are quite specific. So basically, we want to follow the same steps that we have followed. We are trying to confirm this, developing antibodies against additional MHC peptide complexes to follow this pipeline that we have developed in the lab to take it to the clinic. So with that, this is the summary of my talk. I hope I convince you that P95HER2 is a tumor-specific antigen, and that these CAR-Ts that we have developed that express a CAR-T against P95 and secrete a bispecific antibody that binds only to overexpressed HER2 induce safe and durable responses in different tumor models. And that soon, after the regulatory ordeal, this clinical trial, Catherine, is going to take place hopefully next year at Vall d'Hebron, directed by clinicians at Vall d'Hebron, Irene and Vladi. Based on this experience that we have accumulated in a project that has taken decades, we are trying to expedite development of CAR-Ts targeting additional tumor-specific antigens. We hope that the antibody that I have shown that recognizes FGFR2 very specifically, this mutation, could be taken to the clinic, and we are also developing other uses of this antibody. And finally, once we have done this with cells expressed at the cell surface, we are trying to initiate the development of CAR-Ts or other means to target cancer cells by targeting MHC loaded with some peptides that should be specific or are specific of tumor cells. With that, I have to acknowledge, of course, the members of the group. The P95, the leader of the P95 project is Macarena Roman, is here, and she's in the audience. And no, sorry about that. Here, oh, Jesus, here, there she is. And Jade Liu, sorry, Jade. She is the leader of the FGFR2 project that I have just shown. And Marta Bort, that is also here, is developing the MHC peptide antibody. So if you want to talk to them, also Pau, I have not forgotten about you. You are also working on this kind of model. The four of them are here, and they are very talented, so if you have any questions, I'm sure that they will be happy to answer them. And of course, a big thanks to the funding agencies, particularly Spanish Association Against Cancer, not because Marta is in the audience, but because they were the first ones that really trusted this project. They funded it in 2018 when we had not produced these results, and they were keen on funding this clinical trial that will start soon, and also the Instituto de Salud Carlos III that is also funded this trial. Of course, also a big thanks to the Breast Cancer Research Foundation from the United States that has been funded my lab for 20 years, and without them, we could not make all this. So thank you very much, and happy to take any questions.
there's time for one or two questions in the audience.
Nope.
Yeah, I think there's one.
Okay.
I will ask one.
Thank you very much, Joaquin. It was an extensive journey, very intensive, and congratulations also for getting the comments from the last comment from the agency. You were discussing about this point of the retroviral and the lentiviral, but in fact, there are also some points that I think that are interesting for developers. That is the interest. I mean, the high demanding requests from the agency regarding the quality controls and in the CMC production. So I think that this is also something that maybe you could comment a little bit, because this is something that could be challenging for many projects, and I think that it's important to be aware of this situation that is at this moment.
We have found many steps in which we were wrong, but I mean, it's difficult to predict, in my point of view, what the agencies are going to say. Sometimes they say, you cannot use ampicillin-resistant vectors. What do you mean? You have just approved an ampicillin-resistant vector nearby. Well, no. This time you have to use kanamycin. So I don't know if that answers your question. We have many steps that we should have asked way before, and many of them are unpredictable. And I think that it should be pretty clear that ampicillin either is a problem or is not. So why don't you tell everyone, don't even start? Instead of saying, well, go through the whole process and then we will decide if ampicillin can be used or not. I don't know, you are the expert in regulatory affairs, but it's very difficult for us to fight.
No, in fact, this is something that we are discussing in other projects, and I think that it's important to take the experience of the things that are happening. But one thing that we were discussing in the projects in which we are trying to help modify the regulatory environment is that, okay, it's not the same the first trials that the final trials. So you cannot put the requests in the starting trials the same that in the final trials. But this is something that we have to be aware, because somehow it's important to be conscious that this is happening, and to try to move the field forward towards a simplified environment in which these products can move forward. I was surprised to see that the previous person who was talking about in vivo, he said that he went to Australia. Yeah. We should ask why they went to Australia. So, and this is something that we should take into account to discuss how the future should go.
Definitely.
So really nice talk, actually. So I have a more mechanistic question, actually. So do you have any information about the affinity of the antibodies that you are producing against the peptide MHC complex? Because my question is, in general, interaction between the TCR and this complex is a low affinity interaction. Probably your antibodies are going to be high affinity interaction. Are you expecting any potential problem in this direction?
No, I mean, it's a very good question, but for that we need a cellular model that expresses that haplotype loaded with this peptide, and we have not found that yet to really address what is the affinity in a normal, in a physiological environment. Definitely that is something we want to do. We have started with a protein that is, as I said, is massively expressed on some tumors, and we will see. As you have mentioned, affinity of the TCR is low. I don't know how an antibody, an SFB, is going to work there. This has to be answered.
Okay, thank you.
Joaquin, great talk. I have a question regarding the durability of the persistence that you observed. So you inject CAR T cells and then you re-inject tumor, and tumor didn't grow. So is it because the CAR T re-expansion, or is endogenous immune system somehow got triggered?
Actually, we were very surprised by that result because, first of all, we expected graft versus host. These are human lymphocytes. So usually in the first experiment that I showed, when not CAR T, but T cells are injected, you have a window of 1 or 2 months before the mouse dies, because the lymphocyte, being human, recognizes every tissue of the mouse as foreign. With CAR Ts, that is not the case, as I have shown. Why they are so persistent? It may have to do with the less favorable answer would be this is not really physiologic. I mean, you are putting a human T cell into mice, and that is inducing an activation of the T cell that is enough to keep it alive for longer periods of times. Or is the CAR T? We don't know, but we were surprised to see that, and we don't have an answer why those cells that had been detected 1 year after the tumor had regressed persisted for so long. I hope I have an answer, but I don't. Thank you.
So we will move on to our next speaker, Dr. Roberto Chiale. He's professor of pathology at Boston Children's Hospital, Harvard Medical School, and the University of Torino. And he leads pioneering research on LAK-driven cancers, and he will present a portfolio of cellular immunotherapies against these cancers.
Thank you very much for the introduction. Thank you for the invitation. The meeting is great, both for speakers and the venue, which is amazing. First time here. It's a beautiful place. We are basically connecting well with Joaquin's presentation because he introduced several concepts about tyrosine kinases in cancer, the difficulties of finding a specific target for solid tumor, which is still a challenge for the field, right? And so we are presenting our program that has been running for almost a decade now, in which we focus basically on one particular type of group of cancer, actually, that are driven by this specific oncogene, which is called ALK, for anaplastic lymphoma kinase. And ALK was discovered, as the name stands for, in a type of lymphoma, anaplastic lymphoma, large anaplastic lymphoma, which is there second on the left. But then eventually it was more important in discovery in non-small cell lung cancer, and so far is the most frequent rearrangement found in non-small cell lung cancer, which still accounts only for 5 to 6% of lung cancers. However, there are different flavors by which ALK can be an oncogene in tumors. Some tumors express the full-length receptor. ALK is a physiological and transmembrane RTK. So, for example, neuroblastoma is the prototype of a subset of tumor that express the full-length receptor, thyroid cancer, or they simply can overexpress, for example, glioblastoma, melanoma, and another type of solid tumor, including rhabdomyosarcoma and pediatric tumors. So we have a complexity of ALK-driven cancer by which this oncogene can be found either as a fusion or as a receptor, and so makes amenable for different possibility for targeted immunotherapy. Key points of this oncogene are summarized in these slides. I mean, of course, we develop all this research through the year, as you can see. But basically, to summarize why we decided to develop this program. First of all, because ALK is an oncogene, so it's bona fide the driver of all this tumor. So these are several important implications also for cancer immunology and also not only for cancer therapy. But first of all, 100% of the cells express this oncogene in any ALK-driven tumor. So it's not like other type of antigens that we have been seeing so far that subset. I mean, we discussed early this morning about the PRAME TCR, very good. However, PRAME can be expressed between 60 to 80% depending on the cancer. So there is always a fraction of tumor cells that do not express the antigen. It's not the case for ALK because 100% the very first initiating cells has an ALK-driven alteration. As a consequence, tumor cells are selectively highly expressing this ALK antigen either via fusion or via the transmembrane receptor. and they show very strong addiction to the tumor, and that is clearly testified by the fact that now there are 6 different targeted therapy ALK TKIs that are approved worldwide to specifically target ALK, starting from crizotinib, first generation, to alectinib, ceritinib, brigatinib, second generation, lorlatinib, third generation, and entrectinib, and now possibly there will be Nuvalent, neladalkib, probably approved maybe next year. So it's a very important target for cancer therapy, and patients experience durable responses. And so because we have this advantage of having a molecule that can keep the tumor at bay for a long time, there is a window also to manipulate the immune system of patients to step in with the next level of immunotherapy. Then, of course, the restriction of ALK is selectively restricted to ALK-positive tumors. We, as a pathologist group, we did an extensive search of ALK expression in any normal tissue. It's basically not expressed, with the exception of few enteric neurons. Is extremely low expression, eventually in some subset of the brain, but not detectable the protein, just the mRNA transcript. So the protein expression is extremely low. ALK-positive tumors tend to have a very MHC class I expression. There are a lot of publications showing that patients that develop an ALK-positive tumor, they immediately react to the presence of the ALK protein in the tumor spontaneously. So they develop anti-ALK antibody, they develop anti-ALK CD4 and CD8 T cells, right? And that's very important because it implies that the protein is immunogenic per se, naturally immunogenic per se, when it's expressed by a tumor. And so we basically decided that we needed to approach this complexity or genetic complexity of a driven tumor by multiple approaches, right? So if a tumor, like in the tumor in the top left, has a fusion, which is typically for lung cancer and lymphoma, the fusion is only cytoplasmic because the break point of the fusion is exactly in the first amino acid for which the cytoplasmic domain of ALK starts, right? So the protein is entirely inside the cell. So in this case, we need to rely on techniques like a vaccine or TCR T cells that basically can target peptides presented by the ALK protein, either in MHC class I or MHC class II. And of course, we have discovered all this process. We developed an ALK vaccine. We discovered all this peptide by mass spectrometry. I will show few data. And very recently, which I will show later, we also discovered a T cell receptor and generated T cell receptor T cells that target ALK. For the other type of tumors, neuroblastoma, rhabdomyosarcoma, Ewing sarcoma, glioblastoma, etc., that express the full-length receptor, now ALK is on the surface, and so it can be targeted by antibody, and from antibody you can generate ADC conjugated. We have not done that, but other groups, for example, Yael Mosse at Philadelphia has done that. And what we did below is to develop CAR T cells against ALK. So today I'm going to show basically parts of this portfolio. What we call is a portfolio because we want to target ALK tumors by multiple approaches, not by one. And what we think is the beauty of this is that these approaches can be combined. So for example, a vaccine for ALK can be used, of course, to vaccinate patients, and we are going to test that in a phase 1 clinical trial supported in the US by the Breakthrough Cancer Foundation that will start at the end of next year. I'm not going to talk today about the ALK vaccine, which is an mRNA ALK vaccine base for lung cancer specifically, so today is not part of my talk. But today I'm going to focus on CAR T and TCR T cells, right? And so I was saying that this therapy can be combined. We already combined CAR T with TCR T. We combine CAR T with a vaccine, and we are going to combine also TCR T with a vaccine, right? And there are rationale behind this possible combination. Of course, starting from the point of the highly specificity of this therapy targeting the oncogenic driver, right? And so we published recently the part of the CAR T. The TCR T is still unpublished, so I'm going to navigate on these two aspects. So CAR T was applied for pediatric neuroblastoma, which is the most common extracranial solid tumor in the childhood and is the most deadly tumor, accounts for about 15% of pediatric cancer death, is less frequent than leukemia, but more deadly, right? And a patient with high-risk feature, they tend to relapse and basically die in about 50% of them. So there is no really space for a good therapy for them. And ALK is relevant for neuroblastoma because basically almost all neuroblastoma express ALK, 90% express homogeneously detectable on the surface. ALK can be a driver in neuroblastoma, can be mutated, point mutation that can be targeted by ALK inhibitors, as we said, can be amplified. And so basically, there are also evidence that ALK TKI, like lorlatinib to generation, also work quite well, specifically in the mutated subgroup, again highlighting the driver role of ALK in this particular tumor. So we developed a CAR-T because of this wide expression of ALK. You see that by mRNA and by surface expression, ALK is recognized or expressed, detected basically in 100% of neuroblastoma, and very interesting also in subset of rhabdomyosarcoma, alveolar rhabdomyosarcoma in particular. We are currently testing them. Being a pathologist, of course, we rely on a very specific staining. This staining is FDA approved because there is an antibody that is FDA approved as a companion test for ALK-driven diseases, to a point that when you see a positivity in a tumor, you can immediately start therapy with an ALK TKI without even showing a rearrangement of fusion or the molecular oncogenic driver because of the specificity of this antibody. And so you see the strong expression in most of the neuroblastoma. These are cell lines, PDXs, and on the right there are primary tumors. And this is the specificity. So by tissue microarray, basically all the important tissue in the human body, by bulk RNA sequencing, single cell RNA sequencing, you see that basically ALK expression is not detected. And even the tissues in panel B that show some some positivity here, like the brain here in yellow, when you actually do immunohistochemistry, there is basically nothing. See the tumor, how it is, right? So the tumor is, this is a neuroblastoma, this is a lung cancer, and see the difference of expression between tumor and the putatively positive normal tissues in the human body. So there is a big gap of differential expression. So we developed 7 different CAR constructs. We tested in preclinical model, and our comparator is GD2 CAR. GD2 CAR are so far the best CAR for solid tumor developed. You know, the GD2 CAR have a big hype, not only in neuroblastoma, but also in other tumors like midline glioma in pediatric population. There have been multiple high-profile publication by the Stanford group about this. So GD2 CAR are considered probably the best, one of the best CAR for solid tumor, and this is our comparator, right? So golden standard. So this is an example of a neuroblastoma that expresses both ALK and GD2, and you see that the ALK CAR, they perform as well as GD2, with the difference that the ALK CAR has a much better safety profile because you know about the toxicity of the GD2 CAR. And because of this very specific staining, so the antibody only recognizes the ALK-positive cells that are not present in the human body and not present, of course, in the mouse body, we can detect the persistence of tumor cells by even single cells. So you can detect single positive tumor cells. And so we can conclude that these mice treated with an ALK CAR or GD2 CAR basically are completely cured because we could not detect one single residual ALK-positive tumor cells in any tissue, right? So, of course, the difference when you use a model expressing the low end, I mean, if you remember the image that I show on primary tumors, there are majority of tumors express good amount of ALK, but of course there are some tumors that still express ALK, but on the low end, the activity of this CAR is obviously less efficient. However, we discovered a virtuoso circuito, we call it, by which adding an ALK TKI to this type of tumor, we create a double sort of effects. On one side, the ALK TKI, of course, for the intrinsic ability of blocking this oncogenic signaling, slows down tumor growth. But it does another thing. By blocking ALK TKI signaling, basically it creates a feedback loop of compensatory reactivation of the ALK promoter. So basically the tumor cells responds to the blockade of the T-cell signaling by increasing the level of receptor, not only increasing the overall amount you see by Western blot, but also increasing the surface stabilization of the receptor. That can be seen in vitro, can be seen in vivo. So, and this is perfect for CAR-T because using an ALK TKI, now the ALK level on the surface of the tumor cells is clearly increased and more stable, right? And so by combining an ALK TKI with CAR-T, so a drug specific for the receptor and CAR, now we can see that even in tumors expressing very low level of ALK, we can dramatically extend the survival in mice. We cannot cure completely the mice as we do in tumors expressing high ALK, but still is a very significant advantage. So based on this, we started a phase 1/2 clinical trial. And Joachim, this is a retroviral construct. There was no problem with the FDA. We got the approval in 28 days, so they had no question about retrovirus. And so the trial was opened this spring, 2025, at Dana-Farber Cancer Center and Boston Children's Hospital. The clinical PI is Susanne Baumeister. This is the schema. I'm not going too much into that, but of course there is cytodepletion, CAR-T infusion. The FDA recommended, because this is a first-in-human anti-ALK therapy, cellular immunotherapy. I mean, of course, ALK inhibitors are approved, but cellular immunotherapy is the first-in-human. FDA recommended to start with ALK-CAR alone in a classical 3x3x3 escalating phase, and then to combine with lorlatinib, which is our real goal, to combine with lorlatinib only after safety and be approved. We already enrolled 2 patients. We are enrolling the patient number 3 next week. And of course, it's still preliminary, but I can tell that they're very, very safe as expected. No toxicity against normal tissues, which is always a challenge for CAR-T targeting solid tumors. Primary objectives will be, of course, the identification of MTD, and then, of course, we will see for some responses, persistence of the CAR, and etc. And this is the team basically that led to this part of the project. Everything has been done by my lab in Boston in this case, and of course the clinical trial is run at the Dana-Farber with the CMCF facility led by Jerry Ritz over there. In the last 6-7 minutes, I'm going to talk about the second part, which for us is very, very exciting also, which is the development of a TCR T-cell therapy for the other portion of the ALK tumor, which do not have ALK on the surface, but they have ALK internal in the cytoplasm, right? So we needed to identify basically, and that's already published in the paper that I showed previously, we needed to identify by mass spectrometry the peptides of ALK that are processed by the immunoproteasome and display on the surface of the tumor cells, right? And we identified several of them. They are all published, including this peptide, RPR. Using this peptide, we not only generated vaccine that, as I said, is not the topic of today, but is entering separately another clinical trial, but we were also able to start cloning TCRs, right? Of course, there are typical 2 options. Most of the TCR, including the PRAME TCR that was discussed early this morning, have been cloned by donor human T cells. So basically incubating the peptide with donor cells and then eventually single-cell TCR sequencing the expanded T cells from donors, right? Which is okay. However, it's a very artificial system. You end up having a lot of TCR with low affinity because you basically pump the culture with very, very high concentration of your peptide, right? And we know that TCR depends on the specificity and the right amount of peptide presented in class 1. So we decided to go in another way, prompted by collaboration with the NCI group that has done that before already with KRAS TCR. And so basically cloning TCR from vaccinated mice that have, of course, a human HLA with a human peptide. So this basically recreates in vivo a condition that is more physiologic for the maturation and expansion of the clones. So we vaccinated the HLA B0702 mice in this case because this peptide is presented in that HLA. And of course, we clone TCR by single-cell TCR sequencing. And we end up, of course, with about 50, 60 of them. We ranked them. We validated the top ones. And we end up with these 2, which we call number 1 and number 2, that has some great properties for us. So first of all, as you see, they can transduce T cells very efficiently. Typically, we get more than 85 to 90% of transduction of T cells. But not only that, they properly mispair spontaneously. This is not a given. So for example, again, the PRAME TCR that have been discussed this morning, they needed to be engineered to modify some amino acid to increase the affinity between the alpha and the beta TCR chains, right? Because otherwise you incur in problems, which is mispairing of this chain with the endogenous alpha beta carried by the donor cells, right? These two TCR do not have, with other we had that problems, okay, but these two TCR do not have this problem. They spontaneously mispair properly well. And amazing to us, you can see here on the left, sorry if I can use, okay. Here you can see the endogenous alpha beta chains, okay. When you introduce the exogenous alpha beta, you see that the endogenous basically disappear. So there is a disassemble by competition of the endogenous donor alpha beta T cells and favoring the exogenous that we introduce, right. This is a big advantage because we basically eliminates mispairing and basically focus all the TCR receptor signaling through the engineered TCR. And so we show that these TCR are very potent and very specific. Specificity can be shown in two ways: cells that are B7 positive versus B7 negative, and of course cells that are ALK positive versus ALK negative. And you see here that only cells that are both ALK and B7, they are killed basically 100% by both TCR. In contrast, if tumor cells, this is endogenous presentation, so it's not exogenous peptide. This is what the cells naturally present, right? So if the cells either lack B7, so ALK positive tumor but B7 negative, or ALK negative tumor but B7 positive, there is zero recognition by this TCR, so very highly specific. And in vivo, they work extremely well also. So here is monotherapy in a lung cancer. This is refractory to ALK TKI. So we injected, and you see all the control, which are empty vector, other type of TCR that they don't work. And of course, you see both ALK-specific TCR inducing a profound response in mice that lasts for very long. The survival advantage is, I think, quite good. And eventually, tumors relapse, and we have some explanation, but I do not have time to discuss now. Here is another example. This is neuroendocrine tumor that are ALK positive. Now with the transmembrane receptor, and the transmembrane receptor can still present the same peptide, right? So that's the advantage, and that's why we can combine CAR-T and TCR-T, right? So neuroblastoma on the top, you can see they can be killed 100%. Merkel cell carcinoma, typically ALK positive, they can be killed 100%. And in vivo, they also show very high specificity and potency. So this is an example of neuroblastoma cells that are either B7 positive or B7 negative on the right. And you see that only when you have both the B7 positive and ALK, and not when you lack B7, right, the two TCR that are very potent, and they can basically eradicate disease. Now the follow-up is much longer, and basically the mice are all alive, and we did histology. There are, again, no tumor cells left, so it's really achieving cured. Okay, I'm concluding here, and this is the group done, most of the research done in Boston with the help of Dana-Farber, MIT, Cell-Based Therapies, who provided the antibody for the CAR construct, and of course, Gianpietro Dotti and the clinical team at Dana-Farber. Thank you very much.
Thank you very much, Roberto. And it's time for one or two questions. Or maybe.
The displacement of the TCR, the exogenous TCR has to do only with levels of expression, so you need a high level of expression?
No, it's not level of expression, because we have the other 40 TCR that we cloned that are supposedly specific, despite we express at the same level, and we can tell because we have an antibody for the mouse beta chains, right, that's recognized. They express exactly at the same level, but they basically don't pair this way. They pair much less efficiently. They don't displace the endogenous. It's a property of these two TCR. Probably the pairing of the alpha beta, the complementary region is so strong that is prevalent and dominant over the endogenous one.
And very rapidly, and how about that relapse after those wonderful responses?
Yeah. Yes, I know you were asking. So the tumors are still ALK positive, obviously, because it's a driver. They cannot lose ALK. That has been demonstrated in thousands of patients, that all the patients that become resistant to ALK-TKI, they are still 100% ALK positive, right? So they don't lose ALK, right? They become resistant by other means. They did not lose MHC, which obviously was the case. So they are still MHC positive. They can still present the peptide. So basically, when we explanted those tumors ex vivo, directly in vitro, the TCR were able to kill them again 100%, right? So probably we think it's just a matter of exhaustion of these T cells over this very extensive duration, right? So we are now testing a second injection, like for example Carlos is doing in multiple myeloma patients. So if it's a matter of having another wave of activated and good shaped T cells, we should push the tumor back.
Congratulations, really elegant work. I have two questions. One, in the CAR T trial, I noticed that the phase 1 does not include any cytokine support after the infusion. At least it wasn't on your design. Was that on purpose or?
Yes, because, I mean, the FDA was very cautious. I mean, ALK is putatively a receptor that could be expressed in some normal tissue. So they ask a lot of questions about the expression in healthy tissues, and we provided what they, at the end, were satisfied. So we decided they wanted actually to test really the plain construct first, and then start the combination with lorlatinib, and we are already working on second generation of CAR T with some hardware at introducing interleukin-15, etc. Right? So it's just because it's a first in human, so we need to have safety first, right?
My second question is about the ALK TCR. It looks very, very hot, and you had two. You used the humanized mouse model to isolate your TCR, and you got a B7 restricted. Now, could you remind me the percentage of patients with
20%.
20%. And was that the only restriction element that you isolated TCRs for?
No. We have clone also now for A0201, which is another 40%, as you know, right? So we already cloned them. And we have other candidates, but it's very, very early, right? And by the way, very interesting, this TCR that recognized the B0702, they also work very well against B0705, which adds another 3-4%.
We found the same thing, that they can be a little promiscuous with the B and C alleles. And then the last thing, for the TCR that you did isolate, I didn't see any peptide dilutions. How high affinity a receptor is it?
It's similar to the PRAME TCR.
Yeah.
Same scale. So
and you had no
affinity. Nanomolar, right?
And you didn't change the affinity. You didn't change any of the.
They are untouched.
Yeah.
So there is room for improvement.
Right.
But as you know, there are examples that you start to manipulate and you get off-target activity.
Exactly. It's
right?
a dangerous business.
It's a dangerous business, right? It's a difficult and dangerous business. So these are untouched.
Great work.
Yeah. I'm afraid that due to time reasons we have to move on, but I'm quite sure that Roberto, you can reach him in the break,
Yeah,
right?
sure. I will be around.
Fantastic.
Thank you very much.
Great. So now it's time to move to short talks session. The first one would be Daniel Gomez, a PhD candidate at CNB CSIC. And he will be talking about development of new cellular immunotherapies against cancer based on T lymphocytes trained with bacteria.
Okay, so thank you very much. And historically, the ways to treat cancer have demonstrated to be so inefficacy. So we have embraced immunotherapies, such as, for example, immune checkpoints inhibitors and also the cell-based therapies. However, these cell-based therapies usually relies on chemotherapy for lymphodepletion, the administration of high doses of interleukin-2, and also high numbers of T cells, just to work properly. However, this usually leads to severe secondary effects and also a poor response in solid tumors. They also face other challenges, such as tumor-induced exhaustion, low persistence, and poor trafficking and infiltration to the tumor itself. So focusing on those other challenges, concretely, and the tumor-induced exhaustion, it is well known that chronic antigen stimulation usually leads to an increase of exhaustion markers and metabolic dysfunction, as well as a decrease of effector function. And this is all led by a transcription factor known as TOX. I want also to focus on the severe secondary effects, which reduce the quality of life of the patient. Also, in some cases, they risk their life. For example, with cytokine release syndrome and other nervous system problems. So our laboratory here proposed a new technology, a new next generation immunotherapy, known as TBUS technology. How it is done? Well, first of all, we genetically modified the bacteria to express tumoral antigens. Then those bacteria are given to dendritic cells. Those dendritic cells engulf the bacteria, and when co-cultured with CD4 T cells, those CD4 T cells are able to capture the bacteria from the dendritic cell and process them. From now on, we are going to call those CD4 T cells back T cells. And I show you here a video of the process. So those back T cells then have the ability to act as antigen-presenting cells, stimulating in an antigen-dependent manner CD8 T cells, which from now on I'm going to call TBUS cells. And those TBUS cells show a central memory-like profile and also express low levels of PD-1. So here I want to present you an experiment in which we use two different bacteria, Listeria monocytogenes and engineered E. coli. And for this experiment, we used CD8 T cells, which come from OT1 mice. That means that all the CD8 T cells recognize OVA via their TCR. And want to compare the therapy, the TBUS therapy, to the classical activation therapy, which use BITs anti-CD3 and CD28. So we check their cytotoxicity ability at different ratios against melanoma B16F10 OVA cells. And what we could check is that those CD8 effector T cells activated by Listeria and also by the engineered E. coli kill tumor cells more efficiently than the CD8 effector cells activated with the classical activation with the BITs, while expressing less PD-1. So then we wanted to perform a bulk RNA-seq of those CD8 T cells, and we obtained some astonishing results. First of all, regarding to the reflection phenotype, we could see that those cells were indeed more effector, expressing great doses of, for example, Recinte and perforin. The second thing that we checked was their exhaustion profile, and they were less exhaustion, and I want to highlight that they expressed a low tox. And regarding to their metabolic state, we could find that they depend less on glucose metabolites, while depending more on the mitochondrial metabolites, which is key in order to properly infiltrate and kill tumors inside those harsh conditions of the tumor microenvironment. So, as a conclusion of this first part, we can say that BACT activation yields to a unique transcriptomic CD8 T cell state that we call TBUS, corresponding to an enhanced effector function and a metabolic fitness with reduced potential exhaustion. And this state consists of cells which are more persistent, non-exhausted, metabolically fit, central memory-like, and also effector. So I want to show you here two different videos of TBUS cells trained, reprogrammed with Listeria or with E. coli, killing tumor cells expressing OVA. And this was all captured by a two-photon technique. So now that we know that the therapy could work, we want to move on to set up the conditions for doing it. So we designed an in vivo experiment in which we inoculate B16, F10 ova cells, melanoma cells, to mice, and after that we gave them several doses. After measuring them and processing the tumor after 1 month, what we were able to see is that at our condition, the tumor size was significantly lower compared to the other conditions. And also, when focusing on the CD8 T-cell profile, we could find that, that there was a decrease in the green, an increase on the green and pink population, which are the persistent and effector ones, while there was a decrease on the red and blue populations, which are the exhausted ones. So then we also perform a single-cell RNA-seq of the infiltrate of those tumors, and what we were able to see was that there was an increase of the CD8 cytotoxic cells, and also there was a decrease on the immunoregulatory cells, which are associated with bad prognosis of the tumor treatment, such as, for example, the T regulatory cells and also the tumor-associated macrophages and neutrophils. So finally, we also checked the clonality, the repertory of those CD8 T cells, and what we were able to see was that in the case of our therapy, the TBS therapy, there was an increase up to 3 times more expression of the TCR, which recognized OVA inside the tumor infiltrate. So I'm leaving here like a proposal of how the therapy would be done, remarking that it won't need the chemotherapy for lymphodepletion, neither the high interleukin-2 doses, and neither the high T-cell numbers. So finally, I want to acknowledge all my lab companions alongside my PI, Esteban Veiga, as well as the funding members like the Ferro Foundation and the Ministry of Science. And that's all. Thank you.
Thank you very much. Daniel, if there's any question in the audience? It's open for questions. I think there's one there.
Okay, thank you for the talk. Very interesting topic. My question, this is all done in the OT1 system. Is how you tested it in other systems, or are you planning to translate this technology to other modalities of T-cell therapies?
Okay, we are indeed working with tumor antigens itself. That we have a poster number 8, if you want to check it. And we are also trying to move towards the CAR-T therapy, because we are designing bacteria to express viral antigens, which we are all vaccinated for, just to have a large number of those cells as a basis for the CAR introduction.
So we will move to our next speaker, Rémi Perteman, postdoc at Universitat de Lausanne, and he will be presenting identification of clinically relevant T-cell receptors for personalized T-cell therapy using combinatorial algorithms. Rémi?
Good morning, everybody. Buon dia tutto. So I'm very happy to be here and to present today the work that I did actually for my PhD, which was how to combine different algorithms to try to identify very precisely clinically relevant TCRs for TCR therapy. So I want to start with a soft introduction on TIL-ACT. Oops, there's no title? Well, no. So the title says adoptive cell transfer of TILs, where, as you know, TILs are expanded in vitro and reinfused to the patient post-chemotherapy. And in the past year, it has demonstrated a good clinical efficacy, especially in tumors with high mutational burden, like metastatic melanoma. But despite these promises, it still only works in 40% of the patient, which shows that there are still some limitations to tackle. So back at the CHUV, sorry for this. The title is very small. Okay, so back at the CHUV, a few years ago, we had the opportunity to work on a phase 1 clinical trial with 13 metastatic melanoma patients treated with TIL-ACT. And what we saw is that 6 of the patients responded and 7 did not. And in my lab, we were very interested in trying to understand what are the factors associated with this difference in clinical response. And what we found is that the answer is in the TILs. So not all of the TILs inside the tumor are tumor-specific or tumor-active. Some are merely bystanders, and their number really matters. If we look at the responder patients, we see that prior treatment, they have a higher number of tumor-active TILs that can be expanded and treat the tumor. And this is not what we see in non-responder patients. So now we're faced with basically 2 types of patient. One that is very rich in tumor-active TILs that could actually benefit from TILs. And so in my lab, we've been trying to optimize the tumor-active TILs mobilization for this. And for the other types of patients, the non-responders, those that are poor in tumor-active TILs, we're proposing an orthogonal approach, which would be to use personalized TCR therapy. So this would work by modifying autologous cells with TCR to create a product that is highly tumor-specific to treat the patient. And of course, the challenge is to identify those tumor-reactive TCRs, and this has been the project of my PhD, which worked a bit like this, where we have a tumor biopsy and we identify from different algorithms the tumor-specific TCRs for this therapy. So of course, and the TCRs would be private and orphan. And of course, we're not the only ones interested in this in the field. Many have found that tumor-specific TILs are transcriptionally different from the bystanders, and others have even developed predictors for tumor-active TILs. But what we think is we don't think that all of the TILs are equal. We think that some might be more important than others, and we also believe that there might be some redundancy in the TILs. So here I'm showing this approach in which we combined 3 different algorithms to really specifically identify those clinically relevant TCRs. So the first axe is the tumor activity, and I'm going to show you in this presentation how we've developed TRT-PRED, our own predictor. The second axe is the structural avidity. We found in the past that highly structural avidity clones preferentially resides inside the tumor and can control tumor growth. So we want to use a predictor of structural avidity developed by Marta Perez here for our selection. And finally, we want to use a TCR clustering tool called TCR-PC-DIST to cluster together TCRs that have similar physical-chemical properties. That way we can select TCRs from different clusters and try to maximize this TCR diversity to target different parts of the tumor. And we call this approach mixed TRT-PRED. So to build TRT-PRED, first we used a single-cell RNA and TCR sequencing-based approach where we could predict the cell-wise tumor reactivity to know which TCR are tumor-active or not. And for these types of model, we need 3 specific ingredients, the input data, and for this we've used a database of 235 clones that have been tested for tumor activity from 10 metastatic melanoma patients. We've designed a few statistical models and evaluated those models inside a very thorough framework, which consisted of leave one patient out nested cross-validation. And the results look a bit like this, where we have the generalization performance of the 21 model, and we can see that the best model is this EDGE-R-QFL model that discriminates very well between the reactive and non-reactive clones. We can also see in the signature that it's associated with exhaustion markers, which is coherent with what we see in the field, and so we believe that we've selected a coherent model. Then we also wanted to challenge our model on external data. So we've challenged it on different cohorts, and I'm showing 4 here, which is cohorts from melanoma, lung, a pan-cancer cohort that is also mostly GI, and also a GI cohort. And what we see is that TRT-PRED performs quite well on these external cohorts, and that it performs even better than existing models. So now that we have a model that works, we wanted to combine it with the other predictor for this mixed TRT-PRED approach. So we've designed this clinical demonstration in vivo in which mice were engrafted with a tumor cell line and then were treated with a TCR therapy that was built from TCRs identified from mixed TRT-PRED. So it looked something a bit like this, where we had this patient that was not used for training. We identify the tumor-active and high structural avidity clones, and then we clustered those clones to obtain 5 candidate TCRs from 5 distinct TCR clusters. From those 5 candidate TCRs, we validate them in vitro and found that indeed they are tumor-active. And then we've selected 3 of those TCRs for technical limitation for the in vivo validation. And what we found is that in an optimal setting, by transfusing 5 million cells, the single TCR alone could control tumor growth. But now in a more stringent settings with only 1 million cells transduced, we saw that the combination of the 3 TCRs, this TCR cocktail, better controls tumor growth than the single cell TCR alone, thus proving that our selection of TCR is indeed relevant. And here's my conclusion slides, and I hope you appreciated how we've developed and benchmarked TRT-PRED, how we've also designed this mixed TRT-PRED approach, and how we validated it in vivo. I also want to show that with all the patients that we've collected, only one would have not been able to use mixed TRT-PRED because they don't have enough TCRs, but we believe that this patient also have a single-cell TCR sequencing that is not great quality. Anyway, and we also believe that this will pave the way towards better tumor-specific models and to maybe also predict tumor specificity from T cells found in the blood directly. I want to thank everybody who has collaborated with me on this project, especially Professor Alexandre Harry and my colleague Joanna Schiffel, but also Marta Perez from Vincent Souet's team. And I also want to finish by just making a bit of publicity. If you're interested in seeing what our lab is doing, we have 3 posters about circulating TILs and TCR sequencing repertoires. Thank you.
If there's any question in the audience, Elena.
Remy, thank you so much for the talk. It's really beautiful. I'm wondering, is the application particularly now thought for melanoma, or are you planning to do this potentially in other cancer types? And also, how long do you think it will take the day you want to apply this for patient treatment from the biopsy obtention to the treatment?
I'm not sure I got the second question, sorry.
How long would it take to actually make a therapy based on this like personalized TCR therapy?
Okay. So our approach was trained only on melanoma. We're working now on creating another approach trained on a pan-cancer setting. We had really good results with melanoma, so we're expecting also good results with other types of tumor. For how long it will take, this is a tough question, and I'm not sure I have the answer. I think what's hard with this is that the TCR are orphan, so we don't know the target. And so if we have a TCR, we need to check, of course, that it's effective, but we also need to check that it's safe. And this, for being approved and etc., might be complicated. So I don't know. I don't know. Sorry.
Yeah, as we are a bit late on time, I think we will move to our next speaker. Thank you. And the next speaker is Marta Soria, a PhD candidate at EDBAPS, and she will be presenting preclinical characterization of RE7, CD7 negative T cells engineered with an academic anti-CD7 CAR for T cell cancer treatment. Marta?
Thanks for the kind introduction, and also to the organizers for selecting my work. So today I will be presenting the preclinical characterization of RE007, which is CD7 negative CAR T cells, CD7 negative anti-CD7 CAR T cells for the treatment of T cell cancer treatment. Okay, so in the field of T cell malignancies, T cell lymphoblastic leukemias, as well as lymphomas, and CD7 positive acute myeloid leukemias are infrequent diseases that carry poor prognosis and therefore remain underrepresented in commercial clinical trials, sustaining the need for novel targeted therapies. So up to date, allogeneic stem cell transplantation remains the only curative option for these patients. However, they must achieve remission prior to transplantation. So the main aim of our work is to develop anti-CD7 CAR T cells to be used as a bridging therapy in these patients. However, if we want to translate this therapy to this disease, it implies that a CAR T cell is going to be redirected against a T-lineage antigens, such as CD7, that was introduced earlier on by Dr. Sarajil today. And this means that this target antigen will be both expressed on healthy T cells and malignant blasts. So in this scenario, CAR T cell therapy faces different challenges. One of the major challenges that we face is that CAR T cells can attack and kill each other during the manufacturing process, or once they have been infused into a patient in a phenomenon known as fratricide. Also, that in contrast to B-cell aplasia, the loss of the patient T lymphocytes can lead to a life-threatening immunodeficiency. And there are some limitations regarding the use of patient-derived cells, as these patients are likely to be immunodeficient, and also that there's the potential contamination risk of the infusion product due to an accidental transduction of the malignant blast that may still be present in the patient's peripheral blood. So CD7 expression is higher in malignant blasts than in healthy T cells. The CD7 negative population is usually tumor-free. So in this work, we hypothesized that we could generate anti-CD7 CAR T cells from this naturally occurring CD7 negative population to simultaneously prevent fratricide and enhance the safety of our final product. And the main aims of our work were to, in the first place, select a candidate anti-CD7 CAR, also to compare different CAR T cell generation strategies, and lastly, to characterize the CD7 negative T cell population. So first, we developed 3 different CAR constructs containing CFPs derived from hybridomas generated at Hospital Clinic Barcelona. And to compare them, we did knockout CD7 at earlier time points during the T-cell expansion protocol. Once we had generated these CAR constructs, we challenged them in our TALL mouse xenograft models, and we observed that the 142.24 anti-CD7 CAR construct was the one achieving the highest survival rates. So we selected it as our clinical candidate. So as I mentioned earlier, we selected the CD7 negative population, which approximately accounts for 7% of the total T lymphocyte compartment in peripheral blood. So for that, we used custom GMP-grade anti-CD7 beads to deplete the CD7 positive population at the beginning of the expansion protocol. and this allows us to obtain a highly purified population. So by transducing this CD7 negative population, we were able to effectively generate anti-CD7 CAR T cells, as you can see in the flow cytometry plots. However, the population doublings at the end of the expansion protocol were significantly lower in this group. So we wanted to assess whether that was due to some fratricide that was happening in our product or either to CD7 negative intrinsic characteristics. So first, we observed that upon activation, the CD7 negative population re-expresses CD7, peaking at day 3 and progressively decreasing during the rest of the expansion protocol. So we hypothesized that this could lead to some residual fratricide within our product. However, when we co-transduce this CD7 negative population in green with an anti-CD19 irrelevant CAR in this context, we observe no significant differences. So regarding CD7 negative intrinsic characteristics, we did observe a peak of activation-induced cell death in the CD7 negative population at day 3 after activation. However, the surviving population was still able to proliferate in cell culture, as you can see in the histograms on the right-hand side. Taken together, our data suggests that CD7 negative cells show reduced expansion due to intrinsic features. So next, we aim to systematically evaluate the selection of CD7 negative cells with the current strategies that have been used in the field to generate CD7 CAR T cells, such as the CD7 ablation using CRISPR-Cas9, the intracellular retention of CD7 using protein expression blockers, also known as PEBBLE, and the pharmacological inhibition of CAR signaling using dasatinib, which is a tyrosine kinase inhibitor. So using all of these strategies, we were able to effectively generate anti-CD7 CAR T cells, as you can see on the left-hand side of the slide. However, when we challenge these CAR T cells in our in vivo TALL mouse model, we observed that the CD7 negative cells outperformed the rest of the treatment groups, achieving over 80% of mice survival. So due to their increased in vivo anti-tumor control and their enhanced safety profile, we selected the anti-CD7 negative anti-CD7 CAR T cells as our final product, and we named it as ARI-007. So next, at this point, we wanted to characterize in more depth the CD7 negative population. And to gain a deeper insight into the composition of this population, we analyzed a set of data from 13 reactive human lymph node samples, which are enlarged lymph node samples due to benign immune responses. And first, we observed that the CD3 positive population separated into 11 different clusters, throughout which CD7 was widely expressed. So taking a deeper look into the CD7 negative population, we observed that it was quite heterogeneous, with the CD4 positive central memory effector memory as well as CD8 positive effector memory subsets comprising the major phenotypes. And comparative analysis of the total CD3 positive compartment and the CD7 negative population revealed substantial differences. Specifically, we observed a decrease in the naive compartment and also an increase in the central memory and effector memory subsets. So we wanted to further characterize the phenotype of these CD7 negative CAR T cells varying CAR construct, and for that we transduce our CD7 negative cells with an anti-CD19 CAR, irrelevant as I said earlier, and in this case ARI-001 from Hospital Clinic. And in terms of metabolism, we observe no significant differences in terms of basal ATP production rates or glycolysis. However, we did observe a significant increase in the spare respiratory capacity of the CD7 negative cells compared to the bulk population. This suggested an increased plasticity towards metabolic function under stress conditions. And lastly, we perform a single-cell secretome analysis, and we observed that the bulk and the CD7 negative population separated into distinct clusters, highlighting their functional differences. And interestingly, the CD7 negative population showed over twofold increase in the percentage of polyfunctional cells, with a specific significant upregulation on the secretion of effector and chemoattractive cytokines. So to conclude, I would like to highlight some of our main take-home points. First, that we selected the 142.24 base CAR due to its superior in vivo anti-tumor activity. Also that other strategies such as the CD7 knockout, the PEBBLE, the CAR signaling inhibition enable us to generate anti-CD7 CAR T cells, however with variable in vivo efficacy. Out of these strategies, the CD7 negative CAR T cells were the one that achieved the highest survival rates. And despite this population showed reduced expansion rates, they also exhibited a more memory-related phenotype, superior polyfunctionality, and higher respiratory capacity. So we believe that our studies provide a strong rationale to test these CD7 negative anti-CD7 CAR T cells in a clinical trial for the development of treatment of TALL, LBL, and CD7 positive AML, which will hopefully begin soon at Hospital Clinic of Barcelona. And with that, I would like to thank my supervisor, Sonia, also the preclinical development team, Teresa, Álvaro, Manel, Mireia, and Iñaki, and you for your attention. I'll be happy to take any questions.
Thank you very much, Marta. There are questions in the audience? Or maybe I can start with one.
Sure.
Naive question here, but if I got it right, you take CD7 negative cells from patients. They do not expand that well, so I was wondering if you try to generate a CD7 CAR from a patient, if you have enough numbers of CD7 negative cells.
Yeah, exactly. So that was one of our main concerns, if we were able to still generate anti-CD7 CAR T cells from this CD7 negative population from the patient, and it's also reduced in the patient. It's true that we have worked closely with the manufacturing unit, and we were able to effectively produce these cells. We narrowed down the initial numbers from which we begin the GMP production, and we believe that we are able to achieve sufficient numbers. Yeah.
Nice.
Thank you.
Thank you. I'm not sure if there's more questions. If not, it's time for lunch. But before, I would like to thank all the speakers from this first session, and a big round of applause. To take 5 minutes because we will take the group picture. Right, Natalia, if I'm not mistaken? From where? Okay. Fantastic. So we wait outside for the group picture, guys. Thank you very much.
¿Ascoltas? Sí. Vale. Mira el boomerang. ¿Ha cambiado algo? No. Pues nada. No.
Excellent. Thank you, everybody. So it's really my pleasure to introduce our next speaker. She's Marta Pujol. She has a PhD in biochemistry and biotechnology by Kingston University in London. She worked in CENIO with Mariano Barbacid, contributing to the development of novel lung cancer therapy. And she currently is the scientific director of the Spanish Association Against Cancer, AECC, and the deputy director of its scientific foundation, leading research funding, science policy, outreach, and advocacy initiatives. For me personally, Marta is a dear colleague. As you have seen, AECC has really helped us build the cell therapy program in bio, and she's a dear colleague that's going to be speaking about what we need to remember, patients, because all of this is for patients. So it's really my pleasure to welcome her to give this talk, talking about how enhancing patient engagement in cancer research, a focus on patient-centric approaches to scientific discovery. Let's never forget about that. Thank you, Marta.
Hello everyone. Elena, thank you very much for the kind introduction, and thank you, Alex, Natalia, and everyone, for Silvia, for inviting us to be here today. It's a real pleasure to be part of this symposium. And so let's start. This is going to be a change of gear of what you have been hearing lately. And this is, as Elena was saying, we're going to talk a little bit about patient and how patients can be involved in everything that is being done with ATMPs and cell therapy. Let me start with these two pictures. If you see these pictures, what do you think? No, you see an eagle that it looks like, well, like a really big presence, and it's like it has a strong presence, and it really looks like dangerous and threatening. And if you see the lioness, it looks like she's eating her offspring, no? But what happens if you see things from a different angle? If you see it from another way? Well, it's completely different, no? Now the eagle, it looks like a little bird that is sad. And if you see the lioness, she's really taking care of her offspring. So not everything is as it seems. And when you look things from different perspective, you can see different things, no? And this is about that. What is the role of patient and public engagement and involvement? And is looking things from a different perspective. When we're in the lab, we are, of course, with cells, with biopsies, with models, mouse models, with different kind of models. But when you go into the clinics, it's very different. Every patient responds differently to the therapy. Some of them, they look similar, but they respond differently. So and they have many different secondary effects. And this is what we need patients also to be involved in research, because not all of them answer the same way to the same questions as you can see in the lab. Patient engagement, it has been developed throughout the years, especially in US, UK, and Canada. They have like this forum and this focus on patient for a long time. But in Europe, this is coming more and more frequent. For example, in Horizon Europe, there is a role on patients, also EUPATI, that is a very global European organization on patients. And also the EMA. The EMA is also looking at patients and asking patients and asking us to really help them in assessing the different drugs and different technology that are going to be approved. No? An example is the HTA, the Health Technology Assessment, that right now the patients, they have a main role in this assessment of technology that is going to have like this European flavor. In Spain, well, we were pioneers with our model in 2020, and this was like helping, how can patients really help us in making decisions in what are the projects that can be funded. Well, this is our normal scheme of awarding projects. I have to say that today we just open all the calls. So for everyone interested, go to our website. Today we just launched all our calls for new projects. And so today we're in day one, publication. From there, we have reception of the applications, we do a triage, then scientific evaluation, committees, until it is awarded and the project starts and are then follow-up. But what happened in 2020, we incorporated the role of patients in this at the same role as scientific evaluation, also looking from the patient perspective, what are the main concerns and if they were addressed by the applications that they were presented. And what we do is that then we have this chair, a patient advocate, that is the one that goes to scientific committees and that validates and reports all the comments of this PPI evaluation to the scientific committee. And what is the profile that this, but not everyone can be a patient advocate, no? And so we have like a very strict profile of patients that can be part of this patient advocate program that we have. First of all, they, of course, have to be patient, family members, caregivers, survivors of cancer. They have to be in a good emotional state. We are lucky enough that we have many different psycho-oncologists all over the country and in our offices. So they are the ones that evaluate this good emotional state. They have to be interested in research, of course. And this is very interesting. They have to act on behalf of a group. They do not act as a patient, as themselves. They have to act as and talk about all patients. No? So maybe they are going to evaluate projects of breast cancer, let's say, while they have been patients of colon cancer. No? So they have really to talk about on behalf of all other patients. They have to assimilate new information. The confidentiality is also very important because at the end of the day, they are reading proposals, so they have to be confidential. And this is also another one that is very interesting, no? Being able to speak in large groups. And this always happens when they go into scientific committees. When they are in front of you, for them, it's very difficult to talk, no? So that they need to have like abilities and the support to be able to talk in this large group. English, we will say this later a little bit, and they have to be eager to learn. This is about learning and evaluating and seeing what is coming. So we started in 2020, as I was saying, one province, that it was Madrid, just one call, that it was strategic projects. And in 2025, we have 10 provinces that are, and we started with 2 patients. Right now, we have, in 2025, we had 36 patients that were involved in the calls, 6 different calls, most of them our calls, but also international calls. So if you want to apply to international calls, be aware that patients are also being responsible of the evaluation. And we can talk a little bit further about this. And what other activities do patients do? I'm going really fast because I know that we have some time concerns, but we can talk later about this. Of course, the calls that we have, international calls, as I was saying, that's why we need English, because like in all the clinical trials, calls that we have with other countries, such as ATTRACT, or we are right now opening a new one that is called Interlocal for local treatments in phase 2 and 3. That also you can jump into looking at that. But also in research project, no? Sometimes researchers, they come to us and they say, okay, can you participate actively in our projects? And an example of this is the CGI, the Cancer Genome Interpreter, that is led by Nuria López-Vigas, but it has 17 countries participating, and we are leading everything that has to do with patients and understanding, for example, in that one, like for a patient, talking about sequencing. We think that this is normal, and we think that NGS is normal, but it's not. And we have identified by doing interviews to patients. No, none of the patients that we have interviewed know about NGS. None of them had undergone NGS, so this is not the regular, and that's why we need them to be close to the research projects. Also scientific attendance, for example, in ASECA, that I'm sure that some of you were there, or participated in events, but also specific consultations that also the scientists, some of you have asked us for some kind of feedback on protocols, on informed consent, also in data, no? And we can talk also later about the European data space and how patients have an active role in this. And scientific policy and counsel. There are many things that patients have the right to talk and to say. And with this, you can like really fast forward approvals, and we really have to raise our voice in Europe, especially now that we are undergoing the revision of the pharma package and now the Biotech Act. So patients, they have a very important role there, and it is also important that you translate your needs to us and to patients so that we know what are the things that are also worrying you, especially in ATMPs. But there's also a very interesting increasing interest of the scientific community on patient advocacy. An example is that we were asked to write a molecular oncology viewpoint on patients, and we have also coordinated a webinar together with the EACR and OIC regarding patient involvement in cancer research. In this webinar, there were over 100 different people from all over the globe, I would say, that to discuss about the future and the present of cancer and patient involvement. What are the what do achieve with involving patients in advocates and in research? First, to identify projects that they have a real impact. They really want an impact. I can tell you a very short example. In the call of Attract, we had a very nice project on ATMPs, and with a very good evaluation, scientific evaluation, and we really thought that patients, they were really going to put it very high, and then when it went to the patients, it was the lowest of all. And this was because patients, they have to be for a longer while in the hospital, and they have to undergo some kind of therapies that are very important and significant for them, no? So this is why they didn't value this project as well. This year we have run it again, and now the patients, they have understood how this kind of projects, they are the future, and now they are evaluating it much better. But it takes time. They really have to understand a little bit more about what is cell therapy, and we need you to make them understand the needs, but also without creating false expectations, that this is very, very important also. Also to detect unmet clinical needs, to disseminate knowledge, and increase confidence when you tell to a patient that another patient have reviewed the protocol. It's a way of getting confidence for them to be enrolled in a clinical trial or something related. So what role they have in ATMPs, in clinical trial design, as we were saying, participating in clinical trial, but also contributing in the protocol, monitoring the toxicities. For them, the toxicities and secondary effects are critical. And sometimes it just looks like a tick in a box, but for them, it's not a tick in the box. No, the secondary effects are very damaging for their survival and for the life after the disease. And the importance of informed consent and patient education, but also in ethical and regulatory aspects. The patients, they also have a very important role in compassionate use and equitable access that we were talking before, biobanks and genetic confidentiality, and also they are being involved more and more in the EMA and FDA and other kind of national regulations in order to have this kind of treatments approved. And what do we achieve by involving patients in those projects? Well, we talk about patient experience and quality of life. No, we have an increase and an impact on their physical and psychological aspects when they are going this kind of treatments. We also have to manage their expectations that I was mentioning before. This is very important to tell them, and so that they know that that their hopes, they don't have to go super high, no, when they go into this kind of trials. But also that this is a way, no, to get to new treatments and to follow up, and also this multidisciplinary way of working. And patients, they have to believe that they are also co-creators of what it is being built. So there has to be a real collaboration between patient and researchers. There has to be platforms to share data, to share knowledge, to share ideas, to share, well, their thoughts during the disease and during the treatment. And, well, relevance in the patient testimony. It's also very important to improve protocols and the way of planning these clinical trials. What happens with patients? Do they like to be part of patient advocacy? 100%. 100% of them recommend the experience. So they always want to be involved, and 94%, they say that they would repeat. So this is very nice to continue having them and having them involved, because they really, really participate and they're very, very active. Those are some of the things that they say, no? Like when they are diagnosed, it's like, why me? Why this happened to me? But the truth is that when they are part of this patient advocacy projects, then they understand that this is not about them, this is about all of them, and that there are a new level of technologies and progresses that are being made that for the near future, there would be the real medicine in the next few years. And just to conclude, well, the patients, they are active and they want to be active in cell therapies, although right now there's an uneven contribution of them. It also depends on the hospital, depends on the country, and there's not an equal contribution of them in ATMPs or in any other clinical trial. The future, it goes along with a greater collaboration and optimization of the clinical trials to be much more equitable and to all patients have the same access. And this starts with having a long-term relationship between the researchers and the patients. And the EI is also a nice opportunity that is going to break some barriers, especially geographical barriers, language barriers. That is always something that we're working on. And also training barriers. Training the patients is also very important. But we have to be cautious with the EI because we might not be including minorities. So we really have to include everyone when we are training our EI. And there are still many constraints like time, funding, infrastructures, and also a limited PPI participation. But this is the way to go. So thank you very much for inviting us to talk about this in this forum. And this is the real way to go. Thank you.
Questions for Marta? Marta, I'll ask you a question. So you have a room full of scientists, clinicians. If you have to tell us in a nutshell, what do we need to do better?
Well, not seeing the patient as the last slave of the chain, like seeing the patient as the real first one, no? And how they, because like this happened to us when researchers in the scientific committee at the beginning, they were receiving the reports from the patients. At the beginning, they didn't believe in them, and then they read them, and they were extremely good. Sometimes even better than the experts, no? So they know about that because, like, for them, this is their life, no? So they really get very into it. They know a lot, and you really have to hear them because they have very nice ideas, very good ideas, and there has to be a place to exchange the ideas between the patients and the clinicians and researchers.
Thank you. Joaquin?
I have a practical question because it has happened to many of us that we are writing our proposal and then we realize that the proposal requests that patient advocates or patients are involved, and we rush to the ACC to ask for help. What would be a rational protocol to address this? Not waiting for a call, but would you like groups that approach you in advance? What is the rational way to do it?
Yeah, well, the truth is that and now this is very common and very often, and you know it especially, and it is always like, okay, we need to write this work package with patients, you have 1 day, no? And, and so that's, well, we don't have enough time. Like, we have regular jobs and plus write these work packages. So sometimes we don't have enough time to be part of the projects because of the time constraint that we are just like the last ones, but the super last ones, no? So if we could be involved, like when you start thinking about the proposal, just as another collaborator, that would be awesome, no? To have a little bit more of time and to really construct something that it makes sense. Because if not, we can just offer the last minute thing that we can offer you, no? But if we can think about it from the very beginning, it would really make sense and be tailor-made for your project and not something like, okay, we can do, I don't know, a workshop with 4 patients, no? The earlier the better.
Thank you, Marta. Thanks for the great talk and thanks for coming. I have 2 questions. One of them is, do you see any cultural differences among countries? Because traditionally in Spain, we don't have such, the involvement of patients is quite recent. And I've always heard, for example, that in Spain, people are always willing to consent in more than in other countries. I don't know if you, talking to your colleagues across different countries, have you seen differences?
Well, the thing is that I think that in Spain we are very generous in many different ways. And in one of the ways is in adhering to clinical trials. That's why we are so good in clinical trials. If you ask a patient, do you want to participate in a clinical trial? 100%, I don't know, but like the percentage of patients that say yes is very, very high. And they always want to be more and more involved in every kind of decision. But they want their contribution to be meaningful. So the other day I was at a meeting and I was with a patient, and that patient in the program didn't even have a name. It was just called patient. And it's like really hard for them to just speak up and say, okay, well, I'm right now I'm not a patient. Like I was a patient 7 years ago, but now I'm just like a regular person that had 7 years ago I suffered the disease. But this is very hush, no? To be like in a symposium and to be the patient. So this is something that we have to really change our mind. In U.S. and well, it's very difficult. It's very different because they have done it for a couple of years. But in the U.S. right now, everything that has to do with patient, there's no funding, no. So we really have to. Well, I think that we are doing things well, slowly. But another problem that we have is that when you go to patient advocates, most of them are women and with breast cancer. So we have a problem in getting men to be part of this kind of programs, and with prostate cancer it would be great, no? But but always women with breast.
Yeah, and my second question is because we were talking to a patient in one of these fora, and he was saying, I don't like this picture of the patient being in the center of the research. I want to be besides. I'm going to be helping. Do you think there is a conflict here? Are we tending, to put the patient too much in the center? Whether they don't want to be in the center, they want to be part of it.
Yeah, I think that they want to be collaborators of researchers. So not like so, because you say patient-centered, but at the end of the day, it's just like putting the patient in the center in the picture. But if you collaborate, there is a real relationship between both, and you can talk equally. In this way of patient-centric, there's always leaving it to the side, and there's not this strong relationship between them.
Hello, thank you very much for your talk. I was wondering, because what Joaquin said is true, that all the grants suddenly ask for this patient advocate plan is quite detailed sometimes. But I feel there is a lack of a structure, at least in Spain, because I think these patients should be involved since the beginning of the trial, but also they should get some reimbursement, right? And from an academic perspective, it adds another layer of complexity, also in the economical part. Is there a plan in Spain how to centralize this and permit to have this figure in all the trials and projects?
Well, the truth is that right now it's a little bit amateur in Spain, at least, so that's why it's not being reimbursed. but we are getting there, no? And this is the way that it should go. And because at the end of the day, most of them, they are really getting very specialized. They are getting training, and this is a job at the end of the day. There's a lot of work. So right now, there is no way, like this is not working that way, but I think that in the near future, we will see a change of gear in this sense. And if we really want, as Alex was saying, to be like really researchers at the same level as patients or patient advocates, they really have to have at least some kind of reimbursement or whatever in the best way.
Introducing Marta, I forgot to introduce myself. So I'm Elena Garralda. I lead the Early Drug Development Unit in Vall-d'Hebron Hospital here in Barcelona. And it's really my pleasure to chair this second sessions towards clinical translation. And with that, I'm going to introduce our next speaker, Elena Gross. It's really a pleasure for me to introduce her to this symposium. She doesn't really need any presentation here, but as you know, she's a biologist. She did her PhD here in Barcelona and then did a postdoctoral work with Steven Rosenberg. And really, when she joined Vall-d'Hebron, she spearheaded all the cell therapy program that is currently there. Personally, she's been a colleague, she's been inspiring. She's the reason I went into cell therapy, and we've been working together this last 9 years already to bring the cell therapy program in VHIO to life. And so it's really a pleasure to hear you and to listen to your talk. She's going to be talking about harnessing peripheral blood to create next-generation T-cell therapies for solid cancers. Thank you, Elena.
Thank you, Elena, for such a kind introduction. I did add a slide of my conflict of interest. I must say that I'm going to be starting by talking about TIL, and Dr. Uday Kamula already presented the topic and gave a very nice introduction. Really, in the field of TIL therapy, the accelerated approval of lifelucel, which is the TIL product developed by Iovance, was quite a milestone. It was the first one-time T-cell therapy to be approved ever for a solid cancer. And this was based on this clinical data, where we can see the percent change from baseline in metastatic melanoma patients, cutaneous melanoma patients treated with TIL after a conditioning chemotherapy and together with IL-2. And this graph summarizes 2 consecutive clinical trials, including 153 patients treated. And as you can see, well, there was a 31% objective response rate, of which 8 patients achieved a complete response and 40% achieved a partial response. Importantly, this showed that TIL therapy can work in patients that are refractory to immune checkpoint blockade. This approval came after over 40 years of research. And this is important to point out. It took a long time for the first TIL therapy to be approved. But it's, for us in the field, it's important to know what's next. And this is what we're going to be talking about today. So we heard from Uday Kamula about efforts in selecting patients, which could definitely impact and increase the clinical efficacy in these selected patients treated, and also on moving towards other indications other than the more immunogenic tumors that are classically been treated with TIL therapy, which is mainly metastatic melanoma, but also some non-small cell lung cancers. There's definitely a need to overcome the chemotherapy and the high-dose IL-2 that's given associated to the TIL therapy. And this is because the main toxicities associated with TIL therapy really derive from these lymphodepletion and high-dose IL-2. There's also attempts to enhance and augment clinical efficacy. There's efforts from multiple groups by manipulating the T cells to engineer them to express and secrete cytokines, also to knock out, for example, PD-1 on TIL, and also simplified and scalable manufacturing. But today I will talk mainly about generating and developing more defined tumor-reactive T-cell product, which is something that I believe in the field we need to work on. Now, as Uday presented before, TIL can recognize a wide range of antigens, including tissue differentiation, cancer germline, and neoantigens. But really, these neoantigens have been shown to be optimal targets for cancer immunotherapy, mainly because of their exquisite specificity to the tumor. They are only going to be presented by cancer cells. And they can be found, we can find neoantigen reactive T cells in almost 80%, approximately 80% of cancer patients, regardless of the tumor mutational burden. There's a correlative evidence that these neoantigens are important, not only for the efficacy of TIL, but also for the efficacy of other immunotherapies, such as immune checkpoint blockade, suggesting these neoantigen reactive T cells play an important role in the anti-tumor efficacy. And there's several selected case reports, and now small cohorts of patients treated with neoantigen-selected TIL, that suggests that potentially by selecting these neoantigen reactive T cells, we could enhance clinical efficacy. Because of all these reasons, and when I moved to Barcelona after my postdoc from Steven Rosenberg's lab, I started to partner with Elena Garralda here to really establish a preclinical work to start a phase 1 clinical trial using TIL selected based on neoantigen recognition. And very briefly, this starts by excising a small biopsy. And although most TIL therapies are established from resections, we started with biopsies and we still continue with biopsies, but hope to include surgeon and bigger resections in the future. Now, with these biopsies, we cut them in small fragments, we expand them independently in high-dose IL-2, as it's regularly done. In parallel, we sequence the tumor. We perform whole exome sequencing to identify all patient-specific non-synonymous mutations. And with this information, we generate 2 reagents. On the top, you see the tandem genes, which are plasmids that encode for all these mutated candidate neoantigens. And we can concatenate up to 25 in each construct and make as many constructs to cover potentially all neoantigens expressed by the patient's tumor. We typically also, in parallel, synthesize the peptides. So we don't only use the tandem gene approach, but also synthetic peptide pools. Ultimately, these reagents are used to either electroporate RNA encoding for these neoantigens or pulse these peptides onto autologous APCs derived from the patient, which are expressing all the HLA class 1 and HLA class 2 molecules in the patient. And we use these autologous antigen-presenting cells, in our case we use ex vivo expanded T cells, as targets in co-culture assays. We co-culture our TIL, which are expanded from the tumor fragments, and only when and if the TIL are found to be neoantigen reactive, we select those TIL cultures that contain neoantigen reactivity for expansion and patient treatment. And really, this has been quite an important effort for the last years with funding by the Instituto de Salud Carlos III, but also from the Fundación BBVA. And again, thanks to partnering with Elena Garralda. Importantly, Ima Creus is the person from my lab that's highly involved in all these screenings, and also Vladimir and Julia from the clinical team, and Silvia Martin Yuesma, who has brought a lot of expertise in regulatory work. The TILs are currently manufactured at the Blood and Tissue Bank, but as you will see, we hope that we can establish our own TIL product in our own GMP facility soon. As an example of how this therapy works, in this patient was a metastatic melanoma patient, cutaneous melanoma, that had undergone 3 prior lines of therapy, including immune checkpoint blockade. And in this patient, 16 TIL expanded from 24 fragments that were initially put in culture. I'm only showing you here TIL F3 and TIL F4, 2 of the cultures that were tested, where you can clearly see that TIL F3 displayed recognition of a neoantigen present in peptide pool 1, while TIL F4 displayed recognition of a neoantigen in peptide pool 2. And despite we had the tandem genes that exactly encoded for the same neoantigens in the peptide pools. We only saw recognition of the peptide pools, but not the tandem mini-genes. And this is likely because these were CD4 T-cell responses. Once we observed this response, Ima actually performed the deconvolution experiments, which can tell us exactly which are the neoantigens that are being recognized. And as you can see, for TIL3, it was a neoantigen arising from ORC4. And for the second TIL, it was a neoantigen arising from LRP1. And so in this patient, we select 3 different TIL cultures that contain reactivity against these 2 neoantigens. And the patient actually received 2 doses of lymphodepleting chemotherapy instead of the 5 doses that are supposed to be administered due to toxicities. The patient received 2.7 times 10 to the 10 T cells containing reactivity against ORC4 and LRP1, and these were CD4 TIL. As you can see, following the treatment, and although the patient received only 2 doses out of the 6 potential doses of IL-2, we did see some tumor regression. This patient did obtain a partial response, although it was only transient, as you can see here. And in the end, it reached some stabilization, and after that, the patient went on to progress. So there's definitely still room for improvement, and this is only one example of the patient treated. I wanted to mention a little bit of all the ongoing clinical trials using TIL at VHIO very briefly. I think, and I've spoken to Elena several times, we really started this effort, the house from the roof. TIL selected based on neoantigen recognition is pretty much one of the most challenging things one can do because it's extremely personalized. But since this first trial, we have now a few others ongoing. Importantly, Pragmatil with EU Horizon, which is an EU Horizon-funded multi-center clinical trial, together with other European centers, including NKI and Herlev, where the main aim is to reduce the toxicity that's associated with the high-dose IL-2, which I already mentioned is going to be really important. But together also with collaborators at the Instituto Catalan d'Oncology, we have another 2 trials with our own TIL, and this is the standard non-specifically expanded TIL for 2 different rare indications. Importantly, as I mentioned, I'd like to share that the construction of VHIO's own cell therapy accelerator, our GMP facility, just finalized. And I think this represents an inflection point for our ability to translate these therapies into the clinic, and we're all really excited about that. And also, it's important to mention everyone that has been contributing to the possibility of getting these treatments to patients. And importantly, the collaboration with Elena Garralda, but also Silvia Martin Yuesma, the Blood and Tissue Bank, who is right now manufacturing Altil, but also all these other collaborators, all of them playing a very important role. It really takes a village. Now, moving on to the more translational work done at the lab, really, there is definitely room for improvement. Even when we select for TIL recognizing neoantigens, based on the literature and our own work, we need to still improve these T-cell products. And again, I think that despite we're enriching for a neoantigen reactive TIL, we still are not reaching the levels of enrichment that one would want. And really, when we put TIL in culture tumor fragments in the presence of high-dose IL-2, what can happen is that, and what frequently happens, is that you're not only expanding the reactive cells, you can also expand bystander T cells. There is room for really trying to enrich the T cell product administered for neoantigen, for tumor reactive T cells. And this can be done in several ways, and different groups approach this in different ways. One can potentially in vitro sensitize these T cells from the beginning with tumor antigens. One could potentially also select activated T cells following ex vivo antigen-specific stimulation, even with tumor, if one has enough tumor targets to do that. One can select peptide HLA multimer positive T cells that are loaded with candidate tumor antigens. But one approach that has attracted me the most for many years now is the discovery of biomarkers expressed on tumor-reactive T cells, and in particularly in TIL. So it is now pretty established, and you heard from Remy's talk this morning, that tumor-reactive TIL share features across cancer types that can be leveraged to develop personalized TIL, but also TCR cell therapies. And this is just one example of one of the, a publication from Kathy Wu's group back in 2021, where we can see the results of single-cell sequencing of the TIL infiltrating for melanoma tumors. The data were pulled together, and these UMAPs, actually what we can observe is that on the left, the virus-reactive T cells are clustering to parts of the UMAP showing that these T cells are effector memory, but not exhausted, whereas the neoantigen reactive T cells and the melanoma-associated reactive T cell receptors, which were mapped onto this UMAP after very careful identification of these receptors, shows that they're really not co-localizing with the virus-specific TCRs, but rather they're showing this exhausted transcriptomic profile. Now, this is consistent with previous literature, including some of my prior work using flow cytometry, and we know that there are definitely some markers such as PD-1, CD39, and CD103 that can help us define the tumor-reactive population infiltrating cancers. This can be used for potentially selection of these T cells and expansion for patient treatment, but also, as Remy was explaining, to identify TCRs for personalized TCR therapy. Now, for a while as well, I've wondered whether we could develop a personalized tumor-reactive T cell product exclusively using peripheral blood. And this is because currently tumor biopsy obtention or resection is a prerequisite for the development of cell therapies and vaccines targeting neoantigens. We need these biopsies or resections to isolate, extract DNA for whole exome sequencing, and in the case of cell therapies, also for the identification and expansion of TILs or TCRs. And there are many patients that actually don't have accessible tumors and cannot undergo potential operations. Beyond these practical implications of accessing a patient population that we cannot currently access performing biopsies, we also wondered whether a single biopsy or a single tumor resection in a patient that has advanced metastatic disease could miss the complex neoantigen landscape in patients with advanced cancer. And of course, we will have situations like the one on the left, where if there's low heterogeneity, you can biopsy or resect one lesion, and it will represent what's happening in the rest. But in the case of the right, if there's high intratumoral heterogeneity, it's very possible that we're not really getting a representation of all the neoantigens, nor potentially all the reactive T cells. And this can happen at the level of intratumoral heterogeneity, but also intertumoral heterogeneity. And so in order to address that, together with Andrea Garcia-Garijo, a very talented PhD student, but now a postdoc in the lab, we really focused on studying whether we could leverage the peripheral blood in two ways. First, to isolate cell-free DNA and potentially use that to identify and discover neoantigens. And although cell-free DNA and circulating tumor DNA are being explored typically using targeted sequencing, it's not been established to use this cell-free DNA to identify neoantigens and to discover neoantigens. And also in parallel to that, we wanted to explore the possibility of using the peripheral blood lymphocytes. And always, because this is not the standard, we wanted to compare to what we would obtain using a tumor biopsy. As an example of how we did this, I'm going to show you the results for one patient. Is a patient with mismatch repair deficient colorectal cancer we call GOI5. As you can see on the left, in the TC scan, there are multiple tumor deposits in this patient with advanced cancer. And just because it was more accessible, we were able to biopsy the tiny liver lesion, which I'm not sure you see, right? I don't know if this is working. a tiny liver lesion, okay? And we extracted DNA from that liver lesion. In parallel to that, we extracted blood, and we isolated the cell-free DNA, and we used the PBMCs, circulating PBMCs, as normal DNA, and performed exome sequencing to perform somatic variant calling and identify candidate neoantigens that could give rise to neoantigens. We also studied the ploidy, the purity, and the clonality of the variants, and I will take you through this slowly. For this patient, and it was one of the first patients we studied, we were quite impressed, actually. First, it was a mismatch repair deficient cancer, and so we did identify quite a lot of mutations. You can see a total 2,873 mutations, of which 76%, almost 77%, were non-synonymous mutations which could give rise to neoantigens. Importantly, we were very excited to see a high concordance between the mutations that were identified in the biopsy and in the circulating tumor DNA, with 1,697 non-synonymous mutations that were overlapping between the two samples. Importantly, as you can see, there were also some mutations that were uniquely identified in the circulating tumor DNA, but not in the tumor biopsy, and the other way around, which we more or less expected more. With this data, and again, taking into account the variant allele frequency, the purity, the tumor purity in the sample, and the copy number alterations, we studied the clonality of each of these variants in each of the samples studied. So shown here on the x-axis is the clonality, the cancer cell fraction that harbors each of these mutations in the tumor biopsy, and on the y-axis in the cell-free DNA. And as you can see, by in blue, we are showing the mutations that are actually shared between both lesions. And as you can see, most of them have a cancer cell fraction of 1, which means that they are present in basically 100% of all cancer cells. But importantly, again, I'd like to highlight the fact that we identified some mutations that were unique to the circulating tumor DNA, shown here in orange, which were not present at all, and obviously subclonal because they were not identified in the tumor biopsy, and the other way around. So with this data, what we did is, in fact, we generated these reagents that I previously mentioned. We generated tandem mini-genes, but also synthesized the peptide pools of encoding for 186 of these shared mutations, but also screened 155 of the mutations that were uniquely identified in the circulating tumor DNA, but not in the tumor biopsy, and 22 that were unique to the biopsy. And so I'll shift gears a little bit because in order to identify these reactive T cells in the blood, we need to manipulate in some ways. Now, it is known that reactive T cells, if present in the blood, are going to be there at a very low frequency. Now, I previously showed you there are some markers that can be used to define and, in fact, can be leveraged to identify the reactive T cells in tumors. Some of these for CD8 TILs are PD1, TIM3, LAG3, CD39. For CD4s, it is less known. But we wanted to, building on previous data, my own data from when I was in Steven Rosenberg's lab, we wanted to exploit this same strategy for the blood. And in fact, we decided to use two different markers, PD1 and CD39, based on the literature that was available when we first started this project. And it's important to mention that PD1 and CD39 are expressed to some extent in the blood, but in fact, the population that's co-expressing both markers is a tiny population of less than 0.5%. So this is quite challenging, sorting these cells. And because we obtained such few numbers, we need to expand them ex vivo before we can test them for neoantigen recognition or tumor recognition. So we did exactly this for this GOI5 mismatch repair deficient patient. And in fact, indeed, we found both CD8 and CD4 circulating lymphocytes that expressing PD1 and or CD39, which harbored T cells targeting neoantigens. They targeted neoantigens that were found to be shared between both samples, the tumor and the circulating tumor DNA, one neoantigen that was unique to the tumor. But importantly, we detected circulating lymphocytes, but not TIL. that recognized 3 neoantigens that were uniquely identified in the circulating tumor DNA and not the tumor biopsy. So we would have completely missed these reactive T cells if we had only studied the tumor, really suggesting that there's a heterogeneity in some patients that we need to address. Overall, in this cohort of 8 patients, which in fact across different tumor malignancies, including breast cancer, head and neck cancer, 1 colorectal MSS patient, and 2 melanoma patients that were refractory, and 2 anti-PD-1. We did everything that I explained before, and when we compare the exome sequencing, the results, the number of mutations identified in the circulating tumor DNA or cell-free DNA compared to the tumor biopsy, in general, we found quite a nice correlation in the number of non-synonymous mutations identified in both samples, with the exception of 2 patients, GOI2 and GOI3, which, as you can see, they displayed lower numbers of mutations in the blood compared to the tumor. And in fact, when we looked at the % of shared mutations between 2 samples, clearly those 2 patients stood out, and there was barely any sharing. And in fact, we now know this is because there was barely any circulating tumor DNA in the patient's plasma. Which makes sense. This strategy is only going to work if and when the patient's tumor is shedding DNA into circulation. Now, for the rest, as you can see, the % sharing was quite high, ranging approximately around 75%, but in some cases even higher. And we were able to select all these candidate non-synonymous mutations and screen these patients' blood, but also the TIL for neoantigen recognition. And as shown here, in this heat map, you can see the reactivities that were identified, and this is on the top showing the reactivities found in the blood, and in the bottom, you can see the reactivities found in the TIL. Mainly in the blood, what I can really conclude is that most of the reactivities identified were found in the CD8s and CD4s that express PD-1, high levels of PD-1, being the PD-1 high CD39, the positive, double positive population, the one that harbored the highest number of neoantigen reactivities. There were 2 patients where we were not able to identify reactivities in the blood, but this worked across different cancer types, as you can see. And importantly, as you can compare to the TIL, we actually missed most of the CD8 reactivities in the TILs, whereas we were able to identify these CD8 reactivities in the blood. And this is comparing the peripheral blood strategy of selecting the cells compared to the classical ex vivo expansion of TIL in high-dose IL-2. This slide actually highlights this. We were able to identify more reactivities in the peripheral blood compared to the regular TIL expanded in high-dose IL-2. And indeed, 29 reactivities identified in the peripheral blood and 19 in TIL, and really it's the CD8 reactivities that we were not expanding well when we put the TIL in high-dose IL-2, really showing that there is room for improvement compared to the classical product, at least in terms of neoantigen reactivity. Another important thing that we wanted to check is whether we can capture all neoantigens or just a fraction of neoantigens in the circulating tumor DNA. As shown here, when we look at the ctDNA non-synonymous mutations, we could capture most of the non-synonymous mutations, but there was quite a bias towards clonal neoantigens, which indeed would be even more attractive targets for cancer immunotherapy because they are present in all cancer cells. And more importantly, using this ctDNA to perform whole exome sequencing to identify neoantigens was able to detect and capture 30 out of 33 of the neoantigens recognized in these patients. So the vast majority of neoantigens were captured by doing this whole exome sequencing of ctDNA. Finally, I'd like to point out that although our strategy is geared towards using peripheral blood as the sole source for this therapeutic approach, ctDNA whole exome sequencing can also be used to complement tumor biopsies to enhance the clonal resolution of the variants identified. And in this in this graph, I'm showing you all the neoantigens that were identified, and really what we're showing is the clonality in each of the samples that were sequenced. And I'd like to highlight, for example, patient GOI6. You can see the neoantigens that were recognized by the patient's blood and TIL are shown in pink on the right. When we biopsied this lesion, most of the neoantigens appear to be clonal, very close to 1. But in fact, when we looked at the cell-free DNA, these mutations are actually clearly subclonal, at least based on the blood. So really, there's more heterogeneity than potentially we expected. Although this is still suggestive data, this does support that the use of this circulating tumor DNA and whole exome sequencing and identification of neoantigens could really provide additional information to make more directed and precise treatments. As a summary, peripheral blood is an easily accessible source that can capture both neoantigen and reactive T-cell heterogeneity. The tumor-reactive PBLs share phenotypic traits across patients that can be exploited to develop personalized cell therapies, and we have additional projects in the lab with Melissa Thone to look at this in more detail through transcriptomic analysis. But also, I wanted to highlight the potential of these circulating tumor-reactive T-cells. I've really focused only on neoantigens for the purpose of this talk, but this population of PD-1, CD39 positive T-cells actually displays a diverse TCR repertoire that's capable of recognizing different types of tumor antigens, not only neoantigens. And I think this diversity only makes it more interesting. And I also showed you that this population can target potentially different, distinct tumor clones, which will be very important to combat heterogeneity. And so coming back to one of my first slides, I think this circulating derived, peripheral blood-derived product attempts to tackle some of these by trying to define a more tumor-reactive cell product. Also simplified because this is a simple manipulation that requires just selection of this population and expansion, potentially shorter times for treatment. And we hope that this can help us access other indications. But as I mentioned, there's plenty of other things that we need to tackle and address. And also, through these efforts, we have identified multiple TCRs that we're also aiming to exploit in the future. With that, I'll thank everyone that's been involved in this work, especially Andrea Garcia-Garijo, but also Ima Creus and Pierre Levy, all the TIL working group, which I mentioned before. My partnership with Elena Raralda has been very important to really bring these therapies to patients, and I'm really excited of what's coming next. Special mention to all patients who have participated, and I'd like to briefly mention that Judith Diaz, who recently defended her thesis, will be presenting her poster this afternoon, but also a presentation tomorrow about the tumor-intrinsic resistant mechanisms of cancer cells to T-cell mediated killing. And I hope you will enjoy that. Thank you very much.
Thank you, Elena. A beautiful talk, a beautiful summary. Questions?
Great talk, Elena, and meticulous and detailed as usual. Probably the most meticulous surgeon, and meticulous scientist in the surgery branch. My question is about the peripheral blood T cells, which you've been studying for years. So they exist in the peripheral blood, and they have PD-1. Presumably, they have seen antigen. I want to get your philosophical thought on what they're doing in the peripheral blood. Are they leaving the site of antigen, presumably because they are PD-1? Are they seeing circulating tumor cells? Because it is a very attractive source for cells, not just for TCR, but potentially for in vivo therapies. Yeah. And the question is, are these the mediators of therapeutic response in patients receiving checkpoint blockade? Because we all exclusively focus on the TIL, but are these cells functionally doing something? I was curious, you've been in this field of the peripheral blood cells, and I'm sure you've thought about it. What are they really doing?
Yes, it's something that I ask myself still very frequently, and I get this question a lot, actually. This question is very difficult to answer in humans. And generally, it's thought that the tissue-resident cells don't tend to recirculate. But this is a dogma that's now been broken, particularly with mouse models. And I think the latest data come from Rami Youssef, who's generated a gene signature. In fact, shows that reactive cells in the blood, and actually looking at the same exact clonotype in the blood and the tumor, they share some traits. They're not identical. The ones in the blood look less activated. But really doesn't tackle whether they're coming from the lymph node and homing to the tumor, or they're trapped in the blood for whatever reason. So I think addressing this question will require syngeneic mouse models, which is something that we should consider. I get this question a lot, and I think it's important because actually otherwise, until we don't get to treating patients, we don't know if those cells will be able to home to the tumor. But I don't see why they shouldn't.
And I would put this experiment in your head. Really look at your post-TIL patients after ablation, because we do TCR repertoire analysis on the blood before and the blood after. And I'm obsessed with the repertoire after lymphodepletion, because there are some clonotypes that are not from the TIL, they're from the bone marrow. And whether you see a difference in tumor reactivity post-TIL infusion that are not from the TIL. So that would be something that you might be interested in.
That would be after reconstitution? Okay. But that's potentially the engrafted TIL?
Not the engrafted TIL,
Okay.
the
So
endogenous
no clonotypes.
repertoire, yeah.
Okay, thank you.
Great talk, Elena, as usual. Congratulations. I have a very naive question. What is this real neoantigen-specific T cell? Like, what is the true neoantigen-specific T cell? All the TILs that you have identified, do they have similar potential to kill the tumors? And
You mean if all of them have the same capacity of killing tumor?
Yeah, and is it like a gene signature you could identify that could say, okay, that particular TIL gonna work, or you're at the end, or we're thinking about TIL therapy, not a cocktail of,
Yes.
TILs.
I don't think all neoantigen reactive TCRs are equal, and I think we'll be hearing about that from Cristina Puig tomorrow. I look forward to that talk. And I do think that there's much more layers of complexity than just taking all neoantigen reactive T cells or TCRs. And we will have to learn exactly what are the traits that mediate this anti-tumor response, which we're still struggling to understand. Thank you.
Okay, just one last question there in the back.
Hi, Elena. Thank you for the amazing talk. Those two patients that showed like the lower reactive T cells, did you see any pattern of prior treatments that was related to that, or are you exploring strategies to promote release of the tumor DNA?
Actually, we have not looked at that in detail, but it's something that we should definitely consider. And regarding prior treatments and how that can influence our ability to find them, I think that's also interesting. In fact, with Endica Prieto in the lab, we have a project focused on understanding the reactivity of the T cells in patients treated with immune checkpoint, whether we see more or less after treatment, and whether that could be a better source for adoptive cell therapy. This is something that is being explored. Thanks.
Thank you very much, Elena. And Sine Recker. She's professor and group leader at the Department of Health Technology, Technical University of Denmark, specializing in T-cell immunology and cancer research.
All right, it's coming now.
Her lab develops cutting-edge multiplex strategies for T-cell detection, including DNA barcoding to simultaneously track over 1,000 antigen-specific T-cells, advancing our understanding of tumor reactivity and immune responses to cancer therapies. Welcome.
Thank you very much for the thorough introduction. And thank you so much for inviting me to this wonderful venue. And I'm glad I didn't have to go through the rest of Alina's slides. I'm coming from Copenhagen, from Danish Technical University. And today I will tell you about some novel tools to improve immunotherapy in the context of cell therapy. So I have no disclosure. And the talk for today will focus about two elements. So one is strategies to improve fitness and persistence of T-cell products, and another is to use de Nouveau AI-generated mini-binders as a surrogate for T-cell receptors. So a while back, I've been working with Yi Sun. She's a polymer chemist and a dear colleague of mine. And we wanted to develop a nanoparticle to engage with T cells. So we thought, let's try to first make kind of a little bit a copycat of the Dynabeats using anti-CD3, anti-CD28, just to prove that our nanoparticles can perform similarly as these already established reagents. And as a surprise, we actually saw really improved features of the T cells that were incubated with these nanoparticles that we have generated, and I will show you a bit of those data. So the particles are built on sugar moieties, dextrains, and then they are coupled with anti-CD3, CD28s on the cell surface. So one of the advantages that we have, and we think that this is actually the reason for the great performance of these particles, is that they are very small, they are biodegradable, so they can be taken up by the T cells via receptor-mediated endocytosis, similar to the natural process of T cell activation. And that is in contrast if you have like plate-coated strategies or you have large beads, you have kind of more of an abrupt signature where T cells have to sort of get rid of whatever it sticks to through that stimulation. So we can demonstrate that through this confocal microscopy picture that these particles are indeed integrated into the cell subunits. So what we see is that when we just look at purely expansion short term over 10 days, we see very much expansion on par with Dynabeats or Transact, a bit better than that. But where we really start to see differentiating signatures is when we keep re-challenging our T cells. And in this case, these are CD19 CAR T cells with tumors. So upon repeated stimulations, as you see here in the top corner, we see improved capacity to control tumor with CD19 CAR T cells expanded with T-expand, as we call these type of nanoparticles, versus those that were expanded with Dynabeats. We also see lower expression of exhaustion molecules such as TIM3, PD-1, LAG3, and TIGIT, and we see a higher proliferation capacity. So this really transitioned into also a better in vivo protection, and this is a JACO tumor model with, again, CD19 CAR T cells, and we can go as low as 0.25 to the million of cells in this model and still see tumor control with the T cells that we have expanded with this T-expand strategy. So these assays are, of course, controlled for the number of CD19 expression CAR T cells, so it's really sort of on a one-to-one basis that these T cells seem to perform better. We see that there is a significant transcriptional signature that differs between these cell subsets, and I won't go through all the genes today. The data is also available on bioRxiv and soon also coming out. But we really see stronger both cytotoxic signatures, inflammatory characteristics that are favorable, and also tumor infiltration molecules. So we think that this is in part the reason why we see this better performance also in vivo. So recently we have tried to take this to a more, let's say, antigen-specific level, where we have then, instead of having the CD28 and anti-CD3 surface conjugation, we have replaced that either with a peptide MHC molecule or the antigen that is recognized by a CAR. And then we co-conjugate now with cytokines, so this is also in an effort to try to remove all the cytokines from solution and only provide the T cells with the cytokines that are actually displaced on the surface of these nanomaterials. So we try to use this in the context of either TCR or CAR knock-in strategy with CRISPR or with retroviral strategies. So the data I show you here on the left is with TCR, a CRISPR-based TCR knock-in, where it is often, these strategies are often hampered by a relatively low transduction efficiency, but when we use these antigen scaffolds with the cytokines to expand such T cells, we really reach around 90% of cells that carries the CAR, or sorry, the TCR in this case, in a very sort of selective fashion. And we see the same also for CAR knock-in T cells. So again, we can improve the number of those receptor-carrying cells with a very simple expansion strategy. And we can also have the advantage of doing this in pools of TCRs, for example. So I think many studies are moving towards the direction of making these multi-specific T-cell products, where you would maybe like to have multiple TCRs expressed amongst the cells that you would like to inject. And here we can provide a simultaneous expansion of all of these together. Here is as a mixture, shown as a mixture of 5 TCRs. And that is, again, really on preference compared to if you don't have a selective strategy for expansion. You end up with very low frequencies of actually receptor-expressing T cells. So again, we see these improved phenotypes. Now we are a little bit focusing on the co-stimulatory molecules. They are higher, and we also see increased proliferative capacity of these T cells, a bit similar to what I showed you with the T-expand. And also in this case, in vivo models, we see again improved efficacy of tumor killing. This is again the JCO model with a CD19 CAR, when they are expanded with these specific antigen-selective expansion strategies compared to conventional expansion strategies. So again, we can maintain tumor at a very low dose, or maintain tumor control at a very low dose of T cells. So we think that these can really have some advantages in also simplifying the manufacturing strategy, as was one of the points that Alina also raised. Since these are biodegradable and they don't need to be removed prior to cell injections, it is a fairly simple process to add these to the manufacturing, and it improves the functional characteristics of your cells, and it performs the selective expansion that you would maybe otherwise have to do with sorting your cells, which is a complex strategy in a GMP manufacturing process. So next thing that we are now trying to focus on is seeing if we can actually use these in vivo. So this is just a very early data, and this is something that we're currently working on. So we can actually inject these particles and then get specific expansion of, in this case, these are our TCR specific T cells in a mouse, and see if we can selectively expand and maintain an adaptive transfer product, either being a CAR or a TCR product through longer time, and also to boost maybe the quantity that we have from these cells. And this could also be in combination, for example, with in vivo gene delivery, as we heard about this morning. So with this, I will just sum up that we now have this strategy to really boost antigen-specific T cells and at the same time improve their fitness. This has the potential really to decrease the manufacturing cost and also the handling requirements, and also hopefully we can demonstrate that they can also be used to maintain the T cells in vivo. So with that, I will shift gears and move to something rather different. These are de novo mini binders that can bind specifically to peptide MHC molecules. So this is something that we set out to develop, I guess now 2 years ago, but in fact it has been a pretty sort of fast-moving project. And this summer we published the data that I will now show you at least some of, and also some new data, back to back with 2 other groups that have done this the same. So just to also highlight that there is data out there both from the Garcia and the Baker lab, also demonstrating that these de novo generated mini-binders can indeed be used to recognize peptide MHC. And as was mentioned in this commentary, we strongly believe that this really have the potential to transforming the design of artificial T-cell receptors. So we are really interested and curious how this will develop in the future. So I will show you a little bit about what we've done here. So we really are designing these in an effort to be able to extend the target space and to be better at targeting peptide MHC complexes, which is kind of the only source that allow us to actually target intracellular proteins. And this is normally done by the T-cell receptor, as you're all aware of, but it's also challenging to both identify and also engineer T-cell receptors to modify specificity and affinity. And they are also rather large proteins, so using them, for example, in complexes such as bispecific T-cell engagers is also quite complex. So there's multiple advantages of having smaller molecules that are stable and where we can also really design and select for specificity. So, so yeah, I forgot to mention these were the, oh, no, I'm moving the other way around. This goes wrong now. How do I go back? This way? Okay. Just wanted to mention that Timothy Jenkins is the computational design researcher that I worked close with to facilitate this, and Christopher Biandarian has been really the hands-on people. So we chose to start with VHIO, and this was because that this is an interesting tumor antigen. It's expressed across many different tumor types, but also really for our efforts it was quite important that there is a structure of the peptide MHC complex that we could start with. So starting from this structure, we passed it through the computational models, being RF diffusion, protein NMPN, and AlphaFold 2 in this case, and with circulations of these strategies and various cutoffs that can be generated, we selected a pool of binders that, at least from the computational side, had the capacities to specifically interact with the peptide MHC. And I have to say I was myself quite skeptical that this could at all be feasible. So but we took these 44 top mini binders and expressed them then as a cellular display system. And then we generated tetramers with the peptide MHC in question, and this VHIO A2 peptide. And screened for recognition of these. And as you can see, there was a population of cells in our mammalian display that actually bound the tetramers, and upon sequencing we could find that, this is very lively, one mini binder that were able to bind to this peptide MHC molecule. So this one mini binder we could then further express, and we could actually make a cryo-EM map of the interaction between the mini binder and the peptide MHC. And we were of course glad to see that this cryo-EM very strongly resembled the structures that was also predicted by the AlphaFold models. We also measured the affinity of the interaction to close to 7 nanomolar, so it's a very high affinity interaction compared to most TCRs. So we could see that if we express this mini binder in a structure as a CAR, so with the mini binder on the cell surface and with the transmembrane domain and the CD28 costimatory domain, we could then actually see killing of cells with this receptor in, and cells that are known to express VHIO, so the A37475 cell line. So So the next thing that we wanted to explore was, okay, now we could make binders for this complex where we actually had a crystal structure, but that's of course a major limitation. So we went back, let's say, to our archive, and years back we studied new epitope recognition in TIL products, and one of the new epitopes that we found here is this particular one that is presented in HLA-A0101, and it's therefore, of course, only relevant for this one patient, but there is certainly no crystal structure of this peptide MHC complex. So we predicted this structure by AlphaFold, and then we went again through the same computational process, and what we could see is that we could actually find and identify 2 mini-binders that would also recognize this peptide MHC complex. So this really proved to us that we could actually have a full end-to-end computational pipeline going from the target to the actual binder, and with fairly high success, in this case 2 out of the 40 screened, one was of the 96 screened mini-binders actually recognized the targets. So, so what is really sort of course, super critical for these binders is the specificity and the cross-reactivity. So we evaluated that, and we see that there are some amino acid variances, as demonstrated here, that can be recognized still by the mini-binder compared to the wild-type sequence, which is the one that is to the very left. We tried to also compare that with some computational matrices and see very good correlation, which means that down the line, we are planning to do much more computational cross-binding screen as a first screening strategy, and then only for some selective smaller pool, we can do the actually experimental work. But this, of course, also encouraged us to try to see how can we actually improve these binders as we move along. So we took the original binder and put it into our diversification strategy, and you could do this in various ways. So we did sequence diversification, and we did another round of partial diffusion across the binder that we already had identified, namely this VHIO-BBVA binder. And starting from this sort of template of an already existing binder, as you can maybe see, improved our hit rates dramatically. So with this another diversification, around half of the new binders that we generated would actually bind to the target. So that means that now we have a rather long list of NY-ESO binders, and we can see that when we stain them with multimeres, which is kind of a crude way maybe to identify their affinity, we can see a range coming from very high affinity binders, and the red one is the one that had the 6.8 nanomolar avidity that we tested previously, and then to more low affinity binders, and you can see the TCR there in blue. So this tells us that it's possible now to generate really a large array of various binders and select these based on the properties that is required for the therapeutic application, and of course for the specificity that you would like to see. So one of the strategies that we are using to really dig deeply into the specificity of the binders and of TCR in general is what we call TCR fingerprinting. Here we are using our capacity to build large arrays of peptide MHC multimeres carrying a barcode, and we can then display peptides with variances of amino acids at each of the position. So with such an amino acid variance library, we can then ask what is the positions that is critical for the interaction between the peptide MHC and the TCR, and how does it look like if we vary each of these with all available amino acids. Of course, this data can then also feed into the in silico process, and we are now building sort of active learning models where we can use these data to iteratively train the development of new binders as we move along. So just to show you an example, this is the 1G4 known NY-ESO TCR. This is the fingerprint, and you can see that there is a motif that strongly resembles also the wild-type TCRs, but there are also some positions where there's quite some flexibility in terms of the amino acids presentation. And just to compare, the top one there is our first mini binder that we generated, and I think just by eye you can appreciate that that there is a bit more promiscuity in that than what you would see with the TCR. But then with the iterations of the new set of binders, we can find some that are very specific, as shown as the bottom one, which has a very strong preference for the amino acids that are also there in the wild-type sequence. And you can see a correlation between what we call TCR or mini-binder promiscuity and the affinity that we can measure just by the crude intensity of the tetramer stain. So this is just to demonstrate that I think here we really have a platform where we can really create large arrays and select based on the properties that are required, the specificity and the affinity, which again may differ depending on whether you are intending to use these binders in a cellular therapy context, or maybe, for example, as a T-cell engager, or maybe even as a blocker of T-cell reactivity. So this is really where we are heading now, is to try to iteratively both test this, but also improve the in silico pipeline, and then try to use these as T-cell engagers. Also try to make mini binders now for MHC class 2, and test if we can also use these to actually block T-cell reactivity, for example, of relevance in autoimmune disease. So with that, I will just thank all the people that have been involved. This is my wonderful research group, and here are some of the names that were involved in the data I showed you today. As I mentioned, this could not have been done without the awesome collaboration with Timothy Jenkins, who is also at the Danish Technical University, and also Yisun, who is the polymer chemist. So thank you for listening, and I'm happy to take questions.
Talk. Any questions, Joaquin?
Excellent talk, thank you. Regarding the mini binders, do you expect immunogenicity when you put them into humans, because these are fully synthetic proteins, right?
It's a very great question, and no, we didn't test it yet. So we have inbuilt or built in some, in the computational pipeline, there are some inbuilt strategies to try to minimize immunogenicity, and these are also very small molecules between 100 to 150 base pair, but this is one of the things that we are testing at the moment. It's super critical. Yeah.
Excellent. So in the interest of time, we will continue with our next speaker. Her name is Annette Kunkele. She's professor from CAR T-cell therapies, head of the Berlin Center for Advanced Therapy, and senior physician in pediatric oncology and hematology at the Charité in Berlin. She's a specialist in pediatric oncology with postdoctoral training in Seattle's Children's Research Institute, where she developed a CAR T therapy for neuroblastoma. And she currently leads research on novel CAR targets and solid tumor resistance, and she coordinates the European Joint for ATMP Consortium and charity lead for the UHA Gene and Cell Therapy Center. So that is a long list of things. Very happy to hear you speak.
You should have skipped that in the interest of time, but thank you very much for the kind introduction, and thanks a lot to the organizers for the kind invitation. So I will talk a little bit about the development of neuroblastoma-specific CAR that took place in Seattle, as just mentioned. And then I was actually asked to tell also a little bit what the hurdles were when we tried to implement CAR T-cell manufacturing at the Charité in Berlin. And then, of course, I will tell a little bit about our EU consortium, Joint for ATMP. So when I went to Seattle, there was an interest in the lab to develop a CAR against neuroblastoma, and since I am a pediatric oncologist, that made totally sense. Neuroblastoma was already briefly introduced to you this morning as an example for an ALK-expressing tumor. So it's an embryonal malignancy originating from the neural crest. It's the most common extracranial solid tumor of childhood and a major cause of cancer deaths in children under 5 years of age. Despite multimodal therapy, including high-dose chemotherapy and autologous stem cell transplantation, only below 10% of the patients will survive once they have a relapse of the neuroblastoma. So clearly a medical need to invent novel therapies. And so I joined Mike Jensen's lab in Seattle, and we started to build a CAR and evaluated it preclinically. And when you do develop a CAR, you first have to decide what is actually the target antigen your CAR T cells is supposed to recognize. Then you also have to think about what is actually the spacer length that determines the distance between the tumor cell and the T cell, and we do know that this matters, and I will show this to you in a minute. And then also as a third thing, the intracellular signal for core stimulation. We have heard already about 4-1BB carrying second generation CARs, or for example CD28 carrying second generation CARs. So the target antigen that we chose is called L1CAM, also known as CD171. It's a neuronal cell adhesion molecule, and of course it is expressed in neuroblastoma, but also in several other solid tumors, as for example melanoma or glioblastoma. We looked at tumor biopsies from children with neuroblastoma that had either low, intermediate, or high-risk neuroblastoma, and L1CAM was highly expressed among all of these samples. It's actually a glycosylation of the epitope or of the antigen that our CAR is recognizing, which is making it tumor-specific. Nevertheless, and I think this is the theme of the symposium here, when you want to bring something like this into the clinic, you have to show that it is safe. And since L1CAM is highly conserved among species, so the healthy cells expressing L1CAM in humans also do express the L1CAM in rhesus macaques, and it's the same L1CAM as shown here with the staining. So we used non-human primates and injected them with 10 and 100-fold higher doses of the CAR T cells that we wanted to inject in our patients. And the idea was, if those L1CAM specific CAR T cells are now able to recognize the healthy L1CAM, we would be able to detect this as a toxicity in those monkeys. And then just shown here exemplarily the values for creatinine, a marker for kidney, and you can see that the I call it healthy L1CAM is expressed in kidney. You would have thought to see some increase in the creatinine level as sign of toxicity, and since this was not the case, we decided to move on with this target. Next was the spacer length. So we had 3 different spacer lengths ranging from around 11 amino acids, what we call the short spacer, shown here in red. Then we had a medium spacer shown in green and a long spacer shown in blue. And then we did the typical preclinical testing that you have to do in order to get your preclinical package ready that you need when you want to submit this to the regulatory authorities. And we used the neuroblastoma cell line SKNB2, which is positive for L1CAM, shown here, and we did a so-called chromium release assay. So you incubate your tumor cells with chromium, and then you add your neuroblastoma-specific CAR T cells, and if they are able to recognize and to lyse the tumor cells, the chromium gets released, and you can measure this. We also did cytokine release assays, where we also co-cultured our CAR T cells with the neuroblastoma cell line in vitro, and could see that the CAR T cells were able to release these cytokines as depicted here. You can also appreciate that the blue one, the long spacer one, seemed, at least in vitro, to be the winner. So higher cytolytic lysis, higher release of cytokines. We then went in vivo with our human T cells, because this is what you usually do in a xenograft mouse model. And very surprisingly to us, the long spacer, there was no difference between the long spacer and the negative control, which were just untransduced T cell, but the short spacer seemed to be the winner, which brought us to a point that we actually moved on with the short spacer into the clinic. But it does make you question a little bit the in vitro tests that we use. Next was the co-stimulatory domain, and this is all work already done 10 years ago, so we're still not really known in the field, or maybe it is still not, what is actually the best co-stimulatory domain. Is it 4-1BB, or is it even a third generation CAR harboring, in addition to 4-1BB, also CD28? Again, we did a chromium release assay, comparing those two generations of CAR T cells, we also looked at the cytokine release, and you could think that the third generation is actually better than the second generation, but then again, in vivo, there was no difference, so we actually ended up with a clinical trial design where we had two arms, so the patients either received a second generation L1CAM specific CAR T cell or a third generation CAR T cell product. So we did what you usually do. We tried to answer as good as possible the safety question before we went to the FDA. We also showed that they are effective in vivo and in vitro. We piled all the documents together and submitted it to the FDA. And then the clinical trial started with Julie Park and Naveen Pinto being the PIs of this clinical study, where pediatric patients with neuroblastoma were treated with L1CAM specific CAR T cells, harboring a short spacer, so the red construct, and as said, either 4-1BB alone or 4-1BB and CD28. So the results that the trial showed were that at the end we had 25 patients enrolled and 17 were infused with their CAR T cells. We could show that it was actually feasible to manufacture CAR T cells from those heavily pretreated pediatric patients. We had one patient where the neuroblastoma lesion was relatively easy accessible, so we took a biopsy and could show that the CAR T cells were able to infiltrate in the tumor, and we had transient off-tumor on-target T cell-related toxicity, so we had a rash in 40% of our patients and a hyponatremia in around 12% of our patients, but none of those were actually dose limiting. And interestingly, we saw those in lower doses for the third generation compared to the second generation. So remember, it's a phase 1. It was okay, but it was definitely not as overwhelming as we had seen with the CD19 CAR. So we actually, when I moved then back to Germany, we decided to keep on working with the L1CAM specific CAR. And just two little examples, what we are doing right now in my lab in Berlin is within a very great cooperation with Matthias Feige from Munich. We teamed up. Matthias developed a destabilized IL-12 variant. So as soon as the IL-12 is released, it falls apart because you are not able to give IL-12 to your patient. It would be too toxic. But the hypothesis is that due to the IL-12, we are actually able to activate the innate immunity and could, for example, overcome antigen loss as that was demonstrated in CAR T cell trials before. So when we then compared our CAR T cells harboring now either an IL-12 wild type as kind of a control or the two mutants with the destabilized IL-12, we could show that they are able to release interferon gamma during a co-culture with neuroblastoma cells. We also improved our preclinical in vitro toxicity assay by not just using a one-time co-culture, but we actually added several times tumor cells to our T cells. So we actually challenged our T cells, and then it's maybe a little bit hard to see, but in orange is the normal CAR, and you can see after several rounds of tumor cell challenge, this CAR is not able to eradicate the tumor cells anymore, but then here shown in blue, green, and pink is actually the constructs with the IL-12, and you can see that they do a better job in controlling tumor growth upon repetitive stimulus. What we also saw, which we honestly didn't expect it, was that the amount of central memory cells was higher during the in vitro expansion once the CARs had the IL-12 released. We did not see this for our mutant variants, but when we actually looked at the phenotype after co-culture, then also the mutant ones had a higher percentage of central memory cells, and we think this is quite encouraging. We move further on with this. The mouse experiments are ongoing as we speak, also in terms of safety. We had to establish an immune-competent mouse model, and but as said, the experiments are ongoing. Another one is that we want to look for additional targets next to L1-CAM. So together with colleagues from Berlin, Matthias Selbach and Philipp Mertens, they performed proteomic analysis of primary neuroblastoma samples. Then we actually checked how many of them are expressed in healthy tissue and how many of them not. That was mainly done by Kathleen Anders in my group. And we have actually a specific focus on adrenergic or mesenchymal cells. We have neuroblastoma cell lines that are either mesenchymal or adrenergic, and maybe for the non-neuroblastoma experts in the field, there are proofs coming more and more that these are the two types that neuroblastoma tumor cells can have, and while the adrenergic ones are actually the ones that respond very well to chemotherapy, these are the mesenchymal ones that probably remain and from whom the relapse might occur. So of course it's important that especially the mesenchymal ones do have the antigen that you want to target with your CAR. And if you look at L1-CAM, or as the also already mentioned GD2 antigen, you can appreciate that in the mesenchymal variants, those antigens are not there anymore. So we are now following up with target 4, and I can tell you it's not so easy as it sounds. So we are in the middle of making an antibody. From that we then can take the scFv to make a new CAR, but as said, it's not as easy as it sounds. So developing own CAR constructs, of course, we want to bring them into the clinic. And one thing that is often missing, and I think the colleagues from VHIO are just in the middle of finding a solution for this, are actually the facilities where you can manufacture your CAR T cells. It's a truck, so you have to have a clean room. And I moved back to Germany at the beginning of 2015, and we just got the manufacturing license for our first CAR T product, so it took me 10 years to establish it. And it's actually, I wouldn't say it's easy, but it's totally doable, but it was the room that prevented me from doing it. So we started with one new built clean room that was really just built to make CAR T cells. This is how it looked while it was construction. This is what you can see when you look at it from inside. And we needed this whole thing because we needed one clean room, class B. So this was all built just because for this little room here, where we have now our prodigy placed, but you cannot enter a clean room class B coming from the street. You have to go first in what we call clean room area D, and then from D you have to go to C, and then from C you have to go to B. So all this had to be built in order to be able to make CAR T cells, and we did this with a huge support from Lars Bullinger, who is the head of our oncology department at the Charité. So what are the first CAR T cells that we will manufacture and where we obtained the manufacturing license for? They will be CD19 specific CAR T cells. And now you say, why is she doing that? There are approved products. You are right. But there's for pediatric patients just one single approved product, the one from Novartis. And if this company decides on one day it doesn't make enough money anymore, I would not have access to the CD19 specific CAR T cells to treat patients that I know I could cure with those CAR T cells, but instead I would have to send them home for dying. So I think it's highly relevant, and I think the colleagues also from the hospital Barcelona have nicely shown how important it is to have this in academic hands. Plus, if you have a closer look at the patients treated with Kymriah, then you see if they have the relapse of their leukemia within 180 days after their bone marrow transplantation, then their overall survival totally goes down to 16%. which makes totally sense. I mean, imagine you undergo a bone marrow stem cell transplantation, so a new hematopoietic cell has to be taken out from someone's body, infused in someone new, and while the cell is settling down, there is a leukemia cell growing next to it, then this freshly engrafted cells is taken out again, and then someone tries to make a CAR T cell out of it. I think it's just not the perfect starting material. And in this specific setting, where we had a bone marrow transplantation before, we do have a healthy donor. We do have a healthy donor whose T cells have never seen any kind of chemotherapy before. And this is actually the clinical trial that we will hopefully start next year together with our colleagues from Tübingen, sponsored by Ein Herz für Kinder, and with the support from Miltony. So kids that have a relapse 180 days after they underwent bone marrow transplantation, in that case, we will actually go back to their donor, take his or her totally healthy T cells, make a CAR T cell product out of it, and infuse it in our patients. And while we built this one room, we knew from the beginning this will not be enough. So in parallel, we tried to get enough money to build up a second, a huger clean room facility, really designated also to the production of ATMPs. And hopefully the beginning of next year, it will open. It already looks like this. The outer part is not there yet, but at least the building is already there. So quite confident that this will work. This brings me to the last topic, join for ATMP. When you look at the clinical trials for ATMPs in Europe, then you can see, shown here in yellow, that we are clearly lagging behind compared to our colleagues from Asia Pacific or North America, especially for those early clinical trials. So there's definitely a need to make it easier to enable us to bring our products into these early clinical trials. And as someone who went through this process in the States and now has to undergo it in Germany, I can tell you there is quite a difference. So the EU came up with a call about mapping the hurdles preventing accessibility for patients in Europe to ATMP. And then we decided to apply for this call. And with we, I mean the European University Hospital Alliance, where the 10 or now 11 biggest hospitals in Europe decided to join forces. They're all listed here, and we are happy that VHIO is one of our partners. We teamed up with already existing EU networks like Restore and T2 Evolve. We also asked Fondazione Telethon, Fraunhofer, if they would be interested to team up. And we also asked Eurordis, the patient's biggest representative in Europe. And I can tell you it was a clear benefit that they are on board, because their comments throughout the whole time are really, really valuable. We have a list of supporting partners, which is still growing. So if you are interested, you can just write me an email. I will take you up on this list. We started on the 1st of January 2024. We have a funding for 3 years, so still till the end of 2026, and we got around 3 million euros. What is our mission? We want to come up with an information package where the current hurdles are really listed, what is preventing us from having more phase 1-2 clinical trials for ATMPs in Europe. But we also want to go beyond. We want to come up with a set of recommendations how those hurdles can actually be overcome. And we also want to come up with a sustainable model for long-term consortium operation. So we are in the middle of thinking how a GMP coordination platform within Europe could look like, how the legal entity could be, how the funding could look like, and also an EU ATMP expert platform that, for example, could give advice to scientists developing those ATMPs. So these are our different work packages, and you can appreciate that it's really covering the whole translational pipeline. So we have a work package on preclinical tests, which is led by Antonia Müller, who is here in the audience. We have one about GMP manufacturing, one about clinical translation, led by Silvia Luesma. but also economic schemes, HTA assessment, and stakeholder awareness, which is led by Johan van Eldere, who is also in the audience and General Secretary of VHIO. And then we are also focusing on not only on cancers or on rare cancers, we are also focusing on common diseases and rare non-cancer diseases. Where do we get our information from? What kind of hurdles people are facing? We are actually using real-world use cases. The one with the asterisks were developed within Join for ATMP, and you have heard about Pragmatil before. Also happy to say that we also have a closer look at ARI 0001, which was developed at the Barcelona hospital as one of the examples where we think we can really learn a lot what kind of hurdles were faced within these products, and more important, how the people were able to overcome them. The second part of information that we are getting related to the hurdles experienced from us and our colleagues is coming from a questionnaire, from a survey, and for that we developed 340 questions. But it's not that one person has to go through all the 340 questions. We actually had them specific addressed to developers, payers, regulators, or patient representatives. And depending what you click, where your expertise is, you get your amount of questions, and it said they will be not 340. But the questionnaire is still open till the end of January next year. This is the QR code, so please scan it and participate in our survey. So far, 260 participants have filled out the survey, and I will show you an interim results in a minute. We do lack patient representatives, we do lack industry, so if you belong to these groups, please fill out the survey, which was a huge amount of work, especially for Nara Marella, who is also in the audience, and Franka Klatte. The first preliminary interim results analysis shows that we were able, and I'm sorry it's a little bit small, that from a lot of different countries, mainly from Europe, people filled out the survey. Most of them came from academic institutes or hospitals, as said, only a few from industry and only a few patient representatives. So we hope that more people from those stakeholder groups will fill out the survey. We asked, for example, what kind of ATMP, the people who said they have experience with ATMPs, what kind of ATMP it is. It was mainly gene therapy, and here, of course, the CAR T cells were among the mostly produced, investigated ATMPs, and there were in different stages. They were at the process validation, hospital exemption, or even in pivotal clinical trials, and then of course only few in post-marketing studies. We do have a poster presented by Vania Lobreste, who is also in the audience, really looking a little bit more into the manufacturing part, and we do have a poster from Charlotte van Yesterday at this meeting, who is really looking at the regulatory challenges, at the clinical trial initiation, at all these different parts that you have to face when you want to bring such an ATMP into the clinic. And then just one slide about what we got as a feedback from our survey, that scarily, surprisingly, a lot of people were not familiar with regulatory knowledge or had only a low-level understanding of the regulatory knowledge, despite working in the field of ATMPs and developing them. And I think this was also something that came clearly out this morning. It is necessary that you have regulatory knowledge when you want to bring them into the clinic, and as earlier as you get in contact with people who do have this knowledge, the easier it will be to bring your product into the clinic. And also here, just one thing, the people do have support mainly on the national level, not so many on the European level, which I also think will be an interesting outcome from the survey. So what we have in this interim analysis, we got quite a broad overview. We have really engaged, experienced, and academically entered ATMP community. most of them working in the field of rare diseases, which is not surprising, thinking that they were mainly coming from academia, and there is a need for more streamlined, accessible, and harmonized regulatory guidance and resources across Europe. And with this, I'm at the end of my presentation. I want to thank all the people involved, especially my lab, of course, Mike Jensen, giving me the opportunity to start working with CAR T cells, Lars Bollinger for his support in setting up the manufacturing in Berlin, and then this is a picture from our last year annual meeting from the Joint 418P Consortium, and thanks to you for your attention.
We have time for one question.
Hi, great talk. I have a question regarding your data about IL-12. How much of the response you do see is because of IL-12, and how much is because of the CAR? I mean, CAR here, is it as a delivery vehicle for giving IL-12 in the right place?
So the controls were exactly the same CAR, but just without the IL-12, and then we also had the wild type IL-12, and then we had our mutant differences. And we did see in the in vitro tests that the CARs containing the IL-12 had higher efficacy using the tests we did. So the comparison was to the exact same CAR, just without the IL-12.
You need the CAR. Did you need the CAR, or it could be done, let's say, having a T cell expressing your IL-12?
Yeah. So the tumor specificity from the T cell, that will come through the CAR. So it's always the combination. So the idea is to send in the CAR T cell and then support the efficacy of the CAR by the additional use of the IL-12 at the tumor site.
I was just wondering if you have a T cell, not CAR T cell, but just have IL-12, would that be the same?
Yeah, I mean, there actually were clinical trials trying to administer IL-12 by itself, but they showed a lot of toxicity in those clinical trials. And as said, our idea is really to improve the efficacy of our neuroblastoma-specific CARs, which shown based on the clinical trial data, it doesn't seem to be enough to just send in the CAR. The CAR needs some support.
Thank you very much, and I have a lot of questions I'll ask you in the networking cocktail, the poster session, because now we really need to move on. And with this, I want to introduce the next section. I want to first of all congratulate them for being selected to present their work in this symposium. We're going to have two short talks. The first one by Alessia Buratin, a postdoc at Josep Carreras Leukemia Research Institute. Please join her into the stage.
This is not my...
No, but they will come.
Okay.
Okay. If we could have the slides. There we go.
Okay. Yeah, sure. So, good afternoon, everyone. I would like to thank the organizer for selecting my abstract. Today, I present you our study about anti-BCMA CAR T therapy and our data-driven approach to enhance the response to the therapy. So, the core of the project stands on leveraging complex single-cell multi-omics data to predict which cell phenotypes are more responsive to the therapy and what cell phenotypes are disease-driven by using an explainable machine learning model. So thanks to the previous presentation in this morning, we already know that CAR T therapy have revolutionized the treatment of multiple myeloma patients. But even though initial good response, at the end, almost all patients relapse. So why it fails is one of the biggest questions. In this study, we will harness the single-cell technologies. In particular, we combined the transcriptome with the T-cell receptor from the same cells that can give us a unique window about the heterogeneity, the functional state of the cells, but also the clonal trajectories over time and across the different clinical outcomes. So until now, it is well known that the failure of the CAR-T therapy in multiple myeloma is due to a poor T-cell fitness. That means low proliferation, high exhaustion of T-cells, and these dysfunction signatures can be used as a prediction for the outcome. And this is the core of the project. So understand the mechanisms underlying the failure of the therapy and try to avoid the remission. So to achieve our goals, thanks to a great collaboration with the Hospital Clinic in Barcelona with Dr. Carlos Fernandez de la Rea, we have collected samples from 11 relapsed and refractory multiple myeloma patients that can be divided into two main groups according to the time in which they relapse. We have followed up those patients at two key time points, at the infusion product and after about 1 month, when the CAR-T cell in the blood reached the highest level. At the infusion product, we further separated samples into CAR-positive and endogenous T cells. And from both time points, we have sorted only the T cells, and by using a multimodal approach, we have obtained both T cell receptor and RNA sequencing from the same cells. So we successfully integrated those data and identify 60 different T cell subtypes that are telling us how the therapy is shaping the immune system. In particular, we can observe a clear shift between more proliferative T cells to more effector and cytotoxics between the two time points. By further stratified samples according to the clinical outcomes, we can so identify specific patterns that can characterize the samples between times and time points. No, sorry. Okay. But to better understand the complexity underlying the therapy, we have also analyzed the diversity of the clones by using the Shannon entropy index, that is a measure that takes into account both the abundance and the distribution of the TCR sequences. We can see that the good outcomes, the good patients, have a lower diversity at both time points. This result is in some way telling us that those fewer dominant clones maybe are also those more cytotoxic and persistent over time. So to answer our question, we put together our omics to infer the clone functionality. And it is really interesting to note that the hyperexpanded exhausted T cells at the infusion product for both bad and good responders later transient to more effector and cytotoxic state only for good patients. So all of those results is telling us that we can separate good and bad outcomes already at the infusion product, but can we also predict it? And so here I introduce you the second part of the project that is about the use of machine learning techniques. So using single cell technologies, we can cluster cells according to their markers or clinical information. But in this way, the truly affected cells maybe are hidden. So the idea is to use a classificator model that is able to identify these hidden signatures that can allow us to predict the outcome at an early stage. So what I have done is to systematically train and tune a machine learning classificator model for each T cell subtypes that give us a ranking of the T cell subtypes that are more responsive to the therapy. But at the same time, by using an explainable model, we can also have the importance of each features that in this case are genes. So for each genes, we can have the shape values that can tell us how the genes is more linked to the bad or good outcome probability. So the results are that at first we can obtain a high predictive power from those models, higher than 17%, highlighting at the top of the ranking the endogenous CAR-negative T cells that are that it seems that are the more important and the more relevant for the prediction of the outcome at the early stage. But by moving toward a single cell resolution, we can think to calculate a contribution score of each cell given by the sum of the shape values, so of the genes. And so in this way, we can define those subgroups of cells named resistant to the therapy, those with the highest contribution score. So by looking at the differential gene expression for both endogenous and CAR-positive T cells within these two novel definitions of cells, resistant and protective, we can highlight different enrichment of pathways, but in particular we put our attention on this interferon gamma response that is highly upregulated only on resistant T cells across almost all the different T cell subtypes. This, by looking at the clonotype, it's a high expression only in transient clones. It's in some way telling us that this effect is a kind of a short-term effector push that is also confirmed by this regression model. This regression model where the probability of persistence of the clones over time is strongly reduced as the interferon score is increased. So the next step should be identify alternative and combined therapies that can enhance the T cell fitness and improve the CAR-T expansion and so reduce the time of relapse. And to pursue our dreams, we have used the publicly available data about gene expression signatures of drugs in cancer cell lines. By using, again, a machine learning model, we can obtain this perturbation in silico. And we have validated this model in our data, as we can see by comparing the predicted and observed gene expression before and after the CAR-T therapy. The correlation between the two measures is really good and high across almost all the T cell subtypes. So we suppose that we can also generalize this model to the other drugs present in the public database. And the idea was to apply this drug perturbation in silico to our predefined resistant T cells. In this way, we can obtain a novel phenotype of the new perturbed T cells. And the idea is to compare these phenotypes with the phenotypes from the good responders that can achieve us to ranking the drugs able to, in some way, revert the phenotypes and so reproduce the good phenotype. So in this way, we can obtain a ranking and a selection of the drugs with the highest similarity between the perturbed and the good phenotypes. And in particular, we can highlight this epigenetic regulator, panabinostat, Has been demonstrated that it can modulate the gene expression toward more memory and effector states, reduce the exhaustion. So comparing to our previous results, it should be a good starting point for the drug discovery in this study. So in conclusion, I hope to have convinced you about the importance of using single-cell multiomics and artificial intelligence to predict the patient outcomes at the early stage, but also identify novel and even more tailored therapies that can boost the CAR-T therapy in multiple myeloma. So this is my acknowledgement. I would like to thank all my colleagues from my lab, led by Elisabetta Mareu, all of our collaborators, and all of you for your kind attention.
We have time for one question. So I may ask you, what are your next steps? What are you envisioning? How are you going to continue?
We have started to validate all of our this is a pretty computational study, but now with our collaborators, we are planning to start to validate what we are seeing by in silico in a real world.
Excellent. Thank you very much. Looking forward to hear that results too. And with that, we will continue with our next speaker. So. It's really my great pleasure to introduce the last speaker of the evening. His name is Daniel Lick Christiansen from the Oslo University Hospital. Please join me to join him into the stage. Thank you.
Wow, how intimidating it is to start speaking after so many intelligent and professional people. Hello, everyone, and thank you for giving me the opportunity to speak today. I need to... This is the way? Yeah?
Yes.
I would like to start to introduce my development of next-generation tumor-resilient TCR T cells, and why you should start to introduce GMP thinking early on. So first, I would like to give some introduction of me and why I care about this. I work for the section for cell therapy at the Radiumhospitalet in the capital of Norway, Oslo. And my team, which is headed by Dr. Anne Passetto, provides in Norway a national pre-GMP, GMP infrastructure to guide ATMP projects from research towards clinical application. But although it is a national infrastructure, it can also guide foreign academic projects in the same way. And these are my wonderful colleagues. So it's not easy to make a good product that can be used for humans. Many promising proof of concepts fail to progress, not just because it doesn't work, but because the path to GMP is a long and difficult path to take. We see in my facility that there is often a disconnect between early-stage research and GMP manufacturers, and when researchers aiming to take their wonderful, promising supercar to the clinic have to reinvent to fit into a tight regulatory box, this can cause a loss of sanity for everyone involved. So, I'm sure a lot of you know about GMP, but maybe for the ones that don't know, GMP stands for Good Manufacturing Practice. And it's a set of regulations and guidelines that govern therapeutic products to ensure they are safe and effective for human use. So it's fundamentally a system of control showing that you understand your product, you're able to stay in control of it, and you can document it for everyone to see. So instead of exploring first and trying to adapt it to the clinic later, we asked, how can we embed GMP thinking early without delaying innovation? So T-cell therapy provides the unique ability to target intercellular antigens, as we've gone through today. But efficacy in solid tumors can sometimes be limited by the immunosuppressive tumor microenvironment. And one of our collaborators, Peng, he identified a statistical model using single-cell transcriptomic data to associate certain transcriptomic signatures of T cells in infusion product that were associated with a superior clinical response. And he termed them T-resilient T cells, or TRES, which could correlate with immunosuppressive resistance. One such key signature was the reduced expression of FIB-P gene. And we thought that genetically modifying TCR T cells to mimic this resilient phenotype could enhance therapeutic potential. But how? So we wanted to design the research workflow using GMP-compliant or easily GMP-replaceable reagents, suppliers, and processes to reduce the translational friction later if, of course, the budget or the workload allowed it. And I'll give you some examples. Research often prefers small-scale, budget-friendly, and simple systems in order to maintain the cost and flexibility, while GMP often prefer rigid, closed systems, where kind of cost isn't really the same issue as it used to be. So here is a general research protocol for T-cell workflow. First, you source your cells from buffy coat. You isolate the T cells, the PBMC, we're using gradient separation, and then you activate your cells with plate-bound or magnetic beads before you modify your cells using PCR amplified product on your electroporation device. You then expand your cells in tea flasks, maybe, or culture dishes, using antibiotics to make sure that you have no contamination before you harvest on the centrifuge and you test them functionality with flow cytometry or qPCR. But for GMP, it's a different matter entirely. You source your cells from apheresis product, maybe from patients or from healthy donors. You isolate your cells on closed systems like the Rotea or Sepax, and you activate your cells using GMP-grade reagents, and then you would perform your genetic modification using closed systems with GMP reagents with documented purity and identity. Thereafter, you would expand your cells again in closed systems like the Prodigy or G-Rex without antibiotics, and then you would harvest on a closed system yet again before you would test your cells on a GMP-validated QC system. And all of this within a quality system in GMP-classified cleanroom facilities that have been audited by the authorities. So we chose to use a GMP-light alternative from suppliers we already knew could supply a similar GMP-grade reagent or system. So for cell sourcing and isolation, we used buffy coat and gradient separation. Then we chose to use Transact activation reagent, knowing that they had a GMP-compliant alternative. And then we used research-grade consumables and equipment from MaxSite, and genetically modified our cells using research-grade consumables and equipment, knowing that they had GMP-compliant alternatives. We then used the G-Rex research-grade consumables, knowing yet again that, of course, they had GMP alternatives compliant. Then we separated, then we harvested using a centrifuge before we kind of used our clinical-grade GMP flow cytometer. So to go through some of our experimental design and data. First, we investigated our knockout using a Jurkat cell to identify the best guide. 4 days after gene editing, Jurkat cells electroporated with our second guide showed the best knockout efficiency. And we then moved on to primary T cells, where we sourced our PBMC from healthy donors. We then transduced our T cells with viral transduction with Mark1 TCR before we activated the T cells and then performed the FIB-P knockout using CRISPR technology. We then allowed our T cells to recover for 4 days before we did a functional analysis of our cells. So to look at some of our data, This is our cytokine release panel after specific antigen stimulation. And you can see the negative and positive unspecific controls here as well. Overall, the knockout showed an increased secretion for interferon gamma, IL-2, and TNF-alpha, but on CD8 plus cells, although some effect is still visible for other cytokines and on the CD4 cells as well. Apologies, I'm really nervous. So then looking at our qPCR results, we can see that compared to the wild-type mock cells, our knockout cells exhibit approximately 60% reduced expression of the FibP transcript. So we can reasonably say that the knockout was a success. Our collaborators, Peng, also showed the FibP knockout T cells exhibit reduced cholesterol metabolism which will supposedly inhibit effector T cell function. And we are able to show that FibP knockout does negatively impact the LDL receptor expression. But we don't know the exact mechanism. So for our next steps, we would like to continue expanding our framework, and with a long-term aim of delivering a ready-to-use workflow facilitating TRS-based T-cell therapies with enhanced tumor fitness. So I realized that I've been really very nervous this day, and I hope that you haven't been too bored. But I hope after this presentation, you can at least leave with thinking that GMP is kind of a little bit cool. It's not just boring paperwork. It's a system of control, and GMP is how you prove you're in the driver's seat and have control over your process. And the earlier you build control, the faster and more confidently you can drive your new supercar towards patients. Thank you.
Thank you. Thank you very much, Daniel. It was really a very nice talk, and congratulations. Despite your nerves, you did an amazing job, so congratulations. We've all had our first talk, and we've all been nervous, so yes. It's part of our job, so congratulations. Congratulations on doing it.
So, an entire day without drinking as well.
Yeah, no, that's true. So questions for Daniel? So I would ask you I think it's beautiful from a philosophical perspective, introducing the control earlier. I do wonder, and we've seen the slide previously of the amount of phase 1 clinical trials that are being done in other countries, such as Asia-Pacific region. How do you think GMP systems of control diverse from the different worlds, and should we be incorporating some things but letting go of some other things, or how do we find the equilibrium?
Yeah, so we already heard one example of retroviral transduction being a no-no for the Spanish government, although retrovirus transduction for other authorities can be an okay thing to do. So obviously then one of the most important things that you have to do is identify what kind of GMP regulations are present in your country as of a whole. That is the most important thing, identifying what is there and what can you introduce into your own workflow. And instead of just kind of going off of a general GMP guideline, you can also like introduce a little bit of like this all-encompassing kind of thinking of control. I think that's the most important thing for the research part. That's kind of what I want to convey. I want to convey that look into what kind of things that you can incorporate that kind of increases your control, that is easy to implement.
Thank you very much, Daniel. And with this, we are closing today. It was really a wonderful day and a very tight agenda also for the afternoons. I want to congratulate again all the speakers, and I also want to take the opportunity to thank again Natalia and all the VIO team for organizing the meeting. It has been really an amazing first day. I do think we have now the poster session and networking cocktail, so please join us there, and See you tomorrow, everybody, also.